Universitas Islam Indonesia, Profesor Yandi Shukri, Head of Pharmacy Study Program, Universitas Islam Indonesia, Mr. Saifuddin, PhD, and Head of Pharmacy Study Program, Dr. Farida Hayati. Before starting this event, let us pray together to ask for guidance and blessing from God. According to our religions and beliefs, let us begin our prayer. Amen. Before we move to the main session, let me introduce myself. My name is Hana Trisna Andini, and I am currently studying at the Bachelor of Pharmacy Study Program of the Universitas Islam Indonesia. In this event, I am going to speak to you as a moderator. Dear participants, it is my honor to greet and introduce our judges in our competition today. They are Mrs. Anissa Fitria, Dr. Farida Hayati, Dr. Asih Triastuti, and Mrs. Suci Hanifah, PhD from Universitas Islam Indonesia. And also, there are Professor Patrick Ball and Dr. Hannah Morrissey from Wolverhampton University in United Kingdom. Professor Muhammad Taher from International Islamic University Malaysia, and Mrs. Okti Ratna Mafruhah from China Medical University. Hello, good morning, Mrs. Farida Hayati. How are you today?
Morning. Nice to meet you all.
Nice to meet you too, Miss. Okay, next there is Mrs. Anissa Fitria. How are you feeling today, Miss?
Alhamdulillah, I'm good, Hannah. I hope the same goes for you all.
Okay, thank you for joining us today. Next, there is Mrs. Hannah Morrissey and Mr. Patrick Bell. Good morning.
Hello, good morning.
Hello,
we're looking forward for the lockdown to be over and come in person and meet you and see you all.
Okay, thank you for joining us today. And there are Mrs. Okti from China Medical University. Good morning.
Hello, good morning everyone, and Assalamualaikum Professor and Dr. Hannah. Nice to see you all.
Yes, nice to see you too. Okay, Alhamdulillah, may all of you always be given health because the most important thing. Yes. Okay, may all of you always be given health because the most important thing at this time is none other than health. Okay, there are some informations that I will announce and must be complied by participant during the presentation season. Okay, thank you. The first one, all participants and attendees are required to register on the form that has been distributed by the committee in the chat column. Second, participants who will make presentations are those who enter into the top 10, and the order of presentations is determined by the committee. Third, in the next session, we will present a video presentations of each participant. Are kindly requested to attendees who are not interested to talk can be mute the microphone. Last, after the video, there will be a Q&A session. I invite the audience to ask questions to the presenters via chat in this room. Questions can be submitted since the video is playing. Q&A session will be held for 3 minutes. Okay, without wasting any more time, in the first order, in this oral presentation, with number OP1, there is Zahratun Nafi'ah. Let's take a look at the video. Okay, hello community. Is there any problem to start the video? Okay, let's wait a little bit more.
First thing first, let me introduce myself. My name is Zahra Nafia. Warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. First thing first, let me introduce myself. My name is Zahra Nafia. I'm from Universitas Islam Indonesia, and in this occasion, I would like to present the study result that I have done with my colleagues, Mrs. Vitarani Dwi Anandanirum and Mrs. Rohmi Istihara, with the title of allele frequency distribution of UGT1A6 or S6759892 T into G valproic acid metabolic enzyme encoding gene among healthy Japanese population in Indonesia. Epilepsy is one of the most common chronic neurological disorder affecting approximately 50 million people worldwide. This neurological disease is characterized by unexpected and periodic seizure incidents. In developing countries such as Indonesia, the prevalence of epilepsy is higher compared to the developed countries. To date, anti-epileptic drugs remain a popular choice of therapy. Valproic acid or VPA is one of the most widely used anti-epileptic drugs to control the various types of seizures, including myoclonic, absent, general, and partial seizures in both adult and pediatric patients, since they have the lowest adverse effect with a broad therapeutic index. However, inter-individual variability in its dose and plasma concentration still occurs. It is acknowledged that one of the vectors influencing the variability is the genetic polymorphism in the metabolic enzyme encoding gene that has been proven to correlate with its pharmacodynamic and pharmacokinetic variability. An understanding of the mechanism of VPA metabolism can explain the influence of genetic polymorphism on the changes in plasma concentration and their clearance with an implication for the effective dose. VPA metabolism has three main routes, and one of them is phase 2 metabolism through the formation of glucuronate conjugates by glucuronidase, an enzyme encoded by UGT1A6 gene, one of the most commonly found enzyme compared to the other UGT1A isoforms. This pathway becomes the major route of valproic acid metabolism, accounting for 30 to 50% dose. This study aimed to determine the allele frequency distribution of UGT1A6 or S6759892 T into G in Japanese population as the largest ethnic group in Indonesia. The study used stored biological samples of DNA isolated from 100 healthy respondents, and the respondents were categorized as originated in Japanese ethnic group determined by their three previous generations. This study has passed the ethical review from the ethics committee of the Medicine Faculty of Universitas Islam Indonesia. Genotype analysis of UGT1A6 or S6759892 T into G was performed using the polymerase chain reaction or PCR method with this forward primer and this reverse primer prior to the process of restriction length fragment polymorphism using HHA1 as the restriction enzyme to cut the amplification product. Allele frequencies were calculated using the Hardy-Weinberg equation as in the previous study with the following formula. And this is the materials that we used in our research. The electrophoretic display of digestion by the enzyme that detected the target polymorphism in the study is presented in this figure. The amplification product from the PCR method were then identified and followed by restriction digestion using HHA1 enzyme. The electrophoretic display of digestion by the enzyme that detected the target polymorphism in the study is presented in this figure. The amplification products from PCR method were then identified and followed by restriction digestion using HHA1 enzyme. HHA1 enzyme cut the sequence of GCGC into 233 and 72 base pair DNA templates. GG genotype was recognized and digested by HHA1 enzyme. The digestion of amplification product resulted in 305 base pair fragments of TT genotype or wild type and 233 base pair and 72 base pair fragments of GG genotype or mutant with 305 base pair, 233 base pair, and 72 base pair for heterozygotes or TG genotype. Univariate data analysis was then used to determine the allele frequencies of UGT1A6 or S6759892 T into G gene. From this table, it is known that from 100 healthy Japanese Indonesian respondents involved in the genotyping of UGT1A6 T into G, the frequencies of TL and GL were 0.86 and 0.14 respectively. The frequency of GL in Javanese Indonesian ethnic population is similar to the one in a study involving Chinese, Caucasian, and African populations. Several studies have been carried out to analyze the influence of genetic variation of UGT1A6 gene on inter-individual pharmacokinetics of VBA therapy. A study involving 98 pediatric patients with epilepsy who received VBA showed that polymorphism in UGT1A6 affected its biotransformation, leading to lower plasma concentration of VBA and implied the need for dose increase. The frequency of GL of UGT1A6 or S6759892 among Javanese population in Indonesia was 0.14 or 4%. Therefore, this research recommends further studies regarding the influence of this polymorphism on the pharmacokinetic profile of VBA, as well as the clinical response to valproic acid among Indonesian patients with epilepsy. Further studies related to its influence of the risk of breast cancer, the most common type of cancer among Indonesian women, are also suggested to encourage earlier healthy promotion and prevention programs. The researchers are grateful to the Department of Pharmacy of Universitas Islam Indonesia for the research funding. And this is the references that we used in our research. That's all my presentation. Thanks for your attention and wassalamualaikum warahmatullahi wabarakatuh.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Well, that's the presentation from participant under the number OP1. Okay, please for audience who want to ask can send the question in the chat column, including the origin of the institution, or you can use the raise hand feature in this Zoom room. Okay, there's already a question from Gihat Antar from University of Bakti Kencana. The question is, what future action could be done from this research alone after knowing the effect of UGT1A6 gene to this specific disease? Okay, to Zahrotun Nafiah, please answer the question.
Okay, thank you for your great question for me. Let me answer your question. So the effect of UGT1A6 gene is implied for the decrease of concentration of VPA and implied to the increase of VPA dose. So from this research, we can see the general figure of the allele frequency, especially in the Japanese population, for the basis of the medication, individualized medication. So it is the basis, or this research is basis to the further research, or need the further research, or for the to know the effect of UGT1A6 for the population, the larger population. So it is known that this research just to give the general figure of the allele frequency, especially in Japanese population, to the basis of the individualized medication, and need the further research to know the effect for about to for the effect of this research to the larger population. That's my answer.
Okay, thank you Zeroto Navia for the answer. And for Gihan Antar, I hope the answer can be understood with very well. Once again, thank you I said to Zeroto Navia as the first presenters. Okay. Next, I would like to say thank you once again to our judges who already attending this event today. There are Mrs. Anissa Fitria, Dr. Farida Hayati, Dr. Asih Triastuti, Mrs. Suci Hanifa, Professor Patrick Ball, Dr. Hana Morrissey, Professor Muhammad Taher, and Mrs. Okti Ratna Mafruha. Okay, next we will move on to the next participant with number OP7 from Universitas Bhakti Kencana, Jihad Antar Morsi. Let's take a look at the video.
As a pharmacy student who loves to phone.
Malah loading.
As a pharmacy student who loves phone all-nighter, and well, I'm not very proud of that, but along with the many other students that I personally know. We are very dependent on this particular stimulant drug that can improve, give us a higher energy and just help us focus in general, which is why the topic of this research is actually something I'm very passionate about. Well, I am Gihat Antar, and today I will be presenting to you about caffeine extraction and characterization of coffee beans and tea leaves. Full credits to the authors of this research. It is truly privileged to know more and to study deeper about the process behind extraction and characterization, especially of caffeine. Caffeine itself is an alkaloid compound that acts as a stimulant in humans. It is a central nervous stimulant having the effects of temporarily warding off drowsiness and restoring alertness, which I find very crucial, especially for college students like us. You know that every time we drink tea, coffee, cocoa, chocolate, or cola, we are giving our body a hit of caffeine. Did you also know that along with nicotine and alcohol, caffeine is actually considered one of the most widely used mood affecting drugs in the world? Crazy, isn't it? The objectives for this research is to extract caffeine from tea leaves and coffee beans by liquid to liquid extraction method. And for the characterization, it is to characterize the obtained caffeine with the melting point, ER spectroscopy, and differential scanning colorimetry. And lastly, my personal favorite is to develop an easily adaptable method for qualitative and purity analysis of caffeine. As for the materials and the method of the specific research, of course, they are using samples and extracting agents and solvents, such as for the sample, they're using tea leaves, coffee beans, etc. As for the method, we discussed before that they are doing it based on various physical methods for the characterization, such as melting points, ER spectrum, differential scanning colorimeter, and purity check by HPLC. From the research, it was observed that using chloroform as its extracting solvent has actually higher efficiency than other solvents. You can see in table 2, it shows the extraction efficiency of pure caffeine from tea and coffee. It also shows that coffee beans has actually contain a higher percentage of crude caffeine as compared as to tea. It is higher by 1.6%. To purify the crude caffeine further, similar procedures were utilized. However, caffeine content of the various sources varies also with soil conditions and climate. You can see from table 3 to 5, it shows the characteristic of caffeine in various parameter for both sample and standard respectively. While table 2 to 5, it shows the result of caffeine samples measured, and it was found to melt at 238 Celsius. The infrared spectrum of the isolated caffeine showed the absorption band similar to that given in literature. The spectrum indicates that it is the absolute purity of the purified caffeine. Amazing. What amazes me more is that they have developed a high performance liquid chromatography method for the determination of caffeine. So they will be using that instead of spectroscopy is accessible. And they chose this method because it is said that HPLC is the most widely used qualitative and quantitative determination and separation method. That this method is also popular because it is non-destructive, and unlike gas chromatography, it may be applied to the thermally liable compounds. Moreover, it is also a very sensitive technique as incorporates a wide range of detection methods. So by using this method, they can determine the retention time and the relative peak area of the extracted purified caffeine, as you can see from the figures above. So to conclude this research, we can say that they have found and developed a method for extraction and purification of caffeine from tea and coffee. Caffeine from tea and coffee is first extracted by liquid to liquid extraction, followed by recrystallization. Then after that, the purified caffeine is then analyzed by HPLC method. So the effective characterization of caffeine was achieved by determining the ER spectrum, employing a melting point apparatus and a differential scanning colorimetry. A special thank you for the authors for this astonishing and helpful research done by them, along with the help of they received from the scholars whose articles are cited and included in the reference of this manuscript. I'm also very grateful for SlidesGo for the template of this PowerPoint. I want to end this presentation with a quote I fell in love with from Robert Haynes, that everything is theoretically impossible until it is done. Thank you.
Okay, thank you. There is the presentation from participant under the number OP7. For the participant who want to ask, can send the question in the chat column, including the origin of the institution. Okay, there is one question in the chat column from Indonesia, from Universitas Islam Indonesia. The question is, what is the novelty of your research compared to the other extraction research? Okay, please answer the question.
Thank you so much for the amazing question. I'd be more it'd be a pleasure for me to answer this, because first, the compound used in the extraction on my research is caffeine, and I discussed before the importance of caffeine, especially these days where became a necessity for people, especially college students like us and working people. And the other novelty that is that from this research alone, they have developed a determination method, which is other than determination using by spectroscopy UV visible, they're using HPLC, which is a much faster and sensitive technique, and yeah, which is I find very crucial, especially for extraction in general. Thank you so much for the question. That's all.
Okay, that's the answer from Nihat Antar. I hope it can be understood with well. Thank you, Inesha, for the question. Okay, next let us move on the next participant. Okay, the next participant is Zulfa Nur Afifah with number OP13 from Universitas Islam Indonesia. Let's look at the video.
People around the world willing to extract wealth for youth. There has been a surge in demand for high-end anti-aging and brightening cosmetic products. As we know, vitamin C remains a star ingredient in skincare across the globe, which is water-soluble antioxidant that easily oxidized. On all of these issues, then let me here to offering one of greatest solution to handle this problem. But first, let me introduce myself. I am Zulfa Nur Afifah from Department of Pharmacy, Universitas Islam Indonesia. And here's our research. entitled Comparison Downsizing Methods, Extrusion and Ultrasonication of 3-O-ethyl ascorbic acid nanoliposome. 3-O-ethyl ascorbic acid nanoliposome is one of the ascorbic acid derivatives which has better stability to oxygen and light than other ascorbic acid derivatives. This is due to the addition of ethyl group that can protect the 3OH group from ionization and oxidation. For its whitening effect on skin, this active substance works around stratum basal, the part of epidermis. Therefore, this active substance requires a delivery system that has good compatibility with amphiphilic molecular parts that can handle its hydrophilic properties. So, the liposome delivery system is the solution. Liposome is a colloid drug delivery system that formed based on hydrophilic-hydrophobic interactions between phospholipids and water molecules. Ladies and gentlemen, and the honorable judges, the purpose of this study was to carry out the comparison, downsizing methods, both extrusion and ultrasonication, that affected the characterization and stability of 3-O-ethyl ascorbic acid in nanoliposome dosage form. So, these are the materials that we used during this study. They are rotary evaporator, vortex, particle size analyzer, ultrasonic mini extruder for the tools, and we also have lipoid S75, lipoid DMPGNA, cholesterol, 3-O-ethyl ascorbic acid, lactose monohydrate, methanol, chloroform, and also PBS. The liposome preparation was conducted in several repetitions of the extrusion cycles. They are 1, 3, 5, and 7 cycles. The extrusion process is counted as a half cycle once the liposome mold suspension solution passes through two different polycarbonate filter membranes. Meanwhile, the ultrasonic waves provide additional energy, which causes changes in lipid regulation. The variations are 4, 6, 8 and 12 minutes. Your Excellencies and the Honorable Judges, now I would like to explain the results of this study, starting with organoleptic appearance. This figure 1 showed that the more number of extrusion cycles and duration of ultrasonication in time, the clearer the suspension solution is. The characteristic of the formulas produced based on physical parameters can be seen in Table 1. And now, for the particle size distribution, we can see that the narrower the peak, the homogeneous nanosuspension produced. And for particle size versus zeta potential, we could also see the good relationship between particle size and zeta potential values or 5 and 7 cycles for extrusion, also 4 and 6 minutes for ultrasonication. And for the stability test, in temperature of 2 until 8 degrees centigrade for 1 month is quite stable because they still meet the particle size and polydispersity requirement index. Besides, almost all of the variations are still homogeneous. Ladies and gentlemen, and the honorable judges, nanoparticles contain an encapsulation layer covering 3-O-ethyl ascorbic acid and a nearly spherical shape, as shown in Figure 7. Although some particles seems leakage, this is because the TAEM examination was carried out after 2 months from the nanoliposome was met. Based on the results obtained, it can be concluded that the preparation of nanoliposome 3-O-ethyl ascorbic acid using extruder and ultrasonication is optimal
in 5 and 7 extrusion cycles, and duration of 4 and 6 minutes for sonication. Also, extrusion is considered as superior downsizing methods. Our thanks are due to Department of Pharmacy and the Nanopharmacy Research Center of the Islamic University of Indonesia, or known as Universitas Islam Indonesia, which already supports the facilities and infrastructure for this research. Ladies and gentlemen, these are the topics reference that we use.
Excellencies and the honorable judges, further research is needed to analyze the appropriate components for combining 3-O-ethyl ascorbic acid nanoliposomes, and this will become such a great opportunity that fulfill the future demand of beauty. That's all from me. I rest my case. Thank you very much for your kind attention.
Okay, well, that's the presentation from participant under the number OP13. It's very well presented. And there is question in the chat column from Jihad Antar from Universitas of Bakti Kencana. The question is, what is the use of cholesterol for the solution and how will the absences of the substance affect the solution? Please answer the question.
All right, thank you very much for your great question, Ms. Jihad. Well, actually, to form liposome, we need couple of lipid ingredients, and in this case, we use three things. They are lipid S75, lipid DMPGNA, and also cholesterol. And why we do need to use these three things is to make it more rigid, because liposome will be more create with the addition of several lipid ingredients. And also, how will the absence of that substance affect the solution? Actually, it's we're not talking about the solution, but we are talking about the delivery system that is in a liposome that we know it contains phospholipids. So the cholesterol, as I said before, just like another lipid ingredients, it will make it more rigid. So the encapsulation will be more great. That's it. Thank you very much for your great question. Assalamualaikum warahmatullahi wabarakatuh.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Okay, that's the answer from Zulfa Nur Afifah. I hope it can be understood well. Thank you, Jihad, for the question again. And now let's move to the next participant with number OP16 from Universitas Surabaya with name Hansen Ngaliman. Let's look at the video.
Good day to the panel of judges and viewers. Wishing you a good day and good health in these troubling times. Before I proceed, let me introduce myself. I am Hansen Ngaliman. I came here with my research title, HOLACOVAC, Monitoring the Efficacy and the Safety of COVID-19 Vaccine through the Mobile Application. Let's get started. Let's see how beautiful our Earth is. But when the SARS-CoV-2 virus came to attack our Earth, it became very, very sick. The SARS-CoV-2 virus itself infects our lungs or respiratory tract, causing a coronavirus disease, also known as COVID-19. The SARS-CoV-2 virus is transmitted through when someone infected talks, sing, sneeze, or cough. So you need to cover up your mouth with the mask before you talk, sing, sneeze, or cough. Our country has done a 3M and 3T to prevent the SARS-CoV-2 virus. 3T stands for testing, tracing, and treatment. And 3M stands for washing your hand, keeping the distance, and wearing your mask. But now it is added one V. V stands for the vaccine. And after the vaccination process in our country, hopefully it will form a herd immunity. So the contagion cannot really pass via immunized to susceptible ones. For your information, our country has imported a large batch of vaccine from China, and it's called the Sinovac vaccine. It shows 65.3% of efficacy and is proven halal. Our Indonesian Health Ministry stated that there will be no body reaction due to the vaccination, or if it occurs, it might only just be a mild reaction of our body, such as the fever, headache, muscle pain, and also body weakness. But based on evidence, those who have been given the vaccine, they say they will feel more likely to feel sleepy and hungry. And the vaccination process in Indonesia is divided into 4 stages. And we have done the first stage. Congratulations, Indonesia. And as an evidence, our president, Mr. Joko Widodo, has been given the vaccine. And I also have done the survey on 50 medical workers about making this app. So most of them showed me the very positive feedback. So that is the kickstart for me to make the app. Hopefully it will help the vaccination process in Indonesia. The objective of this study is to find the right design for Holocaust in order to maximize the COVID-19 vaccination in Indonesia. To make the Holocaust application, I use the Just in Mind application. And the first method I'm using is the distribution of the questionnaire. The second is the implementation of application. The third is socialization of application. The fourth is inputting the vaccine database. The fifth is selecting the application features. The sixth is making the application draft on mobile. And last but not least, we are keeping it to the public. This is the research and allow me to demonstrate you. So this is the first page. You will need to choose the language. We are choosing English and you need to sign in with your name and also NIK and press login. It will bring you to the first page. This is the page when you haven't been given the vaccine. So it will remind you the date, the time, the location, and the doctor that will give you the vaccine. If you have given the first shot, you would need to, you can set a reminder for you for your second shot. And also, if you have finished the first and also the second shot of the vaccine, you will need to fill the questionnaire right here. And the questionnaire will come out every day, and also there will be few questions that you will need to answer, such as this one. Do you have fever after vaccinated? Press yes if you have fever. Press no if you don't have fever. And also you need to fill the profile here, and also you will fill your medical record in health data. And also you can contact the pharmacist, hospital near you, and also Satgas COVID due to the vaccination. As a conclusion, based on the data stated, Holocaust is a mobile application that can be utilized to monitor the efficacy and the safety of COVID-19 vaccine exclusively in Indonesia. This application is fairly easy to operate and to install by the users, and also the application includes the very complete features such as the reminder that will notify you the time, the location, and also the doctor or the workers that on duty will give you the vaccination. And also, it is completed with a feature where you also can contact the licensed pharmacist due to the condition after being vaccinated. And also, you can find the nearby hospital. If you feel the side effects of the vaccine, you can contact the nearby hospital based on the current location. This is the references that I use to make this research. And also, I want to say that I've been working really hard on this research only for 2 weeks. And different hurdles have been crossed to achieve this research. And I extend my heartiest thanks to my mentor, Dr. Mariska Evelina Gondokusumo, and also my another mentor, Penny Kusuma Leliga, for their roles in supporting me that this research is made to success. Last but not least, I want to thank my parents, friends who were backbone to this research work. Thank you for your attention, and let's fight this pandemic together. See you and bye-bye.
Okay, thank you, Hansen Ngaliman, for the great presentation. The topic taken is very interesting and it is very presented with very well. Okay, for the participant, those who want to ask can drop the question in the chat column and including the origin of the institution. Is there any question from participants? Okay, there are a question from our judges, Mrs. Oktiratna. The question is, since the application looks very useful, how will you disseminate the application for the general population? Thank you. Okay, please to Hansen can answer the question.
Okay, first of all, good morning, and also thank you for your question. So I'm gonna answer it. It's my pleasure. To distribute the app, thanks for saying this app is very useful. So to distribute the app, so we are going to first socialize the app to the medical workers, such as the pharmacist, the doctor, the nurse, and another medical workers. So after the socialization, hopefully the medical workers that have been socialized will help to distribute the app too. And hopefully, the app is useful for the entire Indonesia because the app is not only um, very attractive, but also it helps tracking the vaccination process in Indonesia. Because if you ask me if there is another app similar to my app, there is, but it's not specific for the COVID-19 vaccine. So thank you. That's my answer.
Okay, thank you, Hansen, for the answer. And thank you, Mrs. Okteratna, for the question. I hope the answer can be understood well. Now let's move to the next participant with number OP21 from Universitas Pertahanan with name Kurnia Sandi. Let's take a look at the video.
Assalamualaikum warahmatullahi wabarakatuh. Here I will make a presentation about application of Pharma as safe alternative blood sampling in COVID-19 pandemic. First, I want to tell about myself. My name is Kurnia Sandi. I'm from Military Pharmacy Faculty, Indonesia Defense University. The methods I use to make this oral presentation is narrative review of 4 journal. Here the journal I use as my reference to make this oral presentation. First, I will tell you about the COVID spread and cases of COVID-19 in Indonesia. The people that have infected by COVID-19 in Indonesia is more than 1 million people. So it's a big number that we must pay attention and make innovative technology to do our activity even though it's increasing. The symptom of COVID, like you can watch here, like cold, shortness of breath, difficulty of breathing, fever or chills, muscle and badiasis, vomiting or diarrhea, new loss of taste and smells. FAMS or volumetric absorptive microsampling is a recent microsampling technique used to generate blood specimens of blood and other biological matters for application to a plethora of bioanalytical purpose. Here's the picture of FAMS. I will tell you about advantages and disadvantages of FAMS. First, draw technique, because it just take a very little sample from your blood, so it will not invasive. Second, accurate volume sample, because FAMS is just take 10 until 20 microliter of your blood. Third, easy self collection of clinic, so you can do microsampling without medical techniques. Fourth, no cold chain shipping required. 5. Specimen stability and storage at room temperature, so you don't need a cool box to save the sample. Last, compatible with standard liquid handling instrumentation. What is the disadvantage of FAMS? First, relatively expensive sampling technique, because the FAMS tools is only can be used just one, so you cannot do it with the same tools. That's why I say relatively expensive for sampling tools. This is the tutorial of FAMS. That you must take the blood with the tips of FAMS, then drying it, and extraction the micro sampling of blood. Why we need FAMS? Our research about drug, because we face the obstacle during the pandemic is social distancing that inhibit the drug development. So difficult to implement a clinical trial. So we need a new innovative technology that is FAMS as a alternative blood sampling technique. Here, the clinical consideration for implementation of FAMS. First, invasive nature is minimal when the sampling and the volume of. Second, an attractive approach for vulnerable population. Third, advantages for clinical trial conducted in remote areas with limited access. Fourth, it does not require anticoagulant and centrifugation device. So you not when you take blood sampling from vena, it don't need any anticoagulant or centrifugation device. 5. The FAMS approach can also save on the cost and storing and of sending sample, because you don't need a cool box. You can send it with a post without need a special treatment. 6. FAMS can be applied immediately for the sampling of volunteer of cancer patient at home. The bioanalytical consideration for implementation of FAMS. The first is fixed volume uptake, because the blood you take with FAMS is have a fixed volume when take it. Second, stability of analyte and metabolite. Third, homogeneity of samples and hematocrit effect. When you use FAMS, it will minimize the hematocrit effect than when you use other sampling tools like DBS. Fourth, the carryover and cross contamination. This is the factor to be considered use of FAMS during COVID-19 pandemic. First, self-sampling without medical staff or
Ular. Kecil.
No medical staff or expert when you take the microsampling, but it may be a variability data that takes. Second, volunteer patients' compliance in taking samples. Third, the discipline of volunteers or patients in sampling with FAMS at home without direct control remains a significant concern. When you have volunteers with bad discipline attitude, it will make your clinical trial biased. So the conclusion is, the use of FAMS as opportunity sampling permits the gathering of samples in places some distance from the relevant health center and may be finished through the affected person at home. This may be carry out the implementation scientific trials on drug development and TDM or through the COVID-19 pandemic. In the factor of bioanalysis, FAMS has gained of minimally invasive remedy and small sampling quantity as compared to sampling methods. However, regardless of its advantages, FAMS has worried approximately the stableness of analyte that aren't found in dry samples. So special interest is needed of stableness of those analytes in FAMS samples. So that's all my presentation. I'm sorry for my speaking and my materi. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Kurnia Sandi, for the great presentations. The topics about funds is very interesting. Yeah. Okay, now we will see in the column chat For participants who want to ask, can drop the question now. Okay, well, it's look like there are no questions from the participants. I invite the judges to ask questions to the presenters via chat in this room, please. Okay, looks like there is question from our other participants, Zulfanur Afifah. The question is, what is the concrete action to handle disobedient volunteer as it will make the research PS from Universitas Islam Indonesia? Please to Kurnia Sandi answer the question.
Okay, thank you for the question from Zulfanur Afifah. I will answer the question. What is the concentrate action to handle disobedient volunteer as it will make the research bias? So we can do, as the researchers, we can do the things like we help the volunteers to explain the tutorial, and we have data from the volunteer and make sure that the volunteer is can we trust the volunteer, and we must have the contact of the volunteer so we can call it every we want to do, or when the volunteer is don't know how to collect the sample with the blood sampling. I think that's what we can do for the action to handle disobedient volunteer. Thank you.
Okay, thank you Kurnia Sandi for the answer. And for Zulfanur Afifah, I hope the answer can be understood with well. Okay, next we will move to the next participant with number OP22 from Universitas Ahmad Dahlan with name Jayanti Evita Saskari. Please take a look at the video.
Hi everyone, I am Jayanti Vita Saskari from Ahmad Dahlan University. Here's my study about formulation of cetirizine hydrochloride fast disintegrating tablets by direct compression using cross-povidone and cross-caramello-sodium as a super disintegrants. Fast disintegrating tablet or FDT is a tablet that dissolve or disintegrate in the oral cavity without the need of the water or chewing. FDT disintegrate instantly to release the drug which dissolve or disperse in saliva. In this study, we use cross-povidone and cross-caramello-sodium as a super disintegrants because those use a combination of wicking and swelling that will disintegration rapidly in water. For the optimization method, we used the simplex lattice design to determine the concentration of cross-povidone and cross-caramello-sodium. Preparation of cetirizine HCl FDT tablet, we use the direct compression method. On the table 1, there is a formula of cetirizine HCl FDT tablet where the cetirizine HCl as a active ingredients, cross-povidone and cross-caramello-sodium as a super disintegrant, mannitol as a sweetener, PAG 4000 as a lubricant, and Avicel as a filler. And on the table 2, there is a formula of concentration cross-povidone and cross-caramello-sodium. The first test is the hardness test, where the hardness of tablet about 3 until 5 kg per centimeter cubic, and the second is friability test, where the required fragility limit is not more than 1%. And the third test is disintegration time. Disintegration time: use a petri dish filled with 20 ml of distilled water where the requirement for FDT disintegration is not more than 3 minutes. And the fourth waiting time, we use a filter paper and the petri dish where the filter paper that has been folded twice into a 5 diameter petri dish, and then the petri dish was filled with 5 ml of distilled water containing strawberry red powder, and then tablet is carefully placed on the surface of the filter paper. The time required for develop red color on the upper surface of the tablet is noted as the waiting time. Result hardness test of combination cross-povidone and cross-caramello-sodium is -2. That means the combination cross-povidone and cross-caramello-sodium can decrease hardness of the tablet, but on the table we can see that the hardness of the tablet still on the range. The second result of reliability test of cross-povidone and cross-caramello-sodium is 0.82, which means this combination cross-povidone and cross-caramello-sodium can increase the variability tablet, but no significant effect. The third result waiting time of combination cross-povidone and cross-caramello-sodium is -3.85, which means combination cross-povidone and cross-caramello-sodium can decrease waiting time of the tablet, so the tablet can disintegration rapidly. The fourth result disintegration test of cross-povidone and cross-caramello-sodium is 111.91, which means combination cross-povidone and cross-caramello-sodium can decrease of disintegration time of the tablet. Disintegration time and waiting time caused by morphology of cross-povidone and cross-sodium, where the cross-povidone particles are tend to be granular and highly porous, which facilitates wicking of liquid into the tablet. For the cross-caramello-sodium, because the fibrous structure of cross-caramello particle allows intra and extra-particulate wicking, result in rapid disintegration. Result of hardness test, friability test, waiting time, and disintegration time put on the simplex lattice design with the goal for the hardness is in range, friability in the minimum, waiting time in the minimum, and disintegration time in the minimum. Simple lattice design will predict formula optimum of SETI region HCl. The optimum formula for SLD result in cross-povidone and cross-caramello-sodium with comparison 3.35% and 2.65% in every 100 mg per tablet had 3.12 kg of hardness response, 0.60% of reliability, 1.32 seconds of waiting time, and 2.23 seconds of disintegration time. One sample t-test analysis showed that there is no significant difference between the predicted parameter values and the verification result. Here's the video of disintegration time test.
Okay, thank you, Jayanti, for the excellent presentation about formulation with combination super disintegrant. Next, we will move to the Q&A session. For participants who want to ask, can drop the question in the column chat. And for participants and attendees that are not filled the register form yet, can fill the register form now. Okay. There's question from Jihad Antar from Universitas of Bakti Kencana. The question is, What is the overall objective of the research? How can the society benefit from the product? And who are the target market of this product? Please, Jayanti can answer this question.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for your question. For the purpose of this study, because we want to get the fast disintegration tablet or the fast tablet with absorb in oral cavity, and how can the society benefit from the product? Not most of people, but some people can get hurt when swallow the conventional tablet, or pediatric or geriatric patient that hard to swallow the conventional tablet. So the target of this is for the people who hard swallow the tablet, and then we formulation the tablet for easily to consume, and people can get this product on the market when that's how can the society benefit from the product, and how to get this product. We can get this in the market if there is a company to produce this formulation. That's all. Thank you.
Thank you, Jayanti, for the answer. And for Jihad, I hope the answer can be understood with well. Once again, I need to remind you, all participants and attendees are required to register on the form that has been distributed by the committee in the chat column. And then for participants and attendees who are not interested to talk, can be mute the microphone. Thank you. Okay, next, let's move on the next participant with number OP24 from Universitas Ahmad Dahlan with name Ayu Marliana Agustin. Please take a look at the video.
Assalamualaikum warahmatullahi wabarakatuh. I'd like to thank the Almighty God because we are here today still feeling healthy and able to participate in this oral presentation. Secondly, I would like to thank the Committee of International Pharma Competition IPCT 2021 for giving me this opportunity to compete in this contest. I am Ayu from the Faculty of Pharmacy at Ahmad Dahlan University. In this opportunity, I would like to present our research about qualitative study, knowledge of the effect of MDR-TB treatment and adherence to medications in MDR-TB patients. Tuberculosis. I don't like to admit it, but it's a deadly infectious disease caused by Aerobacteria microbacterium tuberculosis, which is one of the 10 deadly diseases that cause death. TB occurred in every part of the world. In 2019, the largest number of new TB cases occurred in Southeast Asian regions with 44% cases, followed by Africa region with 25% and Western Pacific with 18% cases. In 2019, Indonesia is the second highest number of TB in the world, with an estimated prevalence of 1.6 million cases. I'm going to be talking about multi-drug resistant tuberculosis, or TB-MDR. TB-MDR is a condition when the Mycobacterium tuberculosis bacteria is immune into anti-tuberculosis drugs. It is against at least two of the most potent anti-TB drugs, namely isoniazid and rifampin. Indonesia is one of 27 countries with a relatively high prevalence of MDR-TB, with a total of 490,000 cases in 2016. The number of TB cases spread throughout the island of Indonesia is dominated by Papua as the highest cases, but has a low success rate. The resistance that occurred in Jayapura, Papua was 11.1% in primary cases, 27.5% current cases, and 4% MDR-TB cases. How did the TB become resistant? It could happen if the drug administration is inappropriate or the patient doesn't complete the treatment. Patient consistency during TB treatment was affected by factors such as their knowledge of the disease, satisfaction with health service, and social support such as family. Our purpose in doing this research is to know about the perspective of patients on their treatment and the things that underlie medication adherence of MDR-TB patients. The method that we use is observation, patient interview, confirmations, and team analysis. To protect the validity of the information obtained from the patient direct observation therapy, we reconfirm with patient family and the patient about the result of the interview. The interview was recorded and will be written if the patient doesn't want to be recorded. During the interview process, the steps prepared to prevent TB transmission were using a semi-structural qualitative approach, which was conducted under the sun and using N95 mask. The research was held at Doc 2 Hospital Jayapura with 4 female patients with MDR-TB as the subject. There are several questions we give patients during the interview. First, the patient knowledge of MDR-TB. 3 out of 4 patients know about the disease, with 50% state the results are positive even they have been taking 6-month medication, and the remaining 25% state they are unaware because they stopped using the drugs, and the other state lung coughing pain. Second, patient knowledge about MDR-TB treatment. 3 out of 4 patients know about this disease, and 75% state requires a long treatment, has side effects but cures, and 25% state that they are unaware about it. Third, we asked what they felt about MDR-TB treatment, and all the patients answered symptoms of coughing disappear but side effects appear. To find out more details about the side effect that appeared, we asked their responses and side effect they feel the most. Patient 1 feels body pain, dizzy, and nauseous. Patient 2 feels nauseous. Patient 3 feels body pain, cramps. Patient 4 feels body pain. And these are the questions we are waiting for. We asked the patient adherence to medications and family support factor during the treatment process. All patients state that they were obedient and supported by the family. The family support such as accompanying the patient, providing a good environment, and also support their physique. So, our conclusion regarding this study is that the patient knowledge about MDR-TB must be improved to support their treatment success. The patient have a good knowledge about the effect of MDR-TB treatment. This statement is proven because they also feel symptoms of side effects, such as vomiting, dizziness, whole body ache, cramps, drug allergy. This number of side effects makes their disease categorized as severe. And from the result of the interview data, all patients adhere to treatment and receive support from their family. That's all for me. I give a special thanks to the director of Doc 2 Hospital Jayapura and the staff of the permissions, and also for the committee of IPCT and all of my audience for listening my presentation. Alhamdulillah. I say wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you Ayu Marliana for the great presentations about MDR tuberculosis treatment. The topic is presented very well and it is very important for us to know about it. Okay, there is already question in the column chat from Universitas Pertahanan. The question is how to convince MDR TB patient to undergo optimal and complete treatment, especially to patients who do not have close family? Please, Ayu can answer the question.
Okay. Good morning, everyone. Before that, thank you for the opportunity and also a great question from Muhraihan. Before that, I'm apologize because I'm not using the background because my laptop doesn't support it. Before answering this question, uh, maybe our research is different from the others. We don't discuss about drugs and drugstores, but we emphasize the role of pharmacy in providing patient knowledge about MDR-TB with the aim of breaking down the number of MDR-TB or TB patients currently of major concern because TB causes a large mortality rate. To answer this question, the next thing we will do is first we're gonna socialize to the public about TB and focus on the adult. The question is, why must be the adult? The tuberculosis mostly affect the adult on their productive years. The risk of TB active is also greater in the person suffering for immune systems and people with undernutrition, and also alcohol disorders and tobacco smoking. For a patient who doesn't have family, uh, we're gonna focus on the socialized for to the hospital to give support for the patient physic. That's all for me. Thank you.
Okay, thank you, Ayu Marliana, for the answer. And for Muhraihan, I hope the answer can understood it well. Okay, now let's move on the next participant with number OP28 from Universitas Islam Indonesia with name Senya Putri Amalia. Let's take a look at the video.
Assalamualaikum warahmatullahi wabarakatuh. Good day, everyone. My name is Senia Putri Amalia, and I'm a representative of the team with member Dr. Apoteker Fitarani Dwi Anandaningrum and Apoteker Ari Wibowo. In this opportunity, I would like to present our review article with the title Bioanalysis of Vancomycin for TDM Application Through Immunosenage PLC, a comparative study. The outline that I will present are introduction, the aim, method, result, and the last one is about the conclusion. First, I will explain about the reason why we do this research. It starts from the vancomycin as first line for MRSA. This is a narrow therapeutic index antibiotic with a high inter-individual pharmacokinetic variability. And also, this has a high risk of toxicity and resistance. Those reasons making it important to be controlled through therapeutic drug monitoring or TDM. TDM application requires an accurate method with high repeatability for quantifying patient vancomycin concentration. Two of the most developed methods are immunosenage PLC, or high-performance liquid chromatography, where each method has its advantage and disadvantage. In Indonesia, TDM is very limited. Therefore, guideline that summarize and compare previous study is needed. So, the aim of this review is to compare a number of studies which have applied immunoassay and HPLC in human biological food to be considered in the selection of bioanalysis vancomycin in TDM service. The method. We start from the collect article with some keyword in the database, and for this review, we choose 34 articles based on the inclusion and exclusion criteria. Then, we got 4 main points for the result. First point is about immunoassay method of vancomycin in human biological food. Immunoassay is divided into various techniques, and we found that almost all of them have a cross-reactivity, like RIA or radioimmunoassay, NFPIA or fluorescence polarized immunoassay that cross-react with vancomycin degradation product, and PETINIA that cross-react with natural biochemical compound immunoglobulin M, and another cross-reactivity. From previous study, also shown correlation between immunoassay method, that category in very strong correlation with the value 0.8. for HPLC method, there are many systems that must be controlled, like stationary phase, mobile phase, internal standard, detector, and another that can affect the result, like present recovery or accuracy parameter, precision, selectivity, sensitivity, and others. And from both of the table, we can see that HPLC has the best sensitivity than immunoassay. And also, HPLC has a high selectivity based on previous research that show the absence of compounds like meropenem, imipenem, analgesic, salicylate, and others that can interfere vancomycin analysis, unlike immunoassay that have cross-reactivity. And from this table too, we can see the accuracy from percent recovery that didn't differ significantly. But fluorescence polarized immunoassay or FPIA has the best precision than another method. And the last one is about comparison between immunoassay and HPLC method. This correlation concentration result was input in the strong correlation category with the value of 0.9. However, several studies had shown immunoassay bias result that a significant difference in some conditions. This is the complete data for correlation between immunoassay and HPLC. And then this is for accuracy and recovery that didn't differ significantly. This is for precision that show fluorescence immunoassay has the best value. The selectivity, HPLC able to separate vancomycin from another compound. Sensitivity show that HPLC has the best value. And the last point is about comparison for efficiency level. For the time analysis, immunoassay has a shorter time due to their simpler step and can be performed in large quantities. Difference from HPLC method that's so complex. A long procedure in HPLC also allow for possible variation and require a specialist technician. About the cost, initial investment of immunoassay is smaller than HPLC, but the cost of analysis HPLC proven to be lower. Finally, the conclusion. From 34 scientific journals, immunoassay can obtain a wrong result because their cross-reactivity in some conditions like renal impairment and high immunoglobulin concentration. In contrast, HPLC needs a long time that can delay patient clinical action. So, immunoassay is recommended for routine TDM because there are multiple samples in a short time. But for some conditions, and a requisite of high sensitivity and selectivity can employ the HPLC method. So this concludes the focus of today's presentation. I'd like to highlight that TDM needs a simple and efficient method, but an accurate and bad method only impact on the patient clinical interpretation, which endanger patient condition. And this is the reference that we are used for this review article. And thank you for all of your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Senya Putri Amalia, for the great presentation about vancomycin. And there is already question from Vicky Daniar from Universitas Islam Indonesia. The question is, what are the inclusion and exclusion criteria that you use for your review article, and how cross-reactivity occur in the vancomycin degradation product? For Senya, please answer the question.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Before I answer the question, I want to say sorry because I'm not used to background in this Zoom because my laptop doesn't support it. And I'm so sorry too for the sound that's not conducive in here. So let me answer for the question. Thank you for Miss Vicky for the question. And the first, for the first question is about the inclusion and exclusion criteria. So the inclusion criteria that we use in our review article is first point is about the articles that publish in English and Indonesia. The second point is about article that result from the research, and then the research article that have a topic about bioanalysis, vancomycin using immunoassay, and or HPLC. And the last point is research article that using human biological fluid sample as the specimen. And for the exclusion criteria that we use are the research, which is a case report or the literature review, the article that doesn't available in the full text, the article that do not include validation parameter, and the article that discuss about pharmacokinetic vancomycin in various patient condition. And the next question is about cross-reactivity between vancomycin and vancomycin degradation product. So here, vancomycin degradation product is CDP1 or crystalline degradation product 1. This degradation is a normal product that results from vancomycin metabolism. But in some condition, like patient that has a renal impairment, they can excrete the CDP1 normally. So the CDP1 will build up in the body. And in the bioanalysis of vancomycin using immunoassay, previous studies show that almost all of them, like radioimmunoassay and fluorescence polarization immunoassay, cannot distinguish between vancomycin and CDP1. So they will read CDP1 as the vancomycin, and it will give a biased result or wrong high yield like that. And maybe that's my answer, and hopefully can satisfy for you, Miss Vicky. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Sanya Putri Amalia, for the answer. And for Vicky Daniar, I hope the answer can be understood well. Okay, next, let's move on the next participant with number OP35 from Universitas Islam Indonesia with name Serina Nabila Putri Hakim. Let's take a look at the video.
Good morning, ladies and gentlemen. My name is Sherina Nabila Putri Hakim, and today I'll be presenting my research, Toxic Evaluation of Nano-Formulation of Propolis Extract. Today's content presentation consists of introduction, objective, method, result, and conclusion. Propolis is a product produced by honeybee and is known to have an immunostimulant effect in increasing the performance of immune system. Propolis contains various complex chemical compounds, one of which is flavonoid, which is responsible for the immunostimulant effect. The flavonoid in propolis have low solubility in water, so that the bioavailability and pharmacological effect are not optimal. The limitation of propolis solubility can be improved by converting it into a nanoparticle preparation, which called SNITs. SNITs has been proven to increase the solubility and bioavailability of drugs. SNIT help dissolve hydrophobic compound by breaking them into tiny particles less than 200 nanometers, and it rapidly forming into oil in water form, nanoemulsion, when exposed to gastric juices. These advantages can increase the drug absorption and avoid first-pass metabolism, and thereby it could increase the immunostimulant effect. SNITs has a lot of benefits, such as small particle size, easily to absorb, delivering drug across the blood-brain barrier, but it also could increase the drug level in our body that may potentiate causing toxicity. Currently, there is no research on the safety of propolis SNITs preparation, so it is necessary to do an acute toxicity test to assess the level of safety. The OECD 425 up and down procedure is used as it is the newest method and has the advantages of using less animal. So, the study was conducted to evaluate its acute toxicity on male with a rat using OECD 425 up and down procedure method. In this research, we're using Western male rats, healthy, aged 2 until 3 months and weighing 140 until 250 grams. If we found significant weight loss, disability, illness, death during study and pregnant, the animal will be excluded from the research. After selecting the animal, we conduct the limit test by administering 2000 mg per kilogram body weight SNE propolis on single male rats. After that, we intensively observe the animal for 4 hours. If the animal survives, we add 4 additional animals and administer with the same dose of propolis SNE. The rats were observed for total 14 days. If 3 animals die or more than 3 animals die, we analyze the LD50 by using AOT for 25 stat PGM, then conduct the main test. If the animal die in administering the single male rats, we evaluate and analyze the LD50 by using AOT 425 and then conduct the main test. The main test will begin by administering 175 mg per kilogram body weight of propolis SNE on single male rats. And then the animal will be observed for 48 hours. If the animal found survive, the next animal will receive higher dose following 3.2 factor progression. But if the animal die, the next animal will receive lower dose following 3.2 factor progression. After analyzing the LD50 using AOT software, the statistical estimate for LD50 in limit test and main test are greater than 2000 mg per kilogram body weight. The most common toxicity signs that were found in 10 male rats were lethargy that was found in 8 male rats and changing behavior that were found in 5 male rats. In conclusion, propolis extract in SNE formulation was non-toxic on Wista male rats. The LD50 value of propolis in SNE was greater than 2000 mg per kilogram body weight. I would like to say thank you to my friends Vina and Bagas for helping me and participating in this research with me. Thank you so much.
Okay, thank you, Serena, for the great presentation about propolis. And now we move on to the Q&A session. There is question from Vina Wulan Junior from Universitas Islam Indonesia. The question is, what is the output for the toxicity assay? Okay, please, Serena, you can answer the question.
Okay, thank you. First, I would like to say thank you to Vina Wulan Junior for the question. That is, what if the propolis output? What is the toxicity assay output? So the propolis we use in this study is a propolis SNE product. And in this research is an optimization step for the product to be able to distribute around Indonesia, because we know that propolis has a very amazing pharmacological effect such immunostimulant. And in order to be able to distribute around Indonesia, we need to obtain distribution permit from BPOM. It's like an Indonesian FDA, which need toxicity profile from the propolis needs. Is that my answer? That's my answer.
Okay, thank you. There are also one question again from Hamnah from Universitas Ahmad Dahlan. The question is, if propolis extract formulated by SNIDs give toxic effect, so any recommendation to formulate SNIDs? Thank you. Please answer the question.
So if there is a toxic effect from the propolis, it's not about the formulation, but it's about the dose. Maybe we could give lower dose for the preparation, because this research is to evaluate the most toxic dose for the SNE propolis.
Okay, thank you, Serena, for the answer, and for Vina and Hamna. I hope the answer can understood with well. Now let's move to the last participant in this oral presentation with number OP36 from Universitas Ahmad Dahlan with name Noura Reni Rahma Putri. Let's take a look at the video.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Firstly, let us introduce ourselves. We are from Faculty of Pharmacy, Universitas Ahmad Dahlan. And in this opportunity, we would like to present our study about Indonesian main active compound as main protease inhibitor of SARS-CoV-2, a molecular dating study. This study was written by Noura Reni Rahma Putri, Muhammad Reski Manu, Khalil Fari, and Dwi Utami. And there are the outlines of this presentation: introduction, objective, material and methods, result and discussion, conclusion, references, and acknowledgement. COVID-19 or coronavirus disease 2019 is a disease of the respiratory system caused by SARS-CoV-2. The virus was identified in Wuhan, China, in the end of 2019. The virus widely spread over many countries, and then in March 2020, World Health Organization declared COVID-19 as pandemic. Indonesia is the largest maritime and archaeological country in the world. The area of sea marine of Indonesia is for about 75% of Indonesia's total area. Indonesia has abundant marine resources such as fish, coral, and seaweed. Some of them have active compound ingredients and potentially developed as antiviral drugs. M protein responsible for the transport of nutrients, the blood release, and the formation of envelope. Main protease of SARS-CoV-2 is the main target of antiviral drugs. Oseltamivir is a first-line antiviral drug. During the ongoing outbreak of COVID-19, most patients of COVID-19 who are symptomatic have used oseltamivir. Oseltamivir carboxylic acid was more favorable to bind to the active site of main protease effectively. And the objective of the study was to determine the potential of conjugated sulfate and calcium in inhibiting the main protease of COVID-19 through drugging analysis. Next, there are the material and methods of the study. The method was drugging analysis using main protease and main peptide crystal structure of SARS-CoV-2. The ligand was oseltamivir, conjugated sulfate, and calcium arginate. And there are the process of drugging molecules. The first step is download the file from protein data bank and PubChem. After that, ligand and receptor isolation using Autodoc Tools 1.5.6, and then docking process by Autodoc Vina. And the last step is visualization using BioVia Discovery Studio. Next, there are the results of this study. The binding affinity between oseltamivir and the 6LU7 receptor was minus 7.3 kilocalorie per mole. And after docking process, visualization are conducted to determine the amino acid residue and the interaction between the ligand and the receptor. And the interaction were conventional hydrogen bond, carbon-hydrogen bond, homologous bond, p-alkyl, unfavorable donor-donor, and p-sigma. The binding affinity between conjugated sulfate and the 6LU7 receptor was minus 8.1 kilocalorie per mole, and the interaction were homologous bond, conventional hydrogen bond, and carbon-hydrogen bond. And the Togic score between calcium arginate and the 6LU7 receptor was minus 10.3 kilocalorie per mole, and the interaction were conventional hydrogen bond, homologous bond, and unfavorable receptor acceptor. Next, there are the results of the docking process between the ligand and the 2-GDP receptor. The binding affinity between oseltamivir and the 2-GDP receptor was minus 7.4 kilocalorie per mole, and the interaction were conventional hydrogen bond, van der Waals bond, alkyl, and diethyl. The total score between conjugate sulfate and the 2GTP receptor was minus 8.9 kilocalorie per mole, and the interaction were van der Waals bond, conventional hydrogen bond, and carbon hydrogen bond. And the binding affinity between calcium arginate and the 2GTP receptor was minus 10.5 kilocalorie per mole, and the interaction were conventional hydrogen bonds, van der Waals bonds, and unfavorable acceptor-acceptor. Based on the result, conjugate sulfate and calcium arginate have lower binding energy than oseltamivir. The amino acid residue allows the ligand to contact the receptor, and then it has inhibitory activity. Conjugate sulfate and calcium arginate have lower binding energy than oseltamivir in 2GTP receptor and in 6LU7 receptor. The smaller binding energy obtained, the more stable the bond form. The binding site is an area of protein to bind ligands which will affect the conformation and function of protein. The remaining amino acid residue play an important role in the interaction between the homologous and the ligand. The amino acid interaction allows the ligand to contact the receptor, and then it has inhibitory activity. Therefore, we can conclude that conjugate sulfate and calcium arginate have a great potential as COVID-19 main protease inhibitor based on the binding affinity and the interaction between the ligand and receptor. Next, there are the variants of the study. We thank Computational Chemistry Research Group of Faculty of Pharmacy, University of Magellan for all supporting this study. That is all. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Nora, for the great presentations. Now let's move to the Q&A session. There is already a question from Kurnia Sandi from Universitas Pertahanan. The question is, how about the effectivity and the cost if we make the seaweed active compound as the main protease inhibitor of SARS-CoV-2? Okay, please answer the question.
Okay, thank you for the question. Is my voice clear now? Is my voice clear now?
Yes, it's clear.
Okay, it's about the effectivity and the cost if we make the seaweed active compound as the main protease of SARS-CoV inhibitor. Okay, the first one is the effectivity. From the result, between the oseltamivir and also the arginate, calcium arginate, based on the binding affinity, we know that calcium arginate have the lowest binding energy than the oseltamivir. So it can be concluded that the effectivity of the calcium arginate is more than the oseltamivir. And then about the cost if we make the seaweed active compound. For the formulation, it will take a long time. So we need some practice to determine the best formulation of the compound. And then for the activity, the binding affinity shows the energy between the ligand and the receptor. So if the lower energy we obtain it, and then we will have the more stable interaction between the ligand and the receptor.
Okay, thank you, Nora, for the answer. There is one more question from Jihad Antar from Universitas Bhakti Kencana. The question is, why did the research use molecular docking for the method compared to other drug discovery application like HKSA? What is the advantage of using that specific method? Please, Nora, to answer the question.
Okay, thank you for the question. Uh, in molecular techniques, in computational study, there are a lot of methods we can use, but there are a specific criteria. The first one, there are the structure of the compound, and then there are the protein structure of the receptor. So if we have those compound and the protein, we can do the uh, the molecular technique study because based on its method, we can determine the binding affinity, and it is most representative of the interaction between the ligand and the receptor.
Okay, thank you, Noro, for the answer and for the everyone that asking. I hope the answer can be understood with well. Okay, now we have arrived at the last series for this oral presentation. I would like to thank all of the judges, participants, and attendees who have participated in this oral presentation from beginning to the end. I would like to remind all participants and attendees that this afternoon at 1 p.m. there will be a talk show with the theme Sustainable Pharma Education at the time of COVID-19. Furthermore, we will continue with the next session in this competition. It is a poster presentation. Okay, I need to remind you, there are some information that I will announce and must be complied by participants during the presentation season. First, all participants and attendees are required to register on the form that has been distributed by the committee in the chat column. Second, participants who will make presentations are those who enter into the top 10, and the order of presentations is determined by the committee. Third, in the next session, we will present a video presentations of each participant. Are kindly requested to attendees who are not interested to talk can be muted the microphone. Okay, without wasting any more time, in the first order with number PP1 from Universitas Islam Indonesia with name Ilya Marsha Rohila. Please take a look at the video.
Assalamualaikum warahmatullahi wabarakatuh. Hello, my name is Ilya Marshruhila. I'm a pharmacy student of Universitas Islam Indonesia. Firstly, I would like to say thanks to God Almighty, Allah Subhanahu wa Ta'ala, because of the blessing and graces we can meet here even through virtually. Salawat and salam always given to Rasulullah Sallallahu Alaihi Wasallam, who bring us from the darkness into the bright era. I also want to say thanks for the committee and the judge who gave me opportunity to joining this competition. The theme of this poster competition is health and medicine information. It's a great honor for me to present my poster about stroke. What do you know about stroke? I chose this disease because, as we know, stroke is usually happens at the geriatric people or the old age people. It's make our younger generation doesn't really care about this disease. So I have ever heard the sentence before, the future is created by what we do today. So I hope we can do an action from now to prevent the stroke later when we get old. Okay, right now I will go to the definition of stroke. There are the definition of stroke by WHO in 2014. It is the blockage of blood vessel that goes to the brain that can inhibit the blood flow to the brain. And we can conclude that stroke is a continuously condition or situation of arteriosclerosis. And then there is a data or statistic you can see here. Stroke get the second ranks after systemic cardiac disease as the highest mortality rate according to WHO in 2020. There are more than 5 million people died on 2020 because of stroke. And then Based on Ministry of Health of Republic Indonesia, stroke divided into two kinds based on the risk factor. First is stroke ischemic. It is because of the blockage of blood vessel, and then the second is hemorrhagic stroke. It is because of the bleeding of blood vessel in intracerebral or uh, subarachnoid. I'm sorry. And then let's move into the symptoms. I will explain it one by one. Firstly, is difficult to speak. I think all of us already seen the patient of stroke. They cannot speak easily because of a disturbance of their neuron system or nervous system that can make weakness of their muscle to control their tongue, lips, or their mouth. And the second is losing balance. It is because of disturbance of their brain that control balance of our body. And the third is weak movement. It is because they cannot produce a lot of energy without an oxygen, because they cannot supply enough oxygen. And the last one is unconsciousness or even died, because it actually it has a same symptoms, same reason like the three symptoms before. It is because of disturbance of their brains. Now, I would like to describe about medicine that usually or common using by the patient of stroke. First, there is antihypertensive drug to lower the blood pressure. Same like ACE inhibitor to lower blood pressure, and there is statin to decrease production of cholesterol in liver, and then anticoagulant to inhibit the blood coagulation, and there is alteplase to inhibit or overcome the blood clotting. And then the last explanation about preventive, how to prevent this disease. Firstly is do a work out every day. Maybe you can work out around 15 minutes every day. Yes, you can decide it by yourself. And second is consume less salt, because consume excess salt will increase the amount of sodium in cells and disturb the balance of fluids in our bodies. And the third is be aware of your history illness. It is especially for people who have blood pressure illness. You must take your drug and control your history illness, your illness every month or weeks to the doctor or the pharmacist. And the last one is do not smoking and drink alcohol. As we know, cigarette contain more than 4,000 harmful chemicals, including nicotine, tar, or carbon monoxide that can disturb our blood circulation. That there's no impossibility to cause stroke. That's all. Thank you for your attention. I'm sorry if there's a lot of mistakes. Uh, I'm sorry if there's a lot of mistakes. I'm sorry again. Thank you for your attention. I hope this helpful for all of us. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Okay, that's the presentation from Ilya. Next, we will move to the Q&A session. I will choose just one question from Muhraihan from Universitas Pertahanan. The question is there a way to educate people with smoking and alcohol addiction to prevent stroke? Okay, please Ilya can answer the question.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning. Thank you for your question. Firstly, I want to say thanks for the judge and the committee that give me a time to join this competition and join this Q&A section. All right. Of course, there are. First, start from yourself. Then maybe you can educate your family, like your father or your uncle or people around you. And then maybe you can join a lot of organizations that care to our health. Something like, maybe, do you know forum anak or something like that? It's educate us to avoid an alcoholic and smoking, etc. That's all. Thank you.
Thank you, Ilya, for the answer, and thank you, Muhammad Raihan, for the question. Now let's move on the next participant with number PP18 from Universitas Al-Ghifari with name Ira Rahmawati. Please take a look at the video.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. First, I want to introduce myself. My name is Ira Rahmawati. I am a pharmacy student from Al-Ghifari University, Bandung. Today, I will present a poster that I made entitled Self-Medication for Cough with Pro Per 3. The reason I chose this title is because currently the COVID-19 pandemic is troubling many people and coughing is the one of the symptoms. However, does the cough you experience come the coronavirus? Not necessarily. Therefore, everyone must know what cough looks like, from the causes to treatment to relieve panic during the current pandemic. And in this poster, I will explain about self-medication for cough with proper tree. What is proper tree? Before knowing proper tree, I will explain about cough. What is cough? Cough is the most effective defense mechanism to eliminate foreign materials, including various pathogens from the respiratory tract. Several causes of cough, such as 1 is clearing the throat, 2 viruses and bacteria, 3 smoking, 4 asthma, 5 medicine, for example, captropil. The last is other condition, for example, complication of disease, dust, and others. What to do when coughing? You can take self-medication using PRO per 3. PRO consists of P is preventive, R is recognize, and O is overcome. Number 1 is preventive. You can prevent coughing in several ways, such as quit the smoking, because cigarette smoke contains chemicals that are very dangerous for the health of our lungs. Number 2 is dietary change. Eat regularly and avoid food with excess oil. Number 3, avoid disease transmission by maintaining distance and maintaining personal hygiene. Next is R, recognize. Identify the causes of cough and the type of cough. In general, cough is divided into two. One is productive cough or wet cough. It's a cough that typically brings up mucus. Wet cough can be acute and last less than 3 weeks, or chronic and last longer than 8 weeks. Number 2 is dry cough or non-productive cough. It's cough that doesn't bring up mucus. It may feel like have a tickle in the back of throat, triggering cough reflex, giving hacking coughs. Next is O. O is overcome. Overcome coughs with per 3. What is per 3? P number 1 is practice cough adequate. Number 1 is cover your mouth and nose with a tissue when you cough. Put your used tissue in the rubbish bin. If you don't have a tissue, cough into your oversleeve or elbow, not your hands. Wash your hand with soap and running water. And the last, dry your hands thoroughly with the disposable paper towel. Pen number 2 is provide at-home treatment, such as number 1, keep hydrated by drinking plenty of water. Number 2, use cough drops to soothe your throat. You can use antitussive or antihistamine for non-productive cough. Example is dextromethorphan. This is non-sedating opiate is a component of many over-the-counter OTC cough remedies and has been shown to suppress acute cough in a single meta-analysis. Another example is diphenhydramine. Diphenhydramine is a first-generation antihistamine. H1 is approved in several countries, including England and the United States, as an OTC antitussive. It is reported to reduce cough reflex sensitivity in patients with cough due to URTI. And you can use expectorant for productive cough. Example, ambroxol. Ambroxol is a bromhexine active metabolite. It also shows other antitussive effects other than expectorant. Other example is guaiacol. Example of an OTC use expectorant and can beneficial for the treatment of a productive wet cough. And next, you can gargle with warm salt water regularly to remove mucus and shut your throat. Next, avoid irritants, including smoke and dust. And the last, use decongestant spray to unblock your nose and easy breathing. And the last is P3, is perform medical treatment. Medical treatment is carried out in case of the condition. Number 1, thick mucus that's green or yellow in color. Number 2 is fever. Number 3 is out of breath. Number 4 is chest pain. Number 5 is night sweats. And the last is weight loss. If your cough is likely due to bacteria, your doctor will prescribe antibiotic. They may also prescribe either expectorant cough syrups or cough suppressant that contain codeine. Okay guys, maybe that's all I can explain. Hopefully, it's useful and easy to understand. Always remember, proper treat if you cough. Don't panic, keep your health protocols in check during in this pandemic. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Ira, for the nice presentation. Now let's move to the Q&A session. For participants and attendees who want to ask, can drop the question in the column chat. Well, it's look like there are no questions. Oh, okay. There are questions from Hansen. The question is all the medicine can be purchased freely in the pharmacy? Thank you from Universitas Surabaya. Please, Ira, answer the question. Halo, Ira. Okay, unfortunately, Ira can't answer the question yet. Okay, because of the time, let's move on the next participant with number PP21 from Universitas Setiabudi with name Rizky Ayu Febriana. Let's take a look at the video.
Assalamualaikum warahmatullahi wabarakatuh. Hello, my name is Rizky Ayu Febriana. I'm from Setiabudi University, Surakarta. Okay, I will present my poster in this time on the sub-theme of medicine and health information. I choose the title for this poster, Some Treatment for Vertigo. Before going into an explanation of the material on this poster, I will explain a little thing about the poster design. I designed this poster with two applications. There are CorelDRAW design and Canva application. Both of these applications can be run on handphone, so their operation is also quite easy. For the base color, I choose a light blue and light Tosca color combination because blue can give the impression of calmness and freshness when the people see it. In this poster, I don't provide a lot of text and animated image because I want to make a minimalist poster, but the message can reach the reader. Other than that, I just wrote the important points of the title. Why? Because I want to invite the readers to find out more about this point and not just stop at the material in this poster. In this material, I raised about vertigo because in the environment around me, many people are still confused and anxious when experiencing vertigo. They don't really know what vertigo is and how it is treated. Before we go to the treatment for vertigo, let me show you what is vertigo. Vertigo is a collection of symptoms or a syndrome due to balance disorder in the vestibular system or disorders of the central nervous system. It should be noted that vertigo is a collection of symptoms, and it means there is not a disease. Vertigo is usually found in complaints of a feeling of spinning or a sense of movement from the surrounding environment. And then, what if vertigo coming? How to solve it? There are three ways to treat vertigo. Vertigo treatment can be done with therapy, traditional medicine, and pharmacological treatment. Therapy for the treatment of vertigo is called by particle repositioning maneuver or PRM. There are 5 types of PRM that can be used according to the type of vertigo complaint, including the Apley maneuver, the Simon maneuver, the Lampert maneuver, the forced prolonged position, and the brain drive exercise. This brain drive exercise therapy is usually used for patients to practice at home. And the second way is by consuming herbal plants. Some herbal plants that can be used to treat vertigo are easy to obtain. These plants are like ginger, magnolia flower, rosella flower, and coriander. Besides being easy to find it, these plants are also easy to process. Brew it with hot water and then drink it regularly. That's enough. And whereas for pharmacological treatment is to use vestibular suppressants like benzodiazepines such as diazepam and clonazepam, and then use antihistamines such as meclizine and diphenhydramine. Benzodiazepines are used to reduce the sensation of spinning, while antihistamines are used as suppressants in the vomiting center so they can reduce nausea and vomiting due to motion sickness because by vertigo. Okay, I think enough for my presentation. If with this little knowledge I have an error, I very apologize. That's all, and thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, that's the presentation from Rizky. Such a great presentation. Now let's move to the Q&A session. There is question from Hansen from Universitas Surabaya. The question is, can vertigo be totally cured? Okay, please Rizky can answer the question.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Firstly, I'm sorry because I cannot go background because my internet connection is not stable if I share in laptop. Okay. Thank you, Mr. Hansen, for your question. Can vertigo be totally cured? Actually, vertigo can cure, but what is that we cured is the disease, not the symptom, because I say that vertigo is a symptom. So we can found first what is the disease of this vertigo symptom. And actually, vertigo can cure with operation or surgery, but it is very, very risky for our hearing system. I think enough for my answer. I hope it can clearly answer for your question. Thank you.
Okay, thank you Rizky for the answer and thank you Hansen for the question. Now let's move on to the next participant with number PP25 from Universitas Islam Indonesia with name Inesha Rayadillah Nurdin. Please take a look at the video. I'm sorry, the committee, but the sound is not here.
The misuse of herbal medicine that are not guaranteed safe and efficacious for the human body. So let's move to the first part of my poster. Did you know that some herbal pillows have been proven scientifically can increase the immune system of the body? First, the Burkina or known as kunyit, an in vivo study showed that the dosage of 5 and 10 milligram per kilogram mice that the immune system. And the second one, we have rhiza or known as temulawak. An in vitro study showed that this extract can increase the immune, increasing the proliferation of the lymphocyte cell, which has responsible to the immune system. And next, we have Phyllanthus neuri, or known as maniran. An in vivo study using tilapia fish showed that this extract can increase the immunomodulatory activity by increasing the activation of neutrophil and the antibody. And then the Sampiloto or Andrographis paniculata, an in vivo study using mice showed that the activation of immunomodulatory is showed by the increasing of immunoglobulin G that responsible to the immune system. There is another Indonesian herbal medicine that can increase the immune system, but these four herbal are the most popular herbal among the other Indonesian herbs. So after know what kind of herbal that can help us to increase the immune system, we also have to know what kind of herbal product that's safe to consume. First is the traditional or homemade herbal medicine. Basically, we can make it at home and based on empirical evidence and hereditary recipes. We can consume it by considering some points. First, we have to make sure that the making process is appropriate, and also we have to consume it in the recommended dosage. And next is the registered herbal product or the industrial herbal product. This is the herbal product that registered by BPOM or the Medicine and Food Regulatory Agency of Indonesia. There are three kinds of this herbal. First is the jamu or traditional herbal with this logo, and then the standardized herbal medicine with this logo, and next logo is the phytopharmaca. So we can consume the herbal product that contain one of these three logos because this product is already guaranteed by BPOM, safe and efficacious for the human body. Talking about the herbal product, there is still cases about the misuse of herbal medicine because there are so many herbal product offered out there. So here is the reaction tips for the society to use herbal wisely and to choose the right and safe herbal product. Reaction is abbreviation. So REA is referred to renal and hepatic toxicity may be induced by several compounds in certain herbal medicine. So we have to make sure that the herbal we consume contain the safe compound, and always talk to your doctor or pharmacist before using herbal in long term, especially for people who has the renal or hepatic disorder. And C is referred to check. Always check the label, check the package, the expiration date, and the distribution permits. We can check the distribution permits in the website here. T is referred to think critically. If we found out a herbal that advertise the fast healing process, because mainly the fast healing process of herbal can be caused by the addition of chemical drug substance. Meanwhile, the herbal medicine should be contain of 100% of natural product. And ion is referred to interaction because there might be interaction of the herbal and the other medicine that people consume. Ask the pharmacist for the potential drug interaction between the combination of medication. So by knowing this information, hopefully can help the society to consume and choose the right and also safe herbal medicine product to maintain the health, mainly to increase the immune system against the infectious disease nowadays. So lastly, I would like to say thank you very much for the attention. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Inesha, for the presentation. Now let's move at the Q&A session. There is question from Karina Primatias from Universitas Ahmad Dahlan. The question is, how we know that the herbal medicine is safe for us? Thank you. Okay, for Inesha, please answer the question.
First of all, I want to say thank you very much for the chance. I'm very sorry for I can rename my name and also for the background. So I will answer the question about how we know the herbal is safe or not. So basically, a safe herbal, there is two kind of herbal. I say before, the traditional one, what we know, is it safe or not? For the traditional herbal, because it's based on the empirical evidence and hereditary recipes, so we can know it by asking for the people who expert on that side about the traditional medicine, because there is no evidence in reported, or also we can search its research about the herbal, so we can know the traditional herbal is safe or not. And for the product, industrial product, we can know it's safe by checking it on the website of BPOM. We can enter the number registration of the product, or we can also know that the product is safe by noticing the logo of the product, because herbal product has the 3 logos that registered by BPOM, so we can choose the safe herbal based on that consideration. So basically, herbal, I want to give the another perspective to the society, because not herbal is not 100% safe. The If we know the other side, it's prohibited for some people that have complication or have another disease before. So always making consultation before using herbal, especially in long term with your doctor and also for the pharmacist. So I think that's all my answer. Thank you very much for all the question.
Okay, thank you, Inesha, for the answer, and thank you, Karina, for the question. Hope the answer can be understood with well. Now let's move to the next participant with number PP31 from Universitas Ahmad Dahlan with name Karina Primatias Ningrum. Let's take a look at the video.
Assalamualaikum warahmatullahi wabarakatuh. The honorable judges and fellow participants. First of all, let me introduce myself. My name is Karina Primatias Ningrum, and I'm from Ahmad Dahlan University. At this time, I'd like to explain my poster that title is Drug Spec Label. Ever wonder how your moms know what medicine to give you when you are not feeling well? A lot of mom knowledge and a little but very important things known as the drug facts label. Ladies and gentlemen, the drug facts label is on all OTC medicine. OTC stands for over the counter and refers to medicine you buy without a doctor prescription. The drug facts label can help you choose the right drug for you and your problem, and to help you use the medicine safely. It's on the box or other container your medicine comes in. The study from assessment of the people self-reporting reading of medicine labels state that just 74.2% of people have adequate information about medicine provided on their medicine labels. Most of them didn't read the label completely. If patient not reading and understanding medication label, this may lead to medication error, such as taking the wrong medication or the wrong dose. The FDA created the regulation to simplify and standardize the drug facts label. These labels are short and simple and typically have 6 main parts. The first part is active ingredients and purpose. Active ingredients are ingredients that make the medicine work. Purpose tell you what type or the category of medicine it is. Second one is uses. Defines what symptoms or illness you can expect the medicine to treat. The third part is warning. Explain when you shouldn't take a particular medicine, when to talk to a doctor, and when to stop taking medicine. The fourth part is direction. Tell you how and when to take a medicine. The fifth part is other information. Provide you important information you should know, that where to store it. The sixth part is inactive ingredients. List the other ingredients in the medicine, such as color and flavoring. Ladies and gentlemen, remember, all medicine, even OTC ones, can cause side effects you don't want. So, check the label first before you taking it. Thank you, and wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Karina, for the nice presentation. Now let's move on the Q&A session. There is question from Alia from Universitas Pertahanan. The question is, what should the public do if a product is counterfeited? For example, the BPOM license is falsified. Please, Karina, can answer the question.
Hello, can you hear my voice?
Yes, it's clear.
I'm sorry, for the question is, what should the public do if the product is counterfeited? For example, the BPOM
license is falsified. So the question is, what should we do if the BPOM the BPOM I'm sorry, browser is falsified. So for the public, sure is we cannot just take the information. So we cannot. I'm sorry. It's my okay. We cannot take the information, all of them. Just take all information. So we need to read the more information to understand what I'm sorry is okay. Yeah. If the webform the browsers is falsified, we need to clarify more and more information so we have more understand for the fact label. For the example, if we buy a medicine, we should ask the pharmacy first what the ingredients of medicine. So we know the clearly what the medicine work for us. If we just many people in Indonesia just buy a medicine, just say the drug and then they leave. I'm sorry.
Yes.
Oh, can you hear me? Okay. Is my internet disconnect that time?
No, it's
Hello?
okay. You still connect with us.
Yeah.
Okay, I think Karina is disconnected. So,
Hello? Can you hear
Alia?
me?
Okay, Karina is back.
Hello, can you hear me?
Yes.
Oh,
You can
okay.
continue the answer.
Yeah, I'm sorry. I don't know how much I will explain because the connection is unstable. But you need to know first that if we bought the medicine, we should ask the pharmacy first what is the medicine work for us. Many people in Indonesia they just bought a medicine, just tell the drug name and then they don't know what to use the medicine properly. So ask the pharmacist first before you buy the medicine, so it can help you to have a medicine properly, so it can work more. It can have a more effective for you. Thank you. Sorry for the bad connection. Okay.
Yes. Thank you, Karina, for the answer. And for Alia, I hope the answer can be understood well. Okay, now let's move to the next participant with number PP32 from Universitas Islam Indonesia with name Dwi Nursita Lestari. Let's take a look at the video.
The Honorable for All Juries International Pharmacy Talk Competition. Assalamualaikum warahmatullahi wabarakatuh. My name is Dwi Nursita Lestari. I'm from Professional Pharmacy Student, Universitas Islam Indonesia. Now, I want to present my poster about vaccine against COVID-19. The body has many ways of defending itself against pathogens or disease-causing organisms. Skin, mucus, and cilia all work as physical barriers to prevent pathogens from entering the body in the first place. When a pathogen does infect the body, our body's defenses, called the immune system, are triggered and the pathogen is attacked and destroyed or overcome. A pathogen is a bacterium, virus, parasite, or fungus that can cause disease within the body. Each pathogen is made up of several subparts, usually unique to that specific pathogen and the disease it causes. The subpart of a pathogen that causes the formation of antibodies is called an antigen. The antibodies produced in response to that pathogen's antigen are an important part of the immune system. You can consider antibodies as the soldier in your body's defense system. Each antibody or soldier in our system is trained to recognize one specific antigen. We have thousands of different antibodies in our bodies. When the human body is exposed to an antigen for the first time, it takes time for the immune system to respond and produce antibodies specific to the antigen. In the meantime, the person is susceptible to becoming ill. Once the antigen-specific antibodies are produced, they work with the rest of the immune system to destroy the pathogen and stop the disease. Antibodies to one pathogen generally don't protect against another pathogen, except when two pathogens are very similar to each other, like a cousins. Once the body produces antibodies in its primary response to an antigen, it also creates antibody-producing memory cells, which remain alive even after the pathogen is defeated by the antibodies. If the body is exposed to the same pathogen more than once, the antibody response is much faster and more effective than the first time around, because the memory cells are at the ready to pump out antibodies against the antigen. This means that if the person is exposed to the dangerous pathogen in the future, their immune system will be able to respond immediately, protecting against the disease. So, how vaccine help? Vaccines contain weakened or inactive parts of a particular organism or antigen that triggers an immune response within the body. Newer vaccines contain the blueprint for producing antigens rather than the antigen itself, regardless of whether the vaccine is made up of the antigen itself or the blueprint, so that the body will produce the antigen. This weakened version will not cause the disease in the person receiving the vaccine, but it will prompt the immune system to respond much as it would have on its first reaction to the actual pathogen. Some vaccines require multiple doses, given weeks or months apart. This is sometimes needed to allow for the production of long-lived antibodies and development of memory cells. In this way, the body is trained to fight the specific disease-causing organisms, building up memory of the pathogen so as to rapidly fight it if and when exposed in the future. When someone is vaccinated, they are very likely to be protected against the targeted disease. But not everyone can be vaccinated. People with underlying health condition that weaken their immune system, such as cancer or HIV, or who have severe allergies to some vaccine components, may not to be able to get vaccinated with the certain vaccine. These people can still be protected if they live in and amongst others who are vaccinated. When a lot of people in a community are vaccinated, the pathogen has a hard time circulating because most of the people it encounters are immune. So, the more the others are vaccinated, the less likely people who are unable to be protected by vaccines are at risk of even being exposed to the harmful pathogen. This is called herd immunity. This is especially important for those people who not only can be vaccinated but may be more susceptible to the disease we vaccinated against. No single vaccine provides 100% protection. And herd immunity does not provide full protection to those who cannot safely be vaccinated. But with herd immunity, these people will have substantial protection, thanks to those around them being vaccinated. Vaccinating not only protects yourself, but also protects those in the community who are unable to be vaccinated. If you are able to, get vaccinated. Thank you.
Okay, thank you, Dwi Nursita Lestari, for the presentations. The topic is presented very well, and it's very important to us to know more about COVID-19 vaccine. And I'm sure that the audience is already sending a lot of questions in the chat column, but I will give this change for the one who never asking before. Okay, the question is from Iwang Davis Setiawan in Universitas Islam Indonesia. The question is, what's the background you choose vaccines as the main topic of your poster? Okay, please, Dwi, you can answer the question.
Okay, thank you for the great question from Mr. Iwang. The question is, what is the background you choose the vaccine as the main topic of your poster? Okay, well, as we know, the vaccine is an interesting issue since last year until now. And with the government programs about the vaccination, I think we are, as the pharmacy students, need to educate the public, need to educate the people about vaccines, because there are so many people that still not understand about how vaccine work, how vaccine can help us. So I think educate the people about vaccine is an important thing now. And as we know that not everyone can be vaccinated. People who have the disease as the cancer, severe allergies, HIV, or autoimmune maybe can to get the vaccinated. So I hope this poster can educate the people to we as healthy people. If we can get the vaccines, please get the vaccine. Maybe that is my answer. Thank you very much.
Okay, thank you, Dwi, for the answer. And for Iwang, I hope the answer is can well understood. Next, let us move to the next participant with number PP33 from Universitas Islam Indonesia with name Iwang Davis Setiawan. Please take a look at the video.
Assalamualaikum warahmatullahi wabarakatuh. Medicine is something that is considered necessary by all life or society. However, unfortunately, in the management of medicine, the community is still very lacking. Starting for getting drugs, using drugs, storing medicinal, and how to dispose it. All of which the community still make mistake. Therefore, together with the Ministry of Healthy of the Republic of Indonesia, with a movement called Germas that stand for Gerakan Masyarakat Hidup Sehat or Healthy Living Community Movement. It is making innovation called Dagusibu. Dagusibu stand for dapatkan or get, gunakan or use, simpan or save, or and buang or dispose. Which is this time I illustrate in the poster with helps it will be easier for the public to understand and implement it. In the poster, it shows how to use the right medicine because there are people who are still wrong in consume medicine. For example, when the pharmacist said the medicine need to take 3 times a day, the community still take in the morning, afternoon, and evening or night. However, 8 hours to get the maximum therapeutic result. Moreover, in terms of drugstore, most people stores in the refrigerator or cabinet that are the easily affordable and are not spread children or sunlight. In addition, there is information on how to problem dispose of drugs to avoid case of drugs about the illegal drug sales. Keeping the medicine according to the corridor in our duty. Therefore, let us jointly abide by rules in the care of drug that can be benefit to all. I am Iwan Davistiawan from University Islam of Indonesia. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you Iwang Devi Setiawan for the good presentation. Now let's move to the Q&A session. Okay, there is question from Zulfa Nur Afifah from Universitas Islam Indonesia. The question is, what underlies you to promote the community by using this method? Please, you can answer the question.
Okay, thank you for Zulfa for the question. And then the urgency of the using the gazebo to treat the drugs properties starting off for the get start storing and using and disposing in order to get the optimum effect. And because there are still many people and they still do not understand how to the use for the drugs. For example, the patient from the etiquette in the 3 times days and the people consume in the morning, afternoon, evening, and the night or the night. It is this is a very wrong, and with salt become for every age times. Thank you.
Okay, thank you Iwang for the answer. And for Zulfa, I hope the answer can understood with well. Now let's move to the next participant with number PP42 from Universitas Ahmad Dahlan, Rosawati Eka Mulyani. But unfortunately, this participant named Rosawati was disqualified because she was practicing at the hospital and was not present at today's event. Because of that, now let's move on the next participant with number PP43 from Universitas Ahmad Dahlan named Zulfikar Muhammad Aditama. Let's take a look at the video.
Hi, my name is Zulfikar Aditama and I'm a student in Faculty of Pharmacy, Ahmad Dahlan University, Yogyakarta. Today I'm presenting educational poster entitled Are COVID-19 Vaccines Really Concerning? As we all know, we are now in a situation that we have never imagined before, a global pandemic that affects almost all countries in the world. Economies are falling, starvations, high unemployment rate, force students to study online, millions of people's dreams are cancelled, and many other bad things happen amid the pandemic. Almost a whole year we've been dealing with COVID-19 health protocols. Although me, personally, I love to stay at home and stay away from people because I'm more like an introvert person. But somehow it's also tiring because it's not only affecting me as a person, but it's affecting the whole world and people's future. Imagine being a student who can't study online because they can't afford internet, or simply because they prefer to study with teacher in real life, or students who have planned to study abroad, or people who plan to go overseas to get a better opportunity in life and their plan got cancelled because of this pandemic. It's so catastrophic, isn't it? I haven't even mentioned about the economic impacts. This pandemic is a disaster and we should end it as soon as possible. One of, if not the only way, is vaccination. But this COVID-19 vaccination plan left people in frustrations. People are debating whether these vaccines are safe or not, while majority others accelerate the process of making the vaccines in order to fasten the availability of the vaccines. So let's start talking about the public concerns. The first question is, can we get a COVID infection from the vaccines? The answer is no. The data showed that COVID-19 vaccines do not have infectious material in them. They are designed to create or inject a harmless piece of coronavirus. There might be some side effects such as fever and soreness, but they are not as bad as having a bad COVID-19 illness. Now, the second most highly asked question is, are the COVID-19 vaccines safe and effective? The World Health Organization determined COVID-19 vaccines should have at least 50% efficacy, and among all vaccines that already distributed and ready to use, all of them have more than 50% efficacy. Now keep in mind that these scientists do not have any intention to harm people. The standard of the vaccines is being determined to help us as mankind to solve this problem as soon as possible. And that's a common sense. Now let's move on to the next section. The question that comes up next is, do we have a choice? Let's say, what if most people reject getting vaccinated? Like what I mentioned earlier, vaccination is likely the only way we can do to end the pandemic as soon as possible. The other thing that comes up in mind might be, oh, why don't we wait for herd immunity to be happening so all people will be immune? Well, herd immunity needs years to be done. And also, COVID-19 virus is a virus that keeps evolving. And every time it evolves, it always getting more and more dangerous. If we keep waiting, the virus could be evolved into a lethal virus and more people die because of this. Let's think in more common sense. More people don't get vaccinated, transmission of the virus keep going. The pandemic lasts longer. International and local restrictions obstruct people to achieve their goals, etc., etc., and then economy can't get any better, and the world is gonna fall into chaos. Well, that sounds a bit exaggerating, but that's possible. Now let's talk about the vaccines. COVID-19 vaccines are produced by many pharma companies. Now, how some of these compare? Oxford Uni AstraZeneca from United Kingdom, a collaboration between Oxford University and AstraZeneca Pharmaceutical Company, with type of vaccines, viral vector vaccines. Viral vector vaccines use a modified of different virus, or the vector, to deliver important instructions to our cells and then use the cell's machinery to produce a harmless piece of spike proteins of the virus. And then our immune system recognizes it doesn't belong there and then produce an antibodies to fight. All of these vaccines that I'm gonna mention need 2 doses of injection to complete the vaccination, and days between doses of Oxford Uni AstraZeneca vaccine is 28 days. There is some news lately stated that someone still get COVID after the injection. Well, this person only get the first injection. He needs to get the second one so he can get completely vaccinated. So you need to finish the whole procedure to be completely vaccinated. The efficacy of this vaccine is 62 to 90%, and the storage temperature of this vaccine is in regular fridge temperature, and the cost of each vaccine is about $4. Sputnik 5 vaccine from Russia, also using viral vector type of vaccine and needs 21 days between 2 doses, with 91.4% efficacy, storage in regular fridge temperature, with the price about $10 each. The Pfizer BioNTech vaccine, a collaboration between American and German pharma companies with mRNA type of vaccine. mRNA vaccines give instruction to our cells to make a harmless spike proteins of the virus, and then the spike protein will triggers the immune system to make antibodies to fight against the real virus. This vaccine needs 28 days between the 2 doses with 95% efficacy, storage in freezing minus 70 degrees Celsius with price about $20 each. And then next, Moderna vaccine from the US is also an mRNA type of vaccine that needs 28 days between the 2 doses with 95% efficacy, storage in freezing minus 20 degrees Celsius, costs about $25 to $37 each. The last one is Sinovac vaccine that currently being used in Indonesia. Vaccine from China. This is an inactivated virus type of vaccine, meaning the vaccine is using killed viral particles to expose the body's immune system to the virus without risking a serious disease response. This is more like a traditional method type of vaccines. This vaccine needs 14 days between the 2 doses with 65.3% efficacy that tested in Indonesia, storage in regular fridge temperature, and costs about $14 to $30 each. Now let's talk about AEFI. What is AEFI? AEFI, or an adverse event following immunization, or in Indonesia is called kejadian ikutan pasca imunisasi, is any untoward medical occurrence that follows immunization and which doesn't necessarily have a connection with the usage of the vaccine. The event might be unfavorable or unintended signs, abnormalities, symptoms, or disease, and we should aware of this. Any further info about this you can check on WHO webpage. In conclusion, vaccination is likely the only way we can do to stop this pandemic as soon as possible. There is nothing we should worry about the vaccines. So we should be more educated and trust the experts and keep following the guidance of medical experts for the sake of human lives. That's all from me. Thank you very much for listening. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Zulfikar, for the nice presentation. Now let's move to the Q&A session. There is question from Hansen in Universitas Surabaya. The question is, why does our country use the Sinovac vaccine rather than the other vaccine that show more efficacy than Sinovac? Okay, please, Zulfikar, you can answer the question.
Okay, thank you very much for the question. Okay, I want to answer the question. that our government are having already have a relationship or commitment with the Chinese companies and also Chinese government prior to the vaccination program, and also that the Sinovac vaccine is one of the vaccines that already distributed in the world and the one that fastest, the one that has already finished the trial before the other vaccine. So our government uh, apparently they want to order the earliest vaccines they already finished, and also that the efficacy that I mentioned on the poster is 62.5% efficacy is the one that tested in Indonesia. The Sinovac vaccines also distributed to Turkey, and they tested that they have a 95 or 91 point something percent efficacy. So the efficacy of 62.5% efficacy is the one that tested in Indonesia. So not all Sinovac vaccines is tested with that presentation of efficacy, but also have another range of efficacy, like the Oxford Uni AstraZeneca vaccines. They have a range of efficacy from 60% to 90% efficacy. So you don't have to worry about that. Actually, that these vaccines are completely safe and useful and helpful to prevent ourselves from the coronavirus or COVID-19 infection or diseases. Thank you.
Okay, thank you, Zulfikar, for the nice answer. And from Hansen, hope you understand the answer with well. Now let's move to the next participant, the last one in this poster presentation series with number PP54 from Universitas Islam Indonesia with name Belva Abrar Irawan. Let's take a look at the video.
Assalamualaikum warahmatullahi wabarakatuh. Hi, my name is Belva Abradirawan and I'm from Islamic University of Indonesia. In this occasion, I want to present my posters. It's about the right way to maintain heart health. So, we all know that heart is a very important organ. The heart pumps the blood to meet the needs of oxygen and nutrients for our body tissues. Therefore, we must keep the heart healthy and make sure the heart able to do its jobs optimally. So, today what I am going to share with you is several ways that can be done to keep our heart in the healthy condition. Firstly, checking the heart regularly. Even though we as a person really look healthy in the outside, but actually we don't know how exactly the condition of our inner body. So we still have to check the condition of our heart, whether the heart rhythm, organs, or other parts are normal or not. And by checking regularly, we can also detect if there is a heart problems, so we can resolve it immediately and it doesn't reach a more severe level. Next, avoiding smoking. As we know, cigarettes contain many hazardous or chemical substances that give no advantages at all. So, and according to health data published by British Heart Foundation, smoking can make the chemicals contained in them stick to the artery. This condition clogs the arteries to carry blood to the heart and cause a heart attack. Also, consuming alcohol can make our heart weak and cause an irregular heartbeat activities. The American College of Sport Medicine, or ACSM, recommends doing physical activity for 150 minutes per week. But if we divide it into frequency, for example, 5 times a week, then we only need to spend at least 30 minutes of exercise every day. And by doing the form of simple activity or sport, it can increase our body metabolisms. So the fat in the body will be metabolized so it doesn't accumulate in our heart. And if overweight, lose weight. Why? Because people who are overweight
our need more blood supply, and this condition will force the heart to work harder, and this certainly will trigger the heart to become depressed on its duties. Then over time it can no longer function properly. And to make the heart work as much as it should, it is really necessary to lose weight in someone who is overweight. Check blood sugar and blood pressure. High sugar level and left uncontrolled can increase the risk of heart disease attack. The reason is excess glucose is flowing in the blood and can damage blood vessel and ultimately trigger a heart attack. The damage caused, among others, the accumulation of fat due to cholesterol is called atherosclerosis. And for the blood pressure, research results from the World Health Organization or WHO showed that nearly half of heart attack cases are caused by high blood pressure. Blood pressure that continues to increase in the long term will cause the formation of the crust or plaque, which can narrow the coronary artery. And the last is eat lots of vegetable and fruits. Besides avoiding and reducing unnecessary things from our body, we also need a booster for our health. And by consuming fruit and vegetable, the body will be healthy. Fruit and vegetable have antioxidant and high in fiber. Fruit and vegetable can lower the blood pressure as well, so they can prevent the heart disease. I think that's enough from me. Thank you for your attention. And wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Belva, for the presentation. Now let's move to the Q&A session. There is question from Gihat Antar from Universitas Bakti Kencana. The question is there any way to check our heart health without regularly going for a checkup in the hospital? And how do we consume fruits and vegetables without increasing our insulin level and increasing the risk of diabetes? Okay, Belva, you can answer the question.
Okay, first of all, I would like to say thank you for all this question, and now I would like to answer it. Is there any way to check our heart health without regularly going for a checkup in the hospital? And how do we consume fruits and vegetables without increasing our insulin level and increasing the risk of diabetes? So actually, we have to know the first sign or the sign of the unhealthy heart. So maybe we can first take a look for the condition of our heart. Uh, the signs of unhealthy heart is like aching in the shoulder and chest. I'm sorry. Puffy legs and feet, irregular heartbeat, shortness of food and fatigue. And I think if we have the signs or those signs, we can know that there is maybe something wrong to our heart condition. But I suggest that we actually have to make sure if there is something really serious in our heart, but we must go to the hospital. But we can know the heart condition from the signs that unhealthy heart first, then we can go to the hospital later. So consume vegetable and fruits. Actually, there is an application to check such as cholesterol or glucose level, so we can know that how much or how is the concentration of glucose or cholesterol that we consume. So I think that would be really helpful to us to know that the presentation of something or food or beverage that we consume it in the safety level or not. So that's, I think that is all of my question, all of my answer. Thank you for the question.
Okay, thank you, Belva, for the nice answer. And for Jihad, I hope the answer can be understood as well. Okay, we have arrived at the last series of events for this poster presentation this morning. I would like to thank all of the judges participants and attendees who have participated in this poster presentation from beginning to the end. Okay, that's all for our agenda in this International Pharma Talk and Competition, or IPTC 2021 today. I would like to remind all the participants and attendees that this afternoon at 1 p.m., there will be a talk show with the theme Sustainable Pharmacy Education at the time of COVID-19 and attended by speakers who are competent in their field. There are Professor Patrick Ball and Dr. Hannah Morrissey from Wolverhampton University in United Kingdom, Professor Muhammad Taher from International Islamic University Malaysia, and Mr. Saifuddin PhD from Universitas Islam Indonesia. So please stay tuned until the end. Before we move to the talk show season, there will be a lunch break. Please attend to the audience to rest from their place while enjoying the committee's video until 1:30 p.m. Before the talk show begins, there will be a quiz with prizes based on the video that has been played. We will be back again at 1:30 p.m. Okay, that's all for me. Thank you for the attention. Sorry if there are mistakes that are intentional or not. See you in the next session of IPTC Talk Show. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Hana.
Selamat datang di Fakultas Matematika dan Ilmu Pengetahuan Alam Universitas Islam Indonesia.
Fakultas Matematika dan Ilmu Pengetahuan Alam telah berkomitmen sejak 1995 dalam meningkatkan penyelenggaraan sistem pendidikan yang bermutu, menghasilkan riset yang berkualitas tinggi, melaksanakan pengabdian masyarakat dan dakwah Islamiyah melalui penerapan ilmu pengetahuan dan teknologi yang berlandaskan pada nilai keislaman. Jurusan-jurusan unggulan FMIPA UII akan memfasilitasi menjadi saintis muda yang berkualitas dan berakhlakul karimah. Jurusan Statistika, jurusan dengan satu program studi tertua dan pertama di FMIPA UII, program studi Statistika UII telah mencetak lulusan unggulan yang tersebar luas. Penerapan kurikulum berbasis enthusiastic statisticians serta didukung dengan sarana dan prasarana penunjang yang lengkap serta modern. Prodi Statistika UII berkomitmen untuk terus mencetak data analis unggulan yang mampu bersaing di dunia global. Jurusan Kimia, jurusan kimia dengan 4 program studi pendukung. Program studi kimia. Prodi Kimia UII telah terakreditasi internasional RSC dan terakreditasi unggul, terus berkomitmen untuk mencetak peneliti-peneliti muda yang akan terus berinovasi, menciptakan riset ilmiah yang berkualitas dan modern. Kegiatan technopreneurship yang terfokus pada pengembangan minyak atsiri menjadi salah satu kegiatan andalan Prodi Kimia UII untuk terus melakukan riset demi mencetak lulusan yang memiliki kemampuan entrepreneurship. Program Studi D3 Analisis Kimia. Sebagai satu-satunya program vokasi di FMIPA UII, Prodi D3 Analisis Kimia terus berkomitmen untuk mencetak lulusan yang terampil dalam penguasaan skill laboratorium. Dengan staf pengajar yang tersertifikasi kompetensi serta praktisi dunia kerja, Prodi D3 Analisis Kimia UII memiliki jejaring industri dan instansi multinasional yang luas. Dibekali dengan sertifikasi kompetensi BNSP, Prodi D3 Analisis Kimia UII menjamin lulusannya untuk dapat bersaing di dunia global. Program Studi Pendidikan Kimia. Prodi Pendidikan Kimia UII terus berkomitmen untuk menyiapkan mahasiswanya menjadi tenaga pendidik yang kompeten pada jenjang pendidikan menengah dan sarjana. Prospek kerja yang sangat luas meliputi trainer, educational manager, bahkan bimbel founder, menjadikan program studi pendidikan kimia UII sebagai pilihan yang tepat untuk mencetak tenaga kerja pendidikan yang kompeten di Indonesia. Program Magister Kimia UII. Sebagai penyelenggara pengelola pendidikan kelas dunia di bidang Magister Kimia, PSMK UII telah melakukan kerjasama dengan berbagai universitas internasional seperti Hokkaido University Japan, University Science Islam Malaysia, dan Curtin University Australia. Jurusan Farmasi dengan 2 program studi pendukung. Program Studi Farmasi. Prodi Farmasi UII terus berkomitmen untuk menyiapkan lulusannya menjadi sarjana farmasi dan calon apoteker yang siap berkarya di sektor farmasi klinis komunitas dan farmasi industri. Ditunjang dengan sarana dan prasarana yang mendukung serta modern, juga kerjasama dengan berbagai universitas kancah internasional, Prodi Farmasi UII siap mencetak lulusan terbaiknya. Praktik kerja profesi apoteker. PKPA UII menerapkan sistem pembelajaran berbasis kasus, memberikan ruang kepada mahasiswa untuk mempelajari kasus real dalam praktik kefarmasian di kalangan masyarakat. Berbekal soft skill komunikasi, berpikir analisis, dan juga kritis, membawa PKPA UII unggul di bidang promosi kesehatan di kalangan masyarakat. FMIPA UII dilengkapi dengan pelayanan akademik yang lengkap serta ramah sehingga memudahkan para mahasiswa untuk merealisasikan rencana studi yang dimilikinya. Didukung dengan fasilitas-fasilitas kampus yang lengkap serta modern, FMIPA UII menjamin untuk terus memberikan pelayanan penunjang kegiatan belajar mengajar yang terbaik bagi mahasiswa. Sebagai bagian dari Green Campus Universitas Islam Indonesia, FMIPA UII juga berkomitmen untuk turut mensukseskan program universitas dan memberikan kenyamanan bagi mahasiswa melalui keasrian lingkungan kampus. Wahai saintis muda Indonesia, selamat bergabung di Fakultas Matematika dan Ilmu Pengetahuan Alam Universitas Islam Indonesia.
Apa yang Anda ketahui tentang Universitas Islam Indonesia? Universitas Islam Indonesia atau UII adalah perguruan tinggi swasta nasional tertua di Indonesia. Para pendirinya adalah tokoh bangsa yang mengerti betul tentang pentingnya pendidikan bagi masa depan negeri ini. UII terus mendorong sivitas akademikanya untuk mengukir prestasi. Sebagai universitas yang terakreditasi A, UII memiliki beragam fakultas yang dapat menjadi pilihan para calon mahasiswa. Program studi farmasi merupakan salah satu program studi di UII yang sangat diminati oleh banyak calon mahasiswa. Terbukti bahwa setiap tahun jumlah calon mahasiswa yang mengikuti tes semakin meningkat. Nah, tahukah Anda apa itu farmasi? Banyak orang masih belum mengenal apa itu farmasi. Farmasi adalah salah satu cabang dari ilmu kesehatan yang berfokus pada dasar-dasar seluk-beluk mengenai obat-obatan. Jika Anda kuliah di program studi farmasi, Anda akan mempelajari ilmu untuk membuat sediaan farmasi yang baik seperti farmasetika, bioteknologi farmasi, teknologi sediaan padat, analisis sediaan farmasi, dan lain sebagainya. Tidak hanya itu, farmasi juga mempelajari ilmu yang berkaitan dengan tubuh manusia. Program studi farmasi UII berdiri pada tahun 1998 dan memperoleh sertifikat ISO 9001-2008 dari TÜV Rheinland, Jerman. Mahasiswa prodi farmasi berasal dari seluruh wilayah Indonesia yang tersebar di 34 provinsi. Untuk menunjang kegiatan belajar mengajar, program studi farmasi menyediakan fasilitas laboratorium yang sangat lengkap, seperti laboratorium teknologi farmasi, laboratorium farmasetika apotek simulasi, laboratorium mikrobiologi farmasi, biologi molekuler, laboratorium farmakologi, laboratorium biologi farmasi, laboratorium kimia farmasi, laboratorium unit teaching hospital, laboratorium praklinis, laboratorium pengujian obat, makanan, dan kosmetik (LPOMK), dan laboratorium instrumentasi terpadu. Seluruh laboratorium yang ada sudah dilengkapi dengan peralatan yang sangat memadai untuk menunjang perkuliahan, praktikum, dan penelitian. Laboratorium farmasi sudah tersertifikasi ISO 17025 dari Komite Akreditasi Nasional atau KAN. Program studi farmasi memiliki pusat studi yang menunjang aktivitas penelitian, antara lain Pusat Studi Obat Herbal, Pelayanan Informasi Obat dan Nanofarmasi Research Center. Proses pembelajaran yang dilakukan baik di dalam kelas maupun proses praktikum dengan menggunakan student center learning antara lain problem based learning, project based learning, collaboration learning, dan metode lainnya yang disesuaikan dengan muatan keilmuan yang ada di dalamnya. Proses pembelajaran secara aktif dilakukan agar mahasiswa terlibat aktif dalam proses pembelajaran secara mandiri, mampu menyelesaikan setiap permasalahan, mampu menyelesaikan tugas dengan baik, dan memiliki soft skill yang unggul sehingga mampu bersaing di kancah global. Bagi kamu yang ingin kuliah dengan beasiswa, tidak perlu khawatir. UII memberikan beasiswa bagi mahasiswa baik dari internal maupun eksternal UII. Lulusan dari program studi farmasi ini biasanya kemudian disebut dengan sarjana farmasi dan kemudian melanjutkan ke jenjang profesi apoteker agar dapat bekerja dan berpraktik sebagai apoteker atau farmasis. Data Badan Pusat Statistik menunjukkan bahwa Indonesia masih kekurangan tenaga kerja dari sarjana sains, khususnya farmasi, yang berarti jumlah permintaan sarjana farmasi lebih besar dari lulusan yang dihasilkan. Contoh beberapa tempat yang memerlukan tenaga farmasi antara lain bidang pemerintahan, terutamanya di Departemen Kesehatan, Dinas Kesehatan, Puskesmas, dan pada Badan Pengawas Obat dan Makanan. Bagian administrasi pelayanan obat dan instansi pemerintahan, Polri, maupun TNI. Menjadi dosen pada Departemen Pendidikan Nasional sebagai dosen di bidang farmasi. Bagian apotek atau instalasi farmasi pada berbagai rumah sakit, pada bidang industri obat-obatan, pada bidang industri kosmetik, pada bidang industri makanan, pada bidang jamu dan obat tradisional, pada bidang industri dan perdagangan yang menjadi penjamin mutu, standar keamanan, dan kualitas produk makanan seperti obat-obatan maupun kosmetik. Bagaimana? Menarik bukan? Ayo, berkuliah di program studi farmasi UII.
JTEM 2020 merupakan salah satu implementasi jurusan farmasi Universitas Islam Indonesia dan University of Rhode Island, Amerika. Kami memperluas kerjasama dengan Politeknik Kesehatan Yogyakarta. Tahun 2020 ini mengambil tema Global Health Indonesia. Kegiatan JTEM 2020, khususnya di Yogyakarta, banyak membahas tentang masalah kesehatan, khususnya penyakit tidak menular. Gesakja series terdiri dari 2 seri, yaitu seri pertama mengenai penyakit tidak menular, yaitu berkaitan dengan penyakit diabetes, stroke, hipertensi, dan kemudian tentang bahaya merokok. Student interprofessional work. Mereka dibuat dalam grup-grup untuk berkelompok untuk berdiskusi tentang apa itu stroke, apa itu obesitas, apa itu diabetes, bagaimana penanganannya, bagaimana kalau di Amerika, bagaimana kalau di Indonesia, sehingga mereka saling bertukar pengalaman. kami bekerja sama dengan 4 puskesmas yang ada di Sleman, Yogyakarta. Nah, kemudian setelah itu mereka dari hasil diskusi itu mereka membuat presentasi dan hasil tersebut dipresentasikan ke masyarakat. Presentasi ke masyarakat merupakan salah satu bentuk bagaimana mereka menyampaikan promosi kesehatan. Dan pengabdian masyarakat dilakukan di Dusun Pelem bekerja sama dengan Afkaruna Islamic School. Di lokasi yang seperti Anda lihat di sini, lokasi yang sangat ideal dan mereka sangat antusias sekali untuk melakukan kegiatan ini.
My name is Matt Burton. I am an assistant professor in biomedical and pharmaceutical sciences at the University of Rhode Island. The goal of the J-term is to see every level of the Indonesian healthcare system, from community outreach in villages to the puskesmas, community health centers, and even a hospital. The students have been able to visit each of those. They have given outreach presentations with respect to diabetes, smoking cessation, stroke, hypertension, and obesity.
Nama saya Kania Ratna Rimbi, saya dari Politeknik Kesehatan, Kementerian Kesehatan, Yogyakarta.
Hi, my name is Luisa. I am from the University of Rhode Island. I study public health and nutrition.
Hi, I'm Katie. I'm a junior, double majoring in cellular and molecular biology and French.
Nama saya Bagas Trianloka. Saya mahasiswa farmasi Universitas Islam Indonesia.
Hi, my name is Kelsey. I'm a nursing major and I'm minoring in nutrition.
Kesan dan pesan saya untuk Global Health J-term 2020 adalah yang pertama, sangat menarik sekali. Yang kedua, selain dapat menambah pengalaman, saya juga dapat bertemu dengan teman-teman baru baik dari dalam negeri maupun dari luar negeri, terutama untuk Amerika Serikat, yaitu University of Rhode Island.
I have learned so much while being here. One of the things that has impressed me the most is just comparing nutrition here to the United States.
I'm very excited to be here in Java with all of these amazing people. Today we worked in the little village, checking people's blood pressures, and it was absolutely an amazing experience. Everyone is so welcoming and so warm.
Jadi dari 5 hari acara ini dapat saya katakan bahwa saya belajar tentang banyak materi-materi penyakit mulai dari stroke, hipertensi, diabetes, obesitas, dan merokok. Kemarin juga kita telah mengunjungi puskesmas. Di sana kita memberikan penyuluhan kepada masyarakat tentang pentingnya untuk menjaga kesehatan.
I love doing nursing work. I love giving blood pressures today. The hospitals were really nice. I was really impressed by the equipment that you guys use. You're very updated. I just am blown away with the hard work and the type of care they want to provide out here.
To me, what I have learned the most, yes, I also love all the people. They have received us with open arms, and we couldn't be any more thankful for their hospitality.
And you know, I just am really grateful to be here, and I love the children here, everyone here really. Everyone's just so nice and so kind, and I think what I'm going to take back to the US is just to spread that peace and joy and to be grateful for everything.
Assalamualaikum teman-teman, selamat datang di Universitas Islam Indonesia. Mau tahu lebih jauh tentang UII? Yuk ikut aku jelajah UII melalui UII Journey.
Udah pada tahu belum kalau UII merupakan perguruan tinggi nasional pertama di Indonesia? UII berdiri sejak 8 Juli 1945. Saat ini, UII mendapatkan peringkat A untuk akreditasi institusi BAN PT. UII juga masuk dalam jajaran 500 besar perguruan tinggi Asia menurut QS Asia University Rankings 2019. Sivitas Akademika UII memiliki kinerja level global, dibuktikan dengan diraihnya 5 bintang dalam kategori Employability QS World University Ranking 2016 and 2019. Untuk fasilitas kampus, kamu nggak perlu khawatir. UII punya fasilitas kampus kelas dunia. Dibuktikan dengan 5 bintang dalam kategori Facilities QS World University Rankings 2016 till 2019. Selain itu, UII juga dinobatkan sebagai PTS paling lestari di Indonesia loh. Dibuktikan dalam Green Metric World University Rankings 2018 dan Green Award dari Latofi School of CSR 2012, 2014, and 2016. Terus UI juga punya fasilitas namanya UII Print. Kamu bisa ngeprint, scan, dan fotokopi sesuai dengan kebutuhan perkuliahan kamu. Caranya gampang kok, kamu tinggal masukin nomor induk mahasiswa dan masukin deh password kamu. Gampang kan? Yang gak kalah penting, UII termasuk perguruan tinggi yang mengaplikasikan akses ramah difabel. Aku bantu ya. Nah, gimana? Seru kan jalan-jalan di UII? Eits, tunggu dulu. Masih banyak sobat aku yang nungguin kamu di fakultasnya masing-masing. Mau berkunjung di fakultas yang mana?
Suatu kebanggaan menjadi bagian dari keluarga besar Universitas Islam Indonesia. Kampus perjuangan yang didirikan oleh para tokoh-tokoh pendiri bangsa pada tahun 1945. Suatu kehormatan bagi kami menjadi bagian dari cita-cita para pendiri bangsa untuk mencetak tunas-tunas bangsa yang dapat menempatkan Indonesia pada kemajuan dan kejayaan. Menggabungkan pengetahuan dengan nilai-nilai keislaman dengan akhlak yang mulia. Kami berusaha mewujudkan keunggulan di bidang pengetahuan dengan melakukan penelitian yang dapat meningkatkan kualitas pembelajaran demi terciptanya Universitas Islam Indonesia sebagai world-class university yang unggul dalam bidang pendidikan dan pengajaran. Dedikasi kami adalah tumbuh dan berkembang bersama dalam harmoni yang selaras dengan visi misi dari cita-cita para pendiri bangsa. Kami wujudkan cita-cita keunggulan dalam bidang penelitian dengan mendirikan Nanofarmasi Research Center. Ini adalah langkah kecil kami agar dapat melompat lebih jauh dan lebih tinggi dalam penelitian di bidang farmasi. Karena penelitian merupakan salah satu denyut nadi dan merupakan roh perguruan tinggi. Ada banyak hal yang memberikan dorongan lebih bagi kami sebagai seorang pengajar. Mendorong mahasiswa agar memahami ilmu pengetahuan tidak hanya suatu rangkaian kata di dalam buku, hadir di ruang-ruang kelas dan jurnal. Pengetahuan ada di sekitar mereka. Mahasiswa dapat mengembangkan ilmu pengetahuan melalui proses berpikir yang mendalam dan kritis dan sesuai dengan tradisi luhur budaya akademik, di mana keberagaman pemikiran mendapatkan penghargaan yang tinggi demi menghasilkan generasi yang inovatif, unggul, dan menebar kebaikan. Mahasiswa merupakan tunas-tunas bangsa yang kami siapkan agar dapat berkontribusi pada kemajuan bangsa, salah satunya dalam bidang penelitian. Kami sadar keteladanan merupakan modal utama dalam menghasilkan generasi penerus bangsa yang unggul dan inovatif. Kami libatkan dan bimbing mahasiswa untuk berperan aktif dalam bidang penelitian, terutama dalam bidang nanofarmasi, sehingga mereka memiliki wawasan global agar mampu memberikan kontribusi bagi dunia, sejalan dengan komitmen Universitas Islam Indonesia untuk menghasilkan lulusan yang berkarakter unggul dan berwawasan global.
My project involved looking at glycoside and changing its drug delivery system to a self-nano-emulsifying drug delivery system. This increases the solubility of the drug and also the bioavailability. So it's something that was really interesting and something that I've always wanted to work on. Yeah, my team worked really hard for me, so we were able to do my project. Hani, Sirin, Asnia and Mr. Bambang. Yeah, I couldn't imagine doing my exchange experience anywhere else other than UAE, and I really hope that one day I can come back and maybe do a different project because I loved it here.
Kami sadar ilmu akan terus tumbuh saat dilandasi dengan semangat dan dorongan untuk terus belajar, meningkatkan keterampilan, berpikiran terbuka, dan memiliki wawasan yang luas. Akhirnya ilmu akan sempurna saat kita memberikan manfaat bagi sesama. Inilah yang kami lakukan di sini, menempa diri di bidang penelitian agar dapat memberikan manfaat bagi sesama dan mengambil peran demi kemajuan bangsa. Dengan nanofarmasi, kami berusaha wujudkan cita-cita pendiri bangsa yang telah melahirkan UII. Semangat dan inspirasi perjuangan mereka selalu hadir menjadikan kami selalu kuat melewati setiap tantangan yang ada. Kami mulai segalanya memberikan apa yang kami miliki untuk kemajuan bangsa Indonesia.
Selamat datang di Fakultas Matematika dan Ilmu Pengetahuan Alam Universitas Islam Indonesia.
Fakultas Matematika dan Ilmu Pengetahuan Alam telah berkomitmen sejak 1995 dalam meningkatkan penyelenggaraan sistem pendidikan yang bermutu, menghasilkan riset yang berkualitas tinggi, melaksanakan pengabdian masyarakat dan dakwah Islamiyah melalui penerapan ilmu pengetahuan dan teknologi yang berlandaskan pada nilai keislaman. Jurusan-jurusan unggulan FMIPA UII akan memfasilitasi menjadi saintis muda yang berkualitas dan berakhlakul karimah. Jurusan Statistika, jurusan dengan satu program studi tertua dan pertama di FMIPA UII, program studi Statistika UII telah mencetak lulusan unggulan yang tersebar luas. Penerapan kurikulum berbasis enthusiastic statisticians serta didukung dengan sarana dan prasarana penunjang yang lengkap serta modern. Prodi Statistika UII berkomitmen untuk terus mencetak data analis unggulan yang mampu bersaing di dunia global. Jurusan Kimia, jurusan kimia dengan 4 program studi pendukung. Program studi kimia. Prodi Kimia UII telah terakreditasi internasional RSC dan terakreditasi unggul, terus berkomitmen untuk mencetak peneliti-peneliti muda yang akan terus berinovasi, menciptakan riset ilmiah yang berkualitas dan modern. Kegiatan technopreneurship yang terfokus pada pengembangan minyak atsiri menjadi salah satu kegiatan andalan Prodi Kimia UII untuk terus melakukan riset demi mencetak lulusan yang memiliki kemampuan entrepreneurship. Program studi D3 Analisis Kimia. Sebagai satu-satunya program vokasi di FMIPA UII, Prodi D3 Analisis Kimia terus berkomitmen untuk mencetak lulusan yang terampil dalam penguasaan skill laboratorium. Dengan staf pengajar yang tersertifikasi kompetensi serta praktisi dunia kerja, Prodi D3 Analisis Kimia UII memiliki jejaring industri dan instansi multinasional yang luas. Dibekali dengan sertifikasi kompetensi BNSP, Prodi D3 Analisis Kimia UII menjamin lulusannya untuk dapat bersaing di dunia global. Program studi Pendidikan Kimia. Prodi Pendidikan Kimia UII terus berkomitmen untuk menyiapkan mahasiswanya menjadi tenaga pendidik yang kompeten pada jenjang pendidikan menengah dan sarjana. Prospek kerja yang sangat luas meliputi trainer, educational manager, bahkan bimbel founder, menjadikan program studi pendidikan kimia UII sebagai pilihan yang tepat untuk mencetak tenaga kerja pendidikan yang kompeten di Indonesia. Program Magister Kimia UII. Sebagai penyelenggara pengelola pendidikan kelas dunia di bidang Magister Kimia, PSMK UII telah melakukan kerjasama dengan berbagai universitas internasional seperti Hokkaido University Japan, University Science Islam Malaysia, dan Curtin University Australia. Jurusan Farmasi dengan 2 program studi pendukung. Program studi Farmasi. Prodi Farmasi UII terus berkomitmen untuk menyiapkan lulusannya menjadi sarjana farmasi dan calon apoteker yang siap berkarya di sektor farmasi klinis komunitas dan farmasi industri. ditunjang dengan sarana dan prasarana yang mendukung serta modern, juga kerjasama dengan berbagai universitas kancah internasional. Prodi Farmasi UII siap mencetak lulusan terbaiknya. Praktik kerja profesi apoteker. PKPA UII menerapkan sistem pembelajaran berbasis kasus, memberikan ruang kepada mahasiswa untuk mempelajari kasus real dalam praktik kefarmasian di kalangan masyarakat. Berbekal soft skill komunikasi, berpikir analisis, dan juga kritis, membawa PKPA UII unggul di bidang promosi kesehatan di kalangan masyarakat. FMIPA UII dilengkapi dengan pelayanan akademik yang lengkap serta ramah sehingga memudahkan para mahasiswa untuk merealisasikan rencana studi yang dimilikinya. Didukung dengan fasilitas-fasilitas kampus yang lengkap serta modern, FMIPA UII menjamin untuk terus memberikan pelayanan penunjang kegiatan belajar mengajar yang terbaik bagi mahasiswa. Sebagai bagian dari Green Campus Universitas Islam Indonesia, FMIPA UII juga berkomitmen untuk turut mensukseskan program universitas dan memberikan kenyamanan bagi mahasiswa melalui keasrian lingkungan kampus. Wahai saintis muda Indonesia, selamat bergabung di Fakultas Matematika dan Ilmu Pengetahuan Alam Universitas Islam Indonesia.
Apa yang Anda ketahui tentang Universitas Islam Indonesia? Universitas Islam Indonesia atau UII adalah perguruan tinggi swasta nasional tertua di Indonesia. Para pendirinya adalah tokoh bangsa yang mengerti betul tentang pentingnya pendidikan bagi masa depan negeri ini. UII terus mendorong sivitas akademikanya untuk mengukir prestasi. Sebagai universitas yang terakreditasi A, UII memiliki beragam fakultas yang dapat menjadi pilihan para calon mahasiswa. Program studi farmasi merupakan salah satu program studi di UII yang sangat diminati oleh banyak calon mahasiswa. Terbukti bahwa setiap tahun jumlah calon mahasiswa yang mengikuti tes semakin meningkat. Nah, tahukah Anda apa itu farmasi? Banyak orang masih belum mengenal apa itu farmasi. Farmasi adalah salah satu cabang dari ilmu kesehatan yang berfokus pada dasar-dasar seluk-beluk mengenai obat-obatan. Jika Anda kuliah di program studi farmasi, Anda akan mempelajari ilmu untuk membuat sediaan farmasi yang baik seperti farmasetika, bioteknologi farmasi, teknologi sediaan padat, analisis sediaan farmasi, dan lain sebagainya. Tidak hanya itu, farmasi juga mempelajari ilmu yang berkaitan dengan tubuh manusia. Program studi farmasi UII berdiri pada tahun 1998 dan memperoleh sertifikat ISO 9001-2008 dari TÜV Rheinland, Jerman. Mahasiswa prodi farmasi berasal dari seluruh wilayah Indonesia yang tersebar di 34 provinsi. Untuk menunjang kegiatan belajar mengajar, program studi farmasi menyediakan fasilitas laboratorium yang sangat lengkap, seperti laboratorium teknologi farmasi, Laboratorium Farmasetika Apotek Simulasi, Laboratorium Mikrobiologi Farmasi, Biologi Molekuler, Laboratorium Farmakologi, Laboratorium Biologi Farmasi, Laboratorium Kimia Farmasi, Laboratorium Unit Teaching Hospital, Laboratorium Praklinis, Laboratorium Pengujian Obat, Makanan, dan Kosmetik, LPOMK, dan Laboratorium Instrumentasi Terpadu. Seluruh laboratorium yang ada sudah dilengkapi dengan peralatan yang sangat memadai untuk menunjang perkuliahan, praktikum, dan penelitian. Laboratorium farmasi sudah tersertifikasi ISO 17025 dari Komite Akreditasi Nasional atau KAN. Program studi farmasi memiliki pusat studi yang menunjang aktivitas penelitian, antara lain Pusat Studi Obat Herbal, Pelayanan Informasi Obat, dan Nanofarmasi Research Center. Proses pembelajaran yang dilakukan baik di dalam kelas maupun proses praktikum dengan menggunakan student-centered learning, antara lain problem-based learning, project-based learning, collaboration learning, dan metode lainnya yang disesuaikan dengan muatan keilmuan yang ada di dalamnya. Proses pembelajaran secara aktif dilakukan agar mahasiswa terlibat aktif dalam proses pembelajaran secara mandiri, mampu menyelesaikan setiap permasalahan, mampu menyelesaikan tugas dengan baik, dan memiliki soft skill yang unggul sehingga mampu bersaing di kancah global. Bagi kamu yang ingin kuliah dengan beasiswa, tidak perlu khawatir. UII memberikan beasiswa bagi mahasiswa baik dari internal maupun eksternal UII. Lulusan dari program studi farmasi ini biasanya kemudian disebut dengan sarjana farmasi dan kemudian melanjutkan ke jenjang profesi apoteker agar dapat bekerja dan berpraktik sebagai apoteker atau farmasis. Data Badan Pusat Statistik menunjukkan bahwa Indonesia masih kekurangan tenaga kerja dari sarjana sains, khususnya farmasi, yang berarti jumlah permintaan sarjana farmasi lebih besar dari lulusan yang dihasilkan. Contoh beberapa tempat yang memerlukan tenaga farmasi antara lain bidang pemerintahan, terutamanya di Departemen Kesehatan, Dinas Kesehatan, Puskesmas, dan pada Badan Pengawas Obat dan Makanan. Bagian administrasi pelayanan obat dan instansi pemerintahan, Polri, maupun TNI. Menjadi dosen pada Departemen Pendidikan Nasional sebagai dosen di bidang farmasi. Bagian apotek atau instalasi farmasi pada berbagai rumah sakit, pada bidang industri obat-obatan, pada bidang industri kosmetik, pada bidang industri makanan, pada bidang jamu dan obat tradisional, pada bidang industri dan perdagangan yang menjadi penjamin mutu, standar keamanan, dan kualitas produk makanan seperti obat-obatan maupun kosmetik. Bagaimana? Menarik bukan? Ayo, berkuliah di program studi farmasi UII.
JTEM 2020 merupakan salah satu implementasi jurusan farmasi Universitas Islam Indonesia dan University of Rhode Island, Amerika. Kami memperluas kerjasama dengan Politeknik Kesehatan Yogyakarta. Tahun 2020 ini mengambil tema Global Health Indonesia. Kegiatan JTEM 2020, khususnya di Yogyakarta, banyak membahas tentang masalah kesehatan, khususnya penyakit tidak menular. Lecture series terdiri dari 2 seri, yaitu seri pertama mengenai penyakit tidak menular, yaitu berkaitan dengan penyakit diabetes, stroke, hipertensi, dan kemudian tentang bahaya merokok. Student interprofessional work. Mereka dibuat dalam grup-grup untuk berkelompok, untuk berdiskusi tentang apa itu stroke, apa itu obesitas, apa itu diabetes, bagaimana penanganannya, bagaimana kalau di Amerika, bagaimana kalau di Indonesia, sehingga mereka saling bertukar pengalaman. Kami bekerja sama dengan 4 puskesmas yang ada di Sleman, Yogyakarta. Nah, kemudian setelah itu mereka dari hasil diskusi itu mereka membuat presentasi dan hasil tersebut dipresentasikan ke masyarakat. Presentasi ke masyarakat merupakan salah satu bentuk bagaimana mereka menyampaikan promosi kesehatan. Dan pengabdian masyarakat dilakukan di Dusun Pelem bekerja sama dengan Afkaruna Islamic School. Di lokasi yang seperti Anda lihat di sini, lokasi yang sangat ideal dan mereka sangat antusias sekali untuk melakukan kegiatan ini.
My name is Matt Burton. I am an assistant professor in biomedical and pharmaceutical sciences at the University of Rhode Island. The goal of the J term is to see every level of the Indonesian healthcare system, from community outreach in villages to the puskesmas, community health centers, and even a hospital. The students have been able to visit each of those. They have given outreach presentations with respect to diabetes, smoking cessation, stroke, hypertension, and obesity.
Nama saya Kania Ratna Rimbi, saya dari Politeknik Kesehatan Kementerian Kesehatan Yogyakarta.
Hi, my name is Luisa. I am from the University of Rhode Island. I study public health and nutrition.
Hi, I'm Katie. I'm a junior, double majoring in cellular and molecular biology and French.
Nama saya Bagas Trianloka. Saya mahasiswa Farmasi Universitas Islam Indonesia.
Hi, my name is Kelsey. I'm a nursing major and I'm minoring in nutrition.
Kesan dan pesan saya untuk Global Health J Term 2020 adalah yang pertama sangat menarik sekali. Yang kedua, selain dapat menambah pengalaman, saya juga dapat bertemu dengan teman-teman baru baik dari dalam negeri maupun dari luar negeri, terutama untuk Amerika Serikat, yaitu University of Rhode Island.
I have learned so much while being here. One of the things that has impressed me the most is just comparing nutrition here to the United States.
I'm very excited to be here in Java with all of these amazing people. Today we worked in the little village, checking people's blood pressures, and it was absolutely an amazing experience. Everyone is so welcoming and so warm.
Jadi dari 5 hari acara ini dapat saya katakan bahwa saya belajar tentang banyak materi-materi penyakit, mulai dari stroke, hipertensi, diabetes, obesitas, dan merokok. Kemarin juga kita telah mengunjungi puskesmas. Di sana kita memberikan penyuluhan kepada masyarakat tentang pentingnya untuk menjaga kesehatan.
I love doing nursing work. I love giving blood pressures today. The hospitals were really nice. I was really impressed by the equipment that you guys use. You're very updated. I just am blown away with the hard work and the type of care they want to provide out here.
To me, what I have learned the most, yes, I also love all the people. They have received us with open arms, and we couldn't be any more thankful for their hospitality.
And you know, I just am really grateful to be here, and I love the children here, everyone here, really. Everyone's just so nice and so kind, and I think what I'm going to take back to the U.S. is just to spread that peace and joy and to be grateful for everything.
Jurusan Farmasi Universitas Islam Indonesia memiliki 7 laboratorium. Di antaranya adalah Laboratorium Teknologi Farmasi, Laboratorium Mini Teaching Hospital, Laboratorium Mikrobiologi, Laboratorium Farmakologi, Laboratorium Kimia Farmasi, dan yang terakhir adalah Laboratorium Biologi Farmasi. Laboratorium Biologi Farmasi juga membekali mahasiswa-mahasiswi dengan kemampuan-kemampuan dalam penelitian bahan alam, yaitu dari proses ekstraksi hingga proses isolasi untuk menemukan senyawa-senyawa obat baru di masa depan. Sehingga diharapkan kedepannya mahasiswa-mahasiswa farmasi Universitas Islam Indonesia menjadi pionir-pionir di dalam penelitian bahan alam untuk menemukan senyawa-senyawa baru yang dapat digunakan sebagai obat di masa akan datang. Saat ini, Laboratorium Biologi Farmasi juga telah berhasil memproduksi standar-standar kimia yang digunakan dalam penelitian. Fasilitas-fasilitas yang dimiliki laboratorium di jurusan Farmasi Universitas Islam Indonesia telah terstandar dan terkalibrasi. Di antaranya adalah TLC scanner. Kemudian kami memiliki rotary evaporator dengan kualitas presisi yang bisa untuk menguapkan air. Selanjutnya, yang terakhir adalah River Phase HPLC yang kami gunakan sebagai isolasi bahan alam. Dari apa yang sudah kami lakukan, kami berharap di masa yang akan datang, kami dapat menjadi inspirasi di bidang penelitian, khususnya di dunia farmasi. jurusan Farmasi Universitas Islam Indonesia memiliki 7 laboratorium. Di antaranya adalah Laboratorium Teknologi Farmasi, Laboratorium Mini Teaching Hospital, Laboratorium Mikrobiologi, Laboratorium Farmakologi, Laboratorium Kimia Farmasi, dan yang terakhir adalah Laboratorium Biologi Farmasi. Laboratorium Biologi Farmasi juga membekali mahasiswa-mahasiswi dengan kemampuan-kemampuan dalam penelitian bahan alam, yaitu dari proses ekstraksi hingga proses isolasi untuk menemukan senyawa-senyawa obat baru di masa depan. Sehingga diharapkan kedepannya mahasiswa-mahasiswa Farmasi Universitas Islam Indonesia menjadi pionir-pionir di dalam penelitian bahan alam untuk menemukan senyawa-senyawa baru yang dapat digunakan sebagai obat di masa akan datang. Saat ini, Laboratorium Biologi Farmasi juga telah berhasil memproduksi standar-standar kimia yang digunakan dalam penelitian. Fasilitas-fasilitas yang dimiliki laboratorium di jurusan Farmasi Universitas Islam Indonesia telah terstandar dan terkalibrasi. Di antaranya adalah TLC scanner. Kemudian kami memiliki rotary evaporator dengan kualitas presisi yang bisa untuk menguapkan air. Selanjutnya, yang terakhir adalah River Press HPLC yang kami gunakan sebagai isolasi bahan alam. Dari apa yang sudah kami lakukan, kami berharap di masa yang akan datang, kami dapat menjadi inspirasi di bidang penelitian, khususnya di dunia farmasi.
Okay, good afternoon, guys. Welcome to join for those who just joined it, yeah. Okay, I will get ready. Okay. The time is yeah. Okay, guys, it's look like the video is finished. Now times for quiz. Previously, I will provide information first. So the material for the quiz will come from the video that was played just now. Yeah. So that's why it's lucky for you to watch it carefully. Yeah. Okay, because it will definitely be easy to answer the quiz, but it doesn't matter if you didn't watch it earlier and maybe later you can answer the quiz. It's no problem, yeah. And the quiz winners will also get the prize from the committee, of course. Just wait. Okay, for the quiz, we will use the Kahoot platform. Yeah. So for friends, please prepare your gadgets. You can use two devices or just one is enough. Don't forget to stay online Zoom meeting and activate the Kahoot because the code will be shared on the screen in Zoom room. Okay. Okay, don't forget to name Kahoot using your full name, you guys, and real name to make it easier for the committee to give rewards later. Come on, guys. This is code game. Okay, and the prizes is not bad, yeah. Okay, come on. Everyone can join without exception. Okay. Okay. Okay, come on. Oh, we are waiting for you guys. Go, go, go. Wow. Okay. Yeah. Using your full name, guys. Or real name is no problem. Okay. We are waiting for you. Okay, come on, come on, come on. Up, up, up. Next. Go, go. Okay, guys, we wait 2 more minutes. Yeah, go up. Come on, come on. Okay, stop at 89. Oh, 85, 84. Okay. Okay. Okay, 86. Mhm. Okay. It's like many have joined. Okay. Let us just start, Mrs. Yossi.
Okay, Sophie, we can
Okay.
start now.
Okay. Okay. Let's go, Mrs. Yossi. 3, 2, 1. Go. Wow. Time for quiz. When pharmacy department was founded. Go. Oh my god. Oh my god. Oh. Wow. Okay. Nice answer. Hmm. Wow. Okay. Okay. Yeah. Oh. Okay, next, Mrs. Yossi. Time is up. Oh my god. Okay. Nice answer. Wow. Okay, next, guys. Okay, good job. It's no problem, yeah. Okay. Oh, nice. Wow. Okay, next, Mrs. Okay, many have answered correctly.
Okay, right now, Vicky is on the top of the scoreboard. Okay, Sophie, looks like we got the winner.
Okay, Mrs. Oh my God. Wow, the game is quite interesting. Many have answered correctly, but some are wrong. No problem. It's just a game, but with prizes. Okay, time is over and the game is over. Amazing, yeah. Congratulations to the champions. In third position is Hadrami, yeah? Okay. And then in second position is Vicky Dahniar. Wow. And I'm sorry, first position? Mrs.?
Yes, we have 5 winners. Okay.
Okay.
Five
Oh,
winners.
yes, really, Ambria. Okay, for the first position. Okay, congratulations, guys. The prizes will be processed immediately from committee. Yeah. Wow, it's good. And good willing after the event will be sent immediately. Thank you for the taking of the quiz session. Yeah, and congratulations for the winner. Yeah, good.
Okay,
Okay,
so
thank you, Mrs.
maybe we
Yossi.
still wait for the fourth and the fifth winner for quiz, Sophie. Okay.
Why, mommy? I'm sorry.
We have 5 winners, but we already have 3 winners that you mentioned before, but we also
Okay.
have fourth and fifth winner.
Oh, okay, okay. Yeah, yeah, yeah. Okay.
Okay, sorry, Sophie, maybe we need more time to recap to just and show the winner.
Yes. Yeah, okay, mommy. I'm waiting for announcement.
The
Yeah.
fourth winner is,
Okay.
yes, oh, there are 5 winners. First is Yasirli,
Okay.
Fikih Daniar, Hadrami,
Yeah.
Vida, and Rianita.
Oh,
Congratulations.
okay. Right. Okay. Thanks, Mrs. Yossi.
Yes, thank you, Sophie. Time is yours. Yes, Sophie, you can continue the session.
Okay. Okay. Okay, Mrs. Oh. Okay, guys, attention please. Ladies and gentlemen, the opening of the talk show going to start in 2 minutes. Please prepare yourself as a participant who are encouraged to start activating your camera and using the virtual background, yeah, has been provided by the committee. Thank you for your attention. Okay, guys, that was the result of the quiz. It was written in the chat column, and the committee will inform you about the door prize, yeah? Yeah, okay. Thank you, Mrs. Mutiara Herawati. Bismillahirrahmanirrahim. The Honorable Professor Dr. Apotheker Yandi Shukri, as a head of Department of Pharmacy, Faculty of Mathematics and Natural Sciences, Universitas Islam Indonesia. The Honorable Mr. Saipuddin PhD, head of Study Program of Pharmacy, Faculty of Mathematics and Natural Sciences, Universitas Islam Indonesia. The Honorable Dr. Farida Hayati, Head of Pharmacist Program, Faculty of Mathematics and Natural Sciences, Universitas Islam Indonesia. Lecturer of Pharmacy Study Program, Faculty of Mathematics and Natural Sciences, Universitas Islam Indonesia. Chief of Drug Information Center, Pharmacy of Study Program, Faculty of Mathematics and Natural Sciences, Universitas Islam Indonesia. The Honorable all the speakers of IPTC 2021, Chief Executive and all participants of IPTC Talk 2021. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and welcome to the speakers and all participants in the International Pharma Talk and Competition 2021 held by the Universitas Islam Indonesia Drug Information Center. First of all, let us pray for gratitude for the presence of Allah Subhanahu wa Ta'ala who has given us health to that we can gather on this day. Not forgetting the blessing and greetings we give to our Lord the Prophet Muhammad Sallallahu Alaihi Wasallam, which has taken us from the time of ignorance to the bright times as it is today. Before we begin this event, let me introduce myself. My name is Binti Sofiatul Nadirah. You can call me Sophie. I'm currently studying at the Bachelor of Pharmacy Study Program Universitas Islam Indonesia. And this event will act as a master of ceremony. Well, here I will slightly explain brief introduction of IPTC. Yeah, IPTC is a program or event held by the Pharmacy Study Program of the Faculty of Mathematics and Natural Sciences in collaboration with the Drug Information Center, Universitas Islam Indonesia. This event was held in an online-based system, yeah, that can be followed by all of Indonesian students, and even the competition was finished this afternoon. Now we will continue this event with a talk show with Professor Patrick Abel and Dr. Hannah Morrissey from Wolverhampton University, United Kingdom, and Professor Muhammad Taher from International Islamic University of Malaysia, and Mr. Saipuddin PhD from Universitas Islam Indonesia. Please follow this event until the end because later we have many door prize for lucky participant, and there will also be an announcement of who becomes the winner of the oral and poster competition. So stay tuned, guys. Okay, the event is about to start. For participants who are not interested in talking, please turn off the microphone and do not forget to turn on the video and to connect the virtual background. Yeah. Okay, ladies and gentlemen, before we start, we will recite the Holy Quran delivered by Muhammad Al-Kamel. Kamel is a pharmacy student from an international program who comes from Yemen. Yeah, at UII Pharmacy, there is a class for overseas students. Okay, Muhammad Al-Kamel, time is yours.
Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم إن الذين سبقت لهم منا الحسنى أولئك عنها مبعدون لا يسمعون حسيسها وهم فيما اشتهت أنفسهم خالدون لا يحزنهم الفزع الأكبر وتتلقاهم الملائكة فتلقاهم الملائكة هذا يومكم الذي كنتم توعدون يوم نطوي السماء كطي السجل للكتب كما بدأنا أول خلق نعيده وعدا علينا إنا كنا فاعلين ولقد كتبنا في الزبور من بعد الذكر أن الأرض يرثها عبادي الصالحون إن في هذا لبلاغا لقوم عابدين وما أرسلناك إلا رحمة للعالمين صدق الله.
Sadaqallahul Azim. Thank you for your time. May those who read and listen get reward from Allah Subhanahu wa Ta'ala. Amin, amin, ya Rabbal Alamin. The next agenda is a report by the head of the Committee of International Pharma Talk and Competition that will be delivered by Kian Alfiati. Please welcome Kian. Time is yours.
Okay, Sophie. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
The Honorable Dr. Yandi Shukri, Saifuddin PhD, Dr. Farida Hayati, and Professor Patrick Ball with Dr. Hannah Morrissey from Wolverhampton University, and Professor Muhammad Taher from International Islamic University Malaysia, and we respect the lecturer who have helped us a lot. Also the participant, thank you for coming and in the International Pharma Talk and Competition 2021. My name is Kian and I am pharmacy student 2018 of Universitas Islam Indonesia. I am as the chairperson of International Pharma Talk and Competition 2021. I would like to say big thank you to all of committee and participants. Alhamdulillah, this competition has been attended by 57 poster participants and 41 oral participants with various pharmacy students and pharmacist professional students, as well as the total participants who took part in International Pharma Talk and Competition 2021. The following were graphic. The following were graphic participants who took part in International Pharma Talk and Competition 2021. All participants come from many universities, start from Medan to Papua, including universities Muhammadiyah such as Surakarta, Malang, Purwokerto, Kalimantan, then I also saw from Cendrawasih University in Papua. This enthusiasm is extraordinary. Hopefully our event can run smoothly. We ask the committee apologies if there are many mistakes and shortcomings during this event. Enjoy the program. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you to Kian Afiati as the head of the student committee for the report. Yeah. The next agenda, let us hear a speech and opening remark from a head of the Pharmacy Department of the Faculty of Mathematics and Natural Sciences, Universitas Islam Indonesia, to Professor Dr. Apotheker Yandi Shukri. Hello, Mr. Okay, time is yours.
Is it clear, my sound?
Yes, clear.
Okay.
Loud and clear, Prof. Yandi.
Okay, right. Okay. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
The honorable invited speaker, Professor Patrick Ball and Dr. Hannah Morrissey from Wolverhampton University, UK, Professor Muhammad Taher from International Islamic University, Malaysia, distinguished guests, ladies and gentlemen, and all participants. First of all, let us pray and thank God for the blessing and good health granted to us, as well as the opportunity to join this memorable event, the International Pharma Talk Competition, IPTC 2021. Thank you for joining us today in the IPTC 2021, jointly organized by the Department of Pharmacy, Universitas Islam Indonesia, and Drug Information Center. It's a great pleasure that is this event, a student of undergraduate and pharmacy professional programs have participated. The main theme of the IPTC 2021 is Sustainable Pharmacy Education at the time of COVID-19. These events aim to be developed to discuss pharmacy education globally during pandemic COVID-19 and sharing sessions and pharmacy-related issues through student competition. On behalf of the Department of Pharmacy and Organizing Committee, I'd like to welcome all of you to this event. We are delighted to have you here to participate and share your work and through oral and poster presentations. Sincere appreciation are expressed to Professor Patrick Ball, Dr. Hannah Morisse, and Professor Muhammad Taher, who willingly give lectures this afternoon. We also would like to thank all members of organizing committee for the good teamwork and the great effort in bringing for this event. This meeting brought together more than 200 student participants from various universities and institutions. The event begins with a poster and oral presentation that has been presented this morning for the top 10 presenters. For this afternoon, a lecture series will be delivered by invited speakers talking about pharmacy education around the world during COVID-19. We hope that this event will give a beneficial contribution to improving pharmacy education in the time of COVID-19. We sincerely hope that every participant will enjoy this event. Ladies and gentlemen, finally, by saying Bismillahirrahmanirrahim, I officially declare this International Pharma Talk Competition IPTC 2021 open. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you to Professor Dr. Apotheker Yandi Shukri for your speech. Yeah. Okay, guys, we will carry out a group photo session. Yeah, first before heading for the next event. Well, please, for the committee on duty to get ready, yeah, because there must a lot of slide on our device, yeah, and every device is not the same. So please cooperate with all the participants. Okay, before starting, I remind you again, don't forget to turn on the video and use the virtual background that has been given. Yeah, immediately I invite the committee in charge of documentation. Come on, committee.
Okay, the next event is photo session with participants. Participants are welcome to prepare for. I will count 1, 2, 3 to start taking pictures.
Okay, please smile. Okay, cheers again. Okay, smile, guys.
Okay, once more.
Okay, has the commit finished or done?
Enough. Yes. Hello, can you hear me?
Yes?
Okay, finish.
Okay, finish. Okay, done. I'm sorry. Okay, many thanks to Professor Yandi and also the speakers today, yeah, that we are willing to the document first. And thank you to also the participant. Okay, guys, before entering the talk show, I will remind all participants about attendance. So later, when the talk show is in progress, when the committee has sent a Google attendance form, yeah, please fill in immediately because that present will be used for the certificate process. Okay, so don't forget to fill it out. Well, now this is the session that we are waiting for. Here it is a talk show together with 4 great speakers. Today, for the talk show section, we will have Mr. Herman as a moderator. Mr. Herman Felani Tanjung, Master of Arts, is a manager of development international program of Universitas Islam Indonesia. Hello, Mr. Herman. How are you today?
Hello, Sophie. Life is good here. I hope everyone is doing fine.
Yeah.
Thank you for having me.
Why, Mr.?
Thank you for having me here.
Okay, yeah. You're welcome, Mr. Omet it always be healthy, yeah, because at this time, health is the most important, right?
Yes, that's why I'm sorry I don't use the background, the graphic background, because I'm on my holiday to stay healthy. You know, if you look at my background, I'm on the beach now. No.
Okay, Mr. It's no problem.
My computer is too old, so I cannot use the background. So that's the reason. I'm not on holiday. I'm trapped in my room, just like you.
Okay. Yeah. Okay, Mr. Okay, guys, for all the participants, please prepare your note and question, yeah, because in this section we'll get a lot of interesting information. Okay, Mr. Herman, time is yours.
Thank you very much, Sophie. Actually, Sophie or Hannah should be the moderator for this session because they make the situation really alive. You know, when first of all, I would like to confirm that I'm not a pharmacy lecturer. I'm just an English teacher. So it is really an honor to be here. You know, to meet you all. Okay, so I'll do the duty. Professor Dr. Safuddin just told me that actually just make sure that the students are not asleep. Okay. So my job is to keep you awake. Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah rabbil alamin. Assalatu wassalamu ala asrofil anbiya wal mursalin wa ala alihi washabihi ajmain. First of all, I would like to say good morning to Professor Patrick Ball. It's 7 in the morning, right? In UK now, right? Professor Patrick.
Yeah, that's correct.
And Dr. Hannah, how is Birmingham now? Is it still like a totally lockdown now?
The UK is still pretty much in total lockdown. It is also very, very cold here at the moment. We have very cold winds.
What's the temperature now, Professor Ball?
Currently -4 degrees centigrade.
Wow, but I don't see Dr. Hannah wearing any sweater or you're wearing any suit with you.
In the house.
Okay. Wow. Okay. We stay together. I think we're with you. Indonesia is also, we're in the warm season now. Actually, it's rainy season, but it's pretty warm here. Like us in Yogyakarta now, I think it's around 34 degree or something outside. Okay, so we're going to have you with the students today. It's really an honor. Like I said, I'm using a different virtual background because I feel like I need a holiday. Everyone needs holiday, but you and the students, they're all great because this supposed to be their weekend, but they choose to stay here to join the international Pharma talk in the competition. Okay. And I would like also to say hi to Professor Taher from IIUM. How is it in Kuantan, Professor Taher? Bye-bye.
Good, good. Thank you very much.
Glad to hear that. Okay,
Yes.
I think Malaysia is having like a significant increase in the number of COVID-19 now, but I think the situation is getting better, right? Is it
Yes,
better or
not really?
not really? Okay, Prof. Tahir, yeah, we would like to know. We need to stay happy, whatever the situation is. And I would like to say hello to Dr. Saifuddin too. Is Dr. Saifuddin here? Okay, okay. I'll tell you a little secret about Dr. Saifuddin here. Dr. Saifuddin is one of our favorite lecturer in pharmacy faculty, but his favorite lecturer is actually Professor Patrick Ball. You know, wherever Professor Patrick goes, he will like running after him. So like, Professor Patrick, you're in Darwin, okay, come to Jogja, and then you're in Wolverhampton, come to Jogja. But now, because of the pandemic, I think Professor Patrick cannot be kidnapped by Professor Saifuddin. Dr. Saifuddin, maybe next time when the situation is all getting better, you can take Dr. Hana and also Professor Taher join us in our lovely campus in Yogyakarta. Okay, and I would like to say hello to all of the students too. I want to know, can you guys make some noise to make sure that you are all still alive, from Papua? I see students from Papua here? Or eastern part of Indonesia, Papua, Maluku, we have NTT. Anybody?
I am in Papua, sir.
Oh, Kian. Oh, you're in Papua. Okay, which part of university are you, Kian? The committee, yeah?
In Jayapura, sir.
Jayapura. Okay. Papua now is 5 p.m. in Papua now, right? I see. Okay, even if we are in the same country, but we have significant time differences. Okay, so the topic this afternoon, I think because I have told you that I'm not a pharmacy lecturer or pharmacist or a clinical pharmacist like Dr. Hannah. So as an English teacher, I say I'm really impressed with what you have been doing this morning, with the presentations. I saw even some Indonesian students already look like a native speaker while using their English. Like I saw like Gihad. Gihad, are you here, Gihad? Very impressive
Yes,
pronunciation. Your English is amazing. This is my opinion as an English teacher. And then Ilya
so much.
and Zarofin, Zulfa, so many great students. And I think I'm so impressed that during the pandemics, the difficult times for you to be a millennial and a students, you guys are still performing very well by seeing what you've been doing with your presentations, with oral presentation, and also poster presentation. And it's quite unique. You normally poster presentation, you don't have to speak to explain your poster. This poster speak for itself, but you also present your poster. That is amazing. So okay, the topic for our discussions, for our talk, because it's a talk, it should be, I don't know, it should be tea time maybe in the afternoon, but I don't know, Dr. Hannah in UK, what time do you normally have tea time? Is there tea time in the morning at 7 a.m.?
Coffee time.
Coffee time. Oh, coffee time.
I
Okay.
just finished mine.
I see. So the good thing of pandemic, the good thing, of course, this is something that we don't like, but the good thing is when you're joining the class during the pandemics, I think first, you don't need to have fancy clothes, right? For some of the students who are wearing hijab, you just need to have one good hijab.
Slippers is the key.
Slippers
Wearing
is the key.
slippers.
Yes, Dr. Hannah, and sometimes some of us, we are teaching using sarong, like Dr. Saifuddin is probably not wearing like pants and suit, but maybe it's just a batik and a sarong. Yeah, Pak Taher, yeah.
Yes.
I think you don't need to have to look, some of you, I think Kian normally spend like 30 minutes preparing or maybe 1 hour before going to campus in the normal time now, right? For makeup, dressing up and everything. But now you don't need that. You don't need fancy perfume too. Sometimes you don't need to take a shower, especially when the weather is cold, like in UK now. And everyone I saw that as an English teacher, I said that everyone had a great improvement in doing presentation, especially in English, because when you do it with the Zoom, you can use a kind of notes and everything. And the good things is maybe it's a bad thing, but some students are good in using social media and following the class in the same time because the lecturer didn't notice, Okay, so the topic this afternoon, I think Dr. Saifuddin told me, and it doesn't have to be very serious, but the point is how to stay and survive and maintain your performance as a student in a pharmacy department during the pandemic's time. So I'm sure that some of you would like to share the problem, but I know that there's a poll in Indonesia last year around July conducted by our Ministry of Education say that almost more than 50% of the students, they don't like online class because they have some problems, including like the internet connections, they need to pay extra money, although there's like a little subsidy from the government, but most of the students, they prefer to have face-to-face class, but we have to change because the pandemic changed everything. So I'm going to have for the first talk, I'm going to give the chance to Professor. Okay, Dr. Hana. In Indonesia, we like to call people by their first name to show, yeah, we like to call people with the first name. So we have two great Hana today, you and also Hana, the moderator. And
we like this too.
Okay, and Professor Patrick Ball, maybe the first session will be both of you. You will be talking from the UK perspective.
Slides are on Dr. Hannah's machine, so you won't want to put her on the screen.
Oh,
but we're in the same room. We're
Please.
only a meter apart.
Please, Dr. Hannah, Professor Patrick, the screen is yours. We'll have about 30 to 45 minutes for your presentation, and later we have Q&A, and then we'll continue with Professor Taher and Dr. Saifuddin. I don't need to introduce you because you've been very popular here and also among the students. So as you know that both of them are in Wolverhampton University, and some of you are very popular with the football club from Wolverhampton, right? And and Birmingham is one of the nicest cities.
Wolverhampton Wanderers are not doing that well at the moment.
I know. The pandemic is hard. Okay, please, Professor Ball and Dr. Marcy. The screen is yours.
Thank you. Now, because of the way our computer set up, so when I share with Zoom, if you give me just a second, I will share actually my desktop rather than my screen. So,
while she just does that, can I just say, as being invited to judge, we have been extremely impressed again by the standard, both of the content of the presentations and also the English that you present with. It's really encouraging to see that the standard is being maintained and even improved. We keep saying when we come to visit, when we're allowed to come to visit, that it is really important to develop English language because it is the international language of science, and all the international conferences do tend to be in the English language. And if you can speak English, you can speak to almost the whole of the rest of the world. So congratulations, you are maintaining a very, very high standard of science and the ability to present it.
Thank you, Professor
I'm not
Patrick.
sure which image you can see,
At the
Patrick.
moment, I can see your Gmail.
Okay, so you can see my slides at all.
No.
Okay, so technical issues.
Go back to what you were doing.
Yeah.
Dr. Hannah, just a little suggestion. If you just need to show the slide, maybe you can send it to one of the comments.
The problem with showing just the slide, it will show you this view.
Yes.
That's okay. That's the slide.
Yeah. Okay.
Yeah, we see it.
So, thank you very much for having us. We have a bit of a unique situation here in the UK. We up to our third lockdown. So I want you to imagine that students went to campus. Our semesters actually start in September. So we've got...
It's the same here.
There is, yeah, lots of feedback. So our semesters usually start in September. So our students in 2019 went to the university face-to-face, attending in class as per usual until the end of semester 1. And then they went in lockdown, finished the year in lockdown. The new students were all hopeful, came back to campus in person, and then suddenly been sent home again at the end of 2020. And we're expecting that they will finish the year also in lockdown. This is our third lockdown so far. Everyone thinks that university canceled, but the bad news was for them, no, really, it's not. You still have to study, you still have to sit the exams, and you have to get it tough as much as everyone else in the country. So how is life usually in Wolverhampton? Our teaching is our curriculum based on team-based learning. What this mean? This mean at the beginning of the year, when the student enrolled, we team them up in between 6 and 8 students in each group. And for the first year and second year, they are in the same group. For third year, they have a new group. And then for fourth year, they are fully independent, but they meet in groups for certain activities. What the groups do, the group attend as bubble, attend the classes as bubble, and within this bubble, we give them topic learning material, and then they actually help each other in learning. So you can imagine we make the groups a combination between strong students and weak students, and from different ethnic backgrounds and so on. So they can actually mix up. Favorable or not favorable, some students like it because other people pick them up, but some students hate it very much because they feel isolated. They feel it's not their forte or, yeah.
One of the things that we have to make very clear is that ours is an extremely diverse population. Although almost all our students come from the United Kingdom, we have Wolverhampton is an area where many families from previous immigration periods settled. So we have a lot of students from a Sikh background from Punjab in India. We have a lot of students from Pakistani and Bangladeshi family origins. We have quite a lot of African Asians, and we have a number of Afro-Caribbean students. And actually, here in Wolverhampton in the UK, we have a remarkably small contingent of white Caucasian students. So our classes are extremely diverse in background, religion, outlook on life, etc. So in actual fact, it makes for quite an interesting teaching group.
Yeah, but personality trait wise, people have preference. Some people like to work alone. Some people don't mind to work in a team. Some people feel empowered by having other people around them. Other people feel intimidated when they have people around them. So while the teamwork is good overall, because this is how we work as pharmacists, after graduation, still the education environment, I will have to say, suit 50-50. So in a good day, in a normal life, we do all our teaching face-to-face, all the students cohort in one huge big classroom like yours, either a lecture theater or in arrangements. We don't do a lot of sitting in rows, but we have tables where, or group arrangement where the 7 or 8 students will sit in a round table to be able to communicate with each other, but all kind of face-to-face. So they all together, we have breaks, so they have time to socialize together, but also they knew us personally. We are in the same building, so walking between classes during lunch breaks and so on, you can stop in the corridor, socialize with the students, answer their questions, address their concerns without really very formal kind of arrangement to be able to talk to them. We have for sure like yours, labs, we have, it's all hands-on. Students can do the experiment and see the impact of it right there in front of them. At the same time, they do placements. So they go to the hospitals, they go to the community pharmacies, they go to the GP clinic, and they can see the profession in the real world. They can be part of the profession in the real world. Exams, all in the exam hall, or if it's an oral examination, so it's one-on-one. So exams in person, group presentations like we did today would be in a classroom with audience, and so on. So it's all face-to-face, in person. Do you want to talk to this?
Okay. So then came the first COVID, and suddenly having opened up, ready to start the second semester of the 2019-20 academic year, suddenly we had to send all the students home again. The UK moved into its first lockdown. Only essential shops and factories and hospitals and places are allowed to open. Everybody else was told to stay at home, work from home if possible. You're only allowed to leave your home to go for essential shopping, medical appointments, or once a day to go for exercise. And if you exercise, you're supposed to go from your own house. You can't get in the car and go somewhere nice. You have to go start from your own house, walk, and come back to your own house. You're not allowed to meet with other people. You have to wear a face mask whenever you are out of doors and in shops and places like that. And all cafes, restaurants, places like that were all closed initially. Then things started to settle. The first wave passed through and the government tentatively started to try to open things up. So we were allowed to open more shops, socially distanced. You have to keep 2 meters away from anybody else as you move around. They tried to open up some cafes and restaurants and allow some social life again. And also, we were being encouraged to start to reintroduce some face-to-face components so that the students, in theory, would return to campus for part of the time. But unfortunately, the infection started to take off again, and we went into various different measures. First of all, we had a tiered system, and then we went into a further lockdown. The lockdown has all sorts of other effects because it causes a lot of distress to our student body and to staff, because families are not allowed to come together even for things like deaths in the family and so on. They're not allowed to, a maximum of 30 people are allowed to attend a funeral. So there are a lot of restrictions. One restriction is even I can't get my hair cut at the moment. And in terms of moving to online learning,
Patrick needs a slide. Yeah.
In terms of moving to online learning, it seems fairly straightforward, but we have, as I said, in our student population, a lot of our students live in the family home. And in the family home, you will have 4, 5, 6 children all trying to use online education on one same connection. So we've had a huge amount of issues with our students not being able to get reliable connections because there just is insufficient bandwidth to cope with everything we need to do. So that's a bit of the background. So let's look at how we've actually put this into place.
Okay, so as Patrick said, we have certain facilities remained open in a university. When we were out of the lockdown, everything closed during the lockdown, but certain facilities remained open as a safe haven for students who can't study at home and would like to remain on campus. And this mainly the library. Sorry, the slides is spilled over the background. So it's a library. Some personal study places in the library remained open, for sure, with extreme measures of surface cleaning and everyone wearing masks and so on. So we moved to the online teaching. So you imagine this was in beginning of semester 2, our university calendar year. So everything curriculum-wise was designed to be taught face-to-face. So we had the panic over 1 week to actually develop all the classes to be suitable to use online, to have, I'm sure you are all familiar with Microsoft Teams. We have also something on Canvas called the Big Blue Button, and I will show you images from it in a few minutes. So we had to reshape all the material to actually be suitable to be taught online with the highest possible level of engagement.
Can I just slip in? Canvas is our university's online teaching platform, similar to Blackboard and Sakai, but our university uses this one called Canvas, which provides for our online provision for most of the year. But again, suddenly the amount of material that was having to be carried through this platform dramatically increased, and the university had to make sudden urgent arrangements to increase the capacity of the Canvas platform to accommodate suddenly, instead of just supporting face-to-face education, suddenly it was the only vehicle.
Look at my background, look at every one of you background. You can imagine if it's the things we require to see your background in Bali, it's good background, Haman. But I want you to imagine not everyone computer savvy. Not everyone actually have a computer. Some students were using or still are using their phones, and you can do limited things with that. But you can think about if it's assessment, and they have to turn their camera on. This extreme pressure to be able to turn the camera on and do exactly what we have now, where you have a feedback because more than one person having their sound on, but also a lot of our students are mature students, mature age students who have families. So children floating in the background somewhere, but also we have a lot of multiple generation houses where you've got another 20 people living in a house, and again, knocking on the door and talking to their kid or mom is very common. So a lot of embarrassment to show your background, a lot of difficulties to be able to participate. So as a requirement from our registration body, it was that the students must have at least a face-to-face, 2 hours of face-to-face sessions. So we had to regulate what we can use face-to-face, and the decision was made professional communication, so dispensing basically, and the calculation, we will do them face-to-face over 2 hours. I'm not saying everyone will come to the university, but we divided the class, the 100 students for each cohort into about 6 bubbles between 8 and 10 people. And they are the only group allowed on campus that day, with only one academic and one marshal. What I mean by one marshal, one like a hawk watching the surface cleaning, the mask wearing, the toilet use, everything infection control wise, and the other person teaching. So you can imagine by the time we run over first year, it's a full one day. The 2 hours session actually in reality is 4 hours, because you need to keep the space not occupied for a period of time to clean the air. You need to clean all the surfaces before the next group come, and so on. But it worked while we were out of the lockdown. So it worked for a period of time.
And also the government regulations dramatically restricted the use of our accommodation. So for example, our main teaching room, which has a normal fire capacity occupancy of 120 people, under government regulations for physical separation, was rated to accommodate 17 people. So clearly anything we were going to do face-to-face on campus was going to have to be run multiple times.
Yeah.
They'd have to do the same class 4 or 5 times just to get everybody through.
Yeah. And then we had to, university bought Microsoft Team license. And you remember I told you we have multiple teams in each cohort. So we now have, I just did assessment. On Thursday, we use Microsoft Teams to do the teams, and I will share a screen with you. Laboratory and placements moved to be simulation. So we bought again softwares. So the placement simulation, it's a background of community pharmacy or a GB clinic or a hospital, and one academic taking the students through the facilities and talking to them about patient problems, simulated patients, and so on. So I all went to simulated.
The other thing, just to say, all our students, we have a system where all our students, our staff all make themselves available for a certain amount of time every week for students to book one-on-one tutorials. And these normally take place face-to-face in a group of bookable rooms where you just have, there's a computer, a table, and you can have one staff member with 1 or 2, maybe 3 students. Of course, once again, all of this has all had to move to online interaction.
Okay, so you remember the personality trait I spoke to you at the beginning. So at the normal life, we had 50-50 satisfied because people didn't like the social encounter day in, day out. In a lockdown online, we also still 50-50 because people didn't like the online environment. Why? Because they lost the ability to talk to us in a corridor, but also some cohort didn't even meet yet. They don't know each other in person. So especially this year, first year, who came for the open day, and after 1 week, after 4 weeks, October, they were sent home again because we had lockdown before Christmas, so beginning of December. So they very shallow know each other, but they know only the bubble they were attending university in. So they maybe know 10 people of their class in person. They met them for a maximum of 8 weeks, and now it's all online. So we have a first year student, second year student who have very, very shallow knowledge of their mates in the same cohort because they didn't meet in person.
However, it made for some interesting teaching opportunities, particularly in the public health arena, because this government's attempts to try and the university also to try and restart face-to-face learning with the spikes in infection it caused, A, when the students traveled from their homes back to campus, and then B, again, when the students were again sent back home and they all traveled from campus back home, there were well-documented enormous spikes in the infection rate in the UK. So this is very good practical examples for teaching how to manage a pandemic for the future.
Yeah, as a future healthcare professionals. But at the same time, university experience, I'm sure all of you will agree, university experience different than high school experience, different than sport team experience. It is a different environment. So people who knew the experience and loved it, they miss it. The new people who didn't have the experience, all what they know about university is the online teaching and seeing people virtually. But people who hated to come to the university, who hated the social encounter, actually extremely surprising. Very large number of very weak students, socially not engaged students suddenly become engaged and actually flourish. So some win, some lose. No usual end of the year social events. We used to go and have a meal with the students at the end of the year. For sure, it didn't happen last year. The graduation were virtual graduation. They didn't have the chance to come on campus and for us to celebrate them. And also they had a chance to see their lecturers and each other for last time before they all disperse all around the country for their jobs. At the same time, the new students didn't know us personally, and they didn't have this hands-on experience because everything virtual and simulated.
So, Dr. Hannah, you're saying that actually because of the online interaction, some students who normally do not speak or engage in the class, so they also become more active? Are you saying?
Absolutely, because they talk in the chat box while we are online teaching. So they don't have to use their voice. And like me, with the English, I have accent. In Australia, I don't. In my head, I don't. But you will have a lot of students who have accent. Some people have speech impairments. Some people have an anxiety. But the fact that they typing message, empowering them to actually engage and participate. So
Yeah, some students, they feel bullied because of their accent, you know, which is actually, it's not really a problem.
Absolutely. But also there is no cameras.
True.
So I don't have to be physically embarrassed if my answer is wrong. So, you know, the embarrassment layer being taken away from people with an anxiety or social phobia, or have an issue they think that been bullied or not well perceived by other people. So some students enjoying it. Some students also save the child care, comfortable at home to actually do their homework from their bedroom, basically, so go on with their life. Other people being compromised because home might be not the safe place for them, and having a break to be in a university is a safe haven, or too many people at home, and they have a family responsibility toward the parents' caring responsibility, and they couldn't actually do the same. Don't forget the kids at home not playing, the kids at home doing exactly what the parents doing.
The other thing that the university very quickly realized was that there were a large number of students who were challenged in their IT facilities at home. And so the university made huge efforts to root out every available laptop computer in the buildings, and made something like 500, 600 machines available for loan for students who didn't have their own adequate facilities.
And internet connection.
Yes, some of these have also got portable internet modems to connect through the cellular phone system in order to provide them with internet connections. Because even where, as I said, even when the house may well have a broadband connection, when you have, as I said, 6, 7 other family members all trying to use the connection for their education, their classes, etc. as well, the connection just can't cope with it. And this has also led, for example, when we've had online assessments and examinations, we've had students actually choosing to travel in to use the study bubbles in the university library to do their assessments from there. Yeah. Or some of the students who live further away. We've even had students booking rooms in hotels to be able to do their assessment and know that the
it is tough for some
connection will hold.
Yeah.
So it's been really tough. But on the other hand, people have been really quite inventive. Many of the students have sort of taken their own initiatives to find ways of coping with the new reality.
Yeah.
And it's been really quite impressive to see how people have adapted.
It was tougher for the students who have their own children at home, the mature age student who were teaching their students, their kids at home, while they also have to study. But in normal life, they will send the kids to learn at school from their teacher, and they come to the university to learn from their teacher. So there is a dedicated learning time for the kids away from the parents. So this was the toughest gig, the mature age students. So I will just take you through a tour to some of our systems. If I have to get out and get you live to one of our systems, I will do. but this will be if you're asking me to do so in questions. So this module, our curriculum is 1 year module. So the 6PY011 is our 3rd year students. So they started their clinical, their therapeutics, their clinical. And as you can see, we divide them to 12 groups. And when we do the team work, I actually initiate meeting from here. And then they all come to one big room like we did in Zoom. And then they go to their break rooms, like you can see here.
So you can see that the top line is the general meeting, which everybody comes to initially, and then they go to their separate group letter subunits for their face-to-face group work.
So before they come here, they do what we call readiness assessment. They do it individually. They don't have to come to the meeting yet. Online, using quiz, and I will show you what I mean by quiz. And then they come to this room, they do the team assessment, which we give them multiple choice case studies. They go to their break rooms, they do the assessment, and then they submit it on another media, which you call the Canvas. This is where we record our sessions. So I'm giving lecture or a workshop. I will go to Canvas, the big blue button, and then I will do my own slides exactly like I do with you. I can see the recording happening in the background. But after the lecture finish, because you remember I mentioned this mature age students, they may not be able to dedicate the time to attend the class at this point of time. But what they can do later on, it's fully, you can play a video where the case on the side or the lecture slides on the side, but we are there and we talking to them about the topic. We have the chat box. While they muting their microphone, we invite them, if they wish, to unmute and turn their camera on and come and communicate with us in front of the class. And this way, their pictures and their messages will appear. I pruned this picture so you don't see the chat side of it because it got a student's name in it and I'm not allowed to share it. But it's usually the recording will have, so if you look at this side here, I cut it off. At this side, all the chat talk actually also recorded. So the students who missed the live conversation, question and answer time, they will see it recorded, but they will see it also written. But we became inventive. We give them activities like go in your group and do posters. We did this with you guys. Whoever remember us from a couple of years ago, we gave them a topic. This topic was pregnancy and breastfeeding for our final year. So it's a clinical final year. And we give them topic and they go make posters. They come back and each group presents their posters. So this is one of theirs, but also we can do very primitive things. This was a communication class. And what I did, I draw a picture on a piece of paper in my hand, and we did the one-way communication, the two-way communication. So they draw a picture without having any possibility to ask questions. I relay description and they draw the picture. It came so funny, you can't imagine what it came out. And then we played the same activity with different picture, and they were allowed to ask questions to clarify things. And then after they finished, they draw it on a piece of paper, they send it to me by email, and I share it with the class. And this was one of the winners. I do winning competitions with them to keep them engaged. So one of them get it absolutely right from just the description, relaying the description, they draw the figure perfectly. So inventive engagement, from my point of view as an academic, I can see the online disadvantage is ability to see people in person, to have this personal skill, to be able to communicate with people, but also, again, for people who the university is safe haven, to have this safe place to learn, study, and perform. But at the same time, I can see there is a big merit of online teaching, because it allows a lot of people to take part who otherwise wouldn't take part. But at the same time, to have independent learning. And this is very, very important. When you are in a university, you can hide in the woods behind someone else and never answer a question, live question in the class, and you make it through. But because you don't learn to be independent, you are one of 100. But when you learn from home, you are 8. You either learn or you don't. So again, like my first slide, it's 50-50. So I think we are up to questions. If we have any time left, and if you want me to navigate live with you with our system, I'm also happy to do that.
Yeah, Dr. Hannah and Professor Patrick, let's give a virtual applause for both of them. Actually, you still have about 10 minutes for the presentation, and we have a special time allocated for the Q&A in the end. But it's okay, we can also do the Q&A now. But one thing which is very interesting for me, actually, in some cases, of course, there is always blessing in disguise. In some cases that the online learning can make people to be, they can have more opportunity to talk to the personally, which is maybe in big class it's hard to happen.
I will share the screen, my screen with you, to actually show you our. Can you see my screen?
It's ongoing.
Okay.
There you go.
So this is our learning media platform. So if I enter one of the years, so this is our 4th year. So you can say I can go to conferences, so I can arrange a class with them over conference, and you can say this is one of our recorded lectures. It was a workshop yesterday, and if I click on the recording will come straight away like this, and you can play the video and share it with later on. You can go over it later on. We can send them announcement to be engaged with our activities, but also we can give them quizzes online, so they can do exams online and so on. I will take you on a tour for another class to show you. This is the 3rd year. We just done this in Thursday. So if I take you to one of the quizzes, which again, this is just we done on Thursday. The students do it online. I can have statistics, which questions they done, but also I can go back and look at the grades for each student, and I mark the question from there. So it gives us good facilities to meet the students.
And it's also really good that it gives us automatically from the marking these analytics that show not only the total grade, but which questions were proving difficult. We can see straight away whether one particular question was failed by a lot of students, and we can, as academics, go straight back to look at that question and try and understand why. Is it something we didn't teach adequately, or is the question badly worded?
Yeah.
So it's a very useful academic tool.
This is team-based assessment, which we give them a task, and then they go work on their group on Microsoft Team, and then after they finish with it, they come and submit their assessment on Canvas. And as you can see, they answer the questions, and the grade granted for the whole group. We even last year done this assessment, the last final assessment on Canvas. So if I go to people, you can see our arrangement. We do case-based learning, again, divided into group. So they divided to the larger group than the TBL group. In the TBL group itself, sorry, I have to move you guys. So in a team group, I can actually, so if I go, I can visit group A while they're working, and I can send them announcement. I can start a discussion with them, which, as you can see, I did a discussion with this group, and then I can pin it for them to see later on. But I can meet with them for a conference, which, as you can see, I did twice to give them formative feedback about oral presentation we did. So it is very, very reliable software.
But to put another side to it, we've been doing online assessments and examinations, oral examinations in particular. Now, one of the things is that when you have an oral examination in the building, you generally have 2 or 3 people running around dealing with problems and issues. So that if somebody doesn't turn up, or if somebody is in the wrong place, there is somebody else who can deal with it. Yeah. But when you have to do the oral examinations online, generally speaking, we've had one academic has got a list of people, and they've got a link that they've been supplied with to connect. But if the examination goes absolutely swimmingly, that is fine. But if, for example, somebody's connection falls over halfway through an assessment, we've got to give them more time. We've got to give them a chance to get their connection back up, or reschedule them to do them later in the day. But at the same time, we've got a day's worth of assessments booked back to back. And for the academic staff, trying to maintain the assessments, listen carefully to what the student is saying, but in the background be trying to email the next 10 people on the list to say, I'm sorry, we've fallen behind schedule, your new time is. It's presented quite a few challenges yeah. on the way.
Yeah. As you can see, first year cohort is quite big cohort, so they have about 15 TBL teams. So it works for all the classes. Now, Patrick mentioned before that we now students have to book appointments through a system we called SAMS. So this is kind of how we do these things. So they can book, I don't know how to move you now. I need to move you around. So here we go. So we can book appointment. They can book appointment.
We're still looking at TNA.
Okay. So is it still not changed?
No.
Okay, I'm trying to change the screen. Maybe I have to stop sharing and share again. Okay, so I will just stop share and share my screen again so you can see the new one. This is what we call our students' appointment.
No, this is email I'm looking at the moment. I'm looking at your Outlook email.
Okay. Stop sharing again.
Yeah.
Here you go. The challenge. The challenge. We're only challenged by this, but let me try this again. Can you see it now?
Yes.
Yeah, we see.
So this our student booking system, where I can offer calendar meetings. So as you can see, I offered this many personal meetings. So one-on-one with the students on Monday, and I've got already 3 booked. And then we go back to either meet them on conference on Big Blue Button, or we meet them on conference on Microsoft Teams. We call the student in and we have one-on-one conversation.
In normal time, do you have this very tight schedule, Dr. Hannah?
Yeah.
Before the pandemics?
Yeah.
Just like this? I see.
I should have shared my calendar with you when I had my emails.
Yeah.
Yes, wall to wall until 9 o'clock at night.
I see.
Yes, but the point is that there are some, you have to fit the appointments in, and not every appointment gets taken. But you have to, other days, for example, you might have teaching or meetings virtually the whole day, and you can't offer any appointments that day at all.
Yeah.
So every academic staff, as part of their contract with the university, is supposed to offer at least 3 hours of face-to-face student contact appointments every week. But we try very hard to always be in excess of that, offer more appointments than we're actually obliged to.
Yeah. And then you get the individual academics who are actually putting themselves out there for people who want to have a one-on-one conversation. It might be after hours, it might be in a weekend. So after I finish with this conference, I have one of my students at 11 o'clock on Saturday. It's weekend for us. So our weekend is Saturday and Sunday, we don't go to work, but you have to accommodate some students if they have difficulty communicating during the weekend. This is the only time available. Not everyone doing it, but for the 2 of us, we do it regardless of time, because we have kids. We know that younger people can feel stranded if you don't communicate with them. It's again, individuality of academic behavior become very prominent, more prominent during the lockdown. Who putting themselves out there and who's not.
It also has other little side effects. Just one very quick comment to finish, because previously we would be based in the office at the university most of the time, and we would only do a very limited amount of time at our desks teaching from home. Because we're teaching almost entirely from our living room, we've had to invest in new chairs that are much more comfortable to sit on.
Yeah, that's
bought
true.
Ourselves proper office chairs, and we've also had to upgrade our computer facilities. We bought ourselves better monitors and
and the house wallpaper too, probably. Professor
Yeah,
Patrick.
I never knew a corner of a room much better than the corner I'm sitting in for 12 months.
Yeah.
Yeah.
You can try my background.
Everything we wanted to say.
Okay, thank you very much, everyone. Let's give big virtual applause for Professor Patrick and Dr. Hannah. We're going to have the Q&A session later, if you don't mind, Professor
Yeah,
Patrick and Dr.
all
Hannah.
good, all good.
And I know it's
switch your microphone off.
Yes, thank you very much. And there are already some questions actually, but I want you to put the question in the chat room, and maybe it's praying time for Indonesia, especially the western part now for Asar. So we don't have a specific time for the prayer. So if you want to do prayer, feel free to turn off your camera and do your prayer, and then you can come back because we are all in different part of the country now. Okay. Wait, I got a message. Oh yeah. Okay. Now we're going to continue with Professor Taher to talk from the, from Kuantan, from the Malaysian experience in IIUM. I know that most of you're impressed with the Canvas, and many Indonesian universities are not using that platform yet. We still use Google Classroom, and some of us use even WhatsApp group for the backing up. So maybe if Professor Yandi, or the person in charge are still here, maybe you should be considering using Canvas too for our LMS, learning management system or learning platform. It's really complicated, but I know it's expensive. Okay, now we have Professor Taher with us. He is a professor in IIUM, and normally in IIUM we don't say the long abbreviation anymore, right, Professor? We just say IIUM. It's already popular all around the world, and in kuliah pharmacy in Indonesia, we also say kuliah for for going to college,
Yes.
So he got his PhD in bio. Oh yeah, in Professor Taher, kuliah pharmacy, yes, and organic chemistry for his master. So maybe we would like to hear more from you, Professor Taher, from the professor perspective, lecture perspective, and also from the students' experience. How is it, being a chemistry or a pharmacy students in the pandemic time? How hard it is? Because when Professor Patrick explained about some students, they cannot get connection. So some of them, they are struggling to get the internet connection. But in Indonesia, many students, they break the rule of the health protocol. They still go to the cafe where they can get internet access, which is actually not allowed. See, Kian and Sophie, I think they have broken the rules, the law sometimes. So and maybe you also need a kind of a motivation too from you, how to keep the quality of your academic performance as a pharmacy student, even if you are we have a different kind of situation at this time. So Professor Taher, we have about 30 minutes for you for the presentation, and later we will continue with the Q&A. Please, the virtual platform now is yours, so the screen is yours. Let's give big round of applause for Professor Taher, everyone.
Thank you very much, Mr. Herman. Assalamualaikum warahmatullahi wabarakatuh.
Sama-sama, Pak. Waalaikumsalam.
Prof. Patrick and Dr. Hannah, how are you? Nice to meet you here in virtual meeting. Let me share my slide.
Are you in Kuantan, with KL, or in which city now?
In Kuantan.
In Kuantan, I see.
Yes. Okay, thank you very much, Dr. Herman, and also UII for inviting me to share my little bit experience in teaching and learning during pandemic times. Since the government has announced the lockdown at around 1 year ago, we as academician and also university was shocked about that, and we don't know what to do. So university has been initiating how to solve and how to survive. Yeah? Indeed, COVID-19 teach us already how to resilience, because COVID-19 has changed everything, including in education. It took quite some time for us after government announced lockdown in March 2020. I think about 2 months for us to prepare, because students need to go home and staff need to work from home, and we don't have experience how to handle this kind of problem. So we get instruction from Ministry of Education, and university has been very active how to overcome the problem, yeah, providing training to all staff. Yeah, even at the time we don't know how to share the screen, and also we don't know how to even to record our lecture in PowerPoint.
Yeah, same problem here.
So it was very shocked. So during the time, that 2 months time, because we don't want to leave no one to left behind. I mean, because you know the topography of Malaysia and also the development of ICT here is not because we have a rural area and remote area, and many students living in that area, and now they need to go home and stay at home. So and also we don't want to we don't want to compromise, yeah, about the quality. Yeah, the quality is everything for us. So we university have established we call it emergency remote teaching and learning. We call it ERTL response team. For that 2 months, they were working hard actually to get to provide skill and knowledge for staff how to deliver online teaching, and also that team also working hard to get a complaint and also problem from the student, because some student they don't have actually internet facilities. So we need to provide teaching and learning materials to them. And at the time, yes, we sometimes we need to record it in pen drive. Yeah, we record our lecture and save it in pen drive and post it to them. Yeah, post it to them. So after 2 months, we started to have online teaching. Yeah, online teaching, and university have also developed their learning system. And about e-learning management in UAE, yeah, sometimes we call it UAE, yeah, sometimes IUM, actually old name for IUM. Yeah. We have a center, actually, a center to respond regarding personal development. Yeah, we call it CPD, yeah, Center for Personal Development. This center is responsible to manage a learning system, yeah, a learning system in IUM. And I would like also to bring you to our learning management system. Yeah, we call it ITALIM, yeah, Integrated Teaching and Learning Environment System. First time after we start online teaching, yeah, I think similar to Canvas in UK, as Prof. Patrick mentioned to us. Yeah? The system not able to cater everything because everyone is online. Yeah, so you still need to upgrade the system, yeah, to accommodate all the online teaching. So in learning management system, we call it ITALIM. Yeah. So every lecturer, yeah, they have their own account for their own course. Yeah, and also for such lecture, yeah, they can also share the folder in ITALIM. So this is actually the ITALIM, yeah, for our university, yeah, the university learning platform, yeah, that can enhance existing teaching and learning environment with a wide range of standard and innovative features. So iTalim itself has been upgraded few times, yeah? Because their capability not really sufficient enough to facilitate, yeah, to facilitate our online teaching. So I will bring you to the content, yeah, of the iTalim. Let's say one lecture they have their account, and let's see for myself, yeah, this for my account, yeah. What we have, I have everything here. The topics that the subject that I taught in this semester, yeah, in this semester. So everything related to teaching and learning need to be uploaded to the system. And for your information, in Kuliah of Pharmacy IUM, we have about 120 students per batch, yeah, in average. Actually, the highest number of students is year 4. It is about 141 students. So all instruction, yeah, all instruction regarding the e-learning, yeah, e-learning, we need to upload to the system. And then we have to provide everything in our account, yeah, to support e-learning process, yeah. So what we can do in learning, we can provide a rubric for particle, a routing manual, you can upload here, yeah, and also we can provide attendance, yeah, attendance. So during online teaching, we have actually a tight schedule as well, yeah, like Prof. Hannah mentioned just now, yeah. We follow the timetables, yeah, but the duration, yeah, of the teaching, yeah, we reduce into 40 minutes, yeah, maximum 40 minutes. So to support, yeah, to support iTalim, because we got some problem last time with the iTalim regarding their capability, yeah, their capacity, and we support our teaching and learning using MS Team, yeah, Telegram, yeah, Gmeet, Zoom, Google Form, and also WhatsApp. There are 5 common problems, yeah, faced by students in Malaysia in general, yeah. The first one regarding the managing time, yeah, because normally the student by default, yeah, in face-to-face, they need to go to classroom and go back, do assignment and so on. But if they are doing online learning, yeah, they need to manage their own time, yeah. They need to manage their own time and also the activity, yeah. And also regarding the stable internet access, yeah, stable internet access, because some students are living in a rural area. So we have to settle this, yeah. So for those students who are living in the rural area, the university have called them to come to university, yeah, to come to university. So some student that has have problem, yeah. So now they are living in the university. And other problem is about lack of quiet or private place, yeah, for them to study at home, because probably perhaps they have a big family, yeah, big family. So in this case, the student also allowed to come to the campus, yeah. We facilitate them to go back to campus. And then regarding unclear communication, yeah, it is regarding the internet quality, yeah, internet quality, even at university, yeah, so we have sometimes got problem with the internet line. And then they have called feeling disconnected, yeah. So one of the challenge in online teaching, teaching and learning is about the student engagement, how to engage, how to engage with the student, So we have to be innovative, We have to be as a lecturer, we have to be innovative, in order to make students to engage with the lecture. And then for lectures, we classified into synchronized and also asynchronized. The synchronized means the student need to sit online with their lecturer, So even though they don't on their camera, but we assume that they are online, mostly students are not opening their camera, so we don't know actually whether they are still there or not, So what we trust them maybe because we treat them as an adult, So we just assume that they are online, and another one is synchronized. Synchronized means we because not all topics that is available in course online should be delivered synchronously. Maybe some topics course coordinator need to identify which one, which topic that to them not really important to be delivered synchronously. So the lectures can just upload their teaching materials into iTalim, and after that the student can access it.
Sorry, Prof. Tahir, can the student download this kind of the iTalim in their mobile phone, or they just need to access it using their laptop or computer?
Yes, they can use their handphone.
Gadget,
gadget.
Mobile phone,
Because the system has been created to be accessed through mobile system.
I see. Yeah, because some students, they don't have laptop, they just have their mobile phone.
Yes. So yes, Mr. Ma. Actually, I forgot to tell you regarding that matters. So some students, they have problem with the device that they don't have that kind of device to support their online teaching, and university has raised some fund, endowment fund to support the student, this for the student who doesn't have a device to support their learning. Now, I would like to share with you reading practical. As I mentioned just now, some students, who got problem there with the internet line, also not the good environment at home, so they are already in the campus. Some students are in the campus. For example, year 4 student, about 25 students are in campus. So in IIUM, because the university really concerned about the COVID-19 spreading, so campus is actually green zone, green zone. Whatever activities that can be done, it is allowed face-to-face, but we have to follow SOP, strict SOP, 1 meter distance, others, and using face mask and other SOP. So the student who are in the campus, they are able to join the practical. So we make practical by group. So one group should be led by students who are in the campus. So they can share their experience. And also during the practical, we make a video. We make a video and edit video. So we follow the manuals. And that video will be distributed to the student. And also we provide previous practical report. So, and then the student will be easy for them to follow. Easy for them to follow. And then and regarding practical result, so we'll ask the student to write their report, and in this kind of report, because the report is in a group report, So, and also they are not sitting together. Yeah, one student maybe they are in KL, one maybe in Kuantan, right? So we ask them to assess their peer. Yeah, it's a table. Yeah, table. About for example, the student one group has 4 students. Yeah, so one student need to assess their peer. Yeah, the click. Yeah, another click, and student B also need to assess student A, C, and D. Yeah? So we have such calculation, yeah, regarding the engagement to the report. Yeah? And then presentation of the practical result. Yeah, we do the video recording, yeah, by group. So everyone should present, yeah, should present the result of the practical in one file. Yeah? And then after that, the student will share with us through Google Drive, and we can access their presentation. And then regarding business project, yeah, final year project, for those who are already in campus, they are allowed to conduct real lab works, yeah? And for those students who are at home, yeah, they need to do review, yeah. So review can be narrative review and also systematic review. Actually, the main problem, yeah, in online teaching and learning is getting student engagement. Yes, so we have to be creative, innovative. Yeah, I'm not sure whether it is what we practice here is quite innovative or what to ensure student engagement. Yeah, so one of the effort is to have a formative test. Yeah. To allow the student to do just light assessment, yeah? And then we conduct tutorials, yeah? A discussion regarding the topics, and also we conduct peer assessment. So at the end of the semester, we did assessment. Yeah, we don't call it examination. Yeah. We conduct assessment using iTa'lim as well, yeah? Online written examination, yeah? Or online written assessment actually, yeah? So the type of the question is in the form of essay and also MCQ, yeah? And then online OSCE, yeah? The Department of Pharmacy Practice, they conducted the assessment for OSCE through online, yeah? So we have to establish our own protocol. Yeah? Protocol must be clear here because it is not like face-to-face that can be instructed very effective. Yeah. So for online OSCE, we have to provide a clear protocol, yeah? For them to follow. Yeah. And then regarding assessment, we also conducted online presentation depend on course online. Yeah. So every course line they have different type of assessment. Yeah. So to ensure the quality and also for audit purpose, we have to follow course online. Yeah.
What about for the practical subject, Professor Taher? What about the assessment for the practical subject? Where is the need to go to the laboratory or?
No, just we do virtual practical. Yeah,
yes.
Yeah, because as for your information, actually Malaysia now at third wave of COVID-19. Yeah? The first third wave already. So we have actually the university what we call COVID-19 task force. Yeah, has been working hard actually to bring students, yeah, to back to the campus. Yeah. The first wave actually the time of the spreading COVID-19 was reduced, yeah, reduced, and we at the time we decided to bring students to campus at a certain date, yeah. Unfortunately, yeah, and unfortunately, actually,
case
at that
increase
day,
again.
yeah, increase again, yeah, increase again, And university need to follow instruction from Ministry of Education. Ministry of Education should follow instruction from what called National Safety Council, under Ministry of Health. So actually the multi-layer of instruction, sometimes I myself, I don't digest directly whatever instruction that coming to me, I don't digest directly. I just let to be, just to wait because the regulation change every time, sometimes every hour, every day. So like that, what the situation. So
I learned about it because my wife is doing PhD in Kuala Lumpur, in University Malaya, and I know that the government of Malaysia is very strict about the decreasing the number of the COVID-19, which is actually it's really good. It's really, I mean, you really care about the safety of the students.
That's correct. Actually, what I would like to share with you is the government how to anticipate the spreading, and any premises, school, whatever person that meet each other, we need to scan My Sejahtera. We call it My Sejahtera QR code. Actually, in My Sejahtera QR code, will record your present at certain location.
Yeah.
So let's say one that location is infected, for example, one restaurant, for example, because before second wave, actually restaurant was open, but you have to sit just only 2% in one table. So when you are entering in the restaurant, you need to scan My Sejahtera. So the Ministry of Health, the team of Ministry of Health can detect actually who entered the premise. Let's say one case, for example, is coming from that restaurant. So all the visitors will be screened. Will be screened. And also entering one complex perumahan, for example, taman, if for example outsiders, not the residents, they need also to scan My Sejahtera. To me, the government is very strict in terms of to control the spreading of COVID-19 in Malaysia. I really appreciate it. So now we can actually monitor every state or every province in Malaysia, which province is the highest, and they will lock down. They limit the movement of the people from state to go to other state. For information, actually Kuala Lumpur and also Selangor have the highest population, actually, and also they have highest case of COVID-19. Okay, and then doing assessment, online assessment, actually no one will invigilate you. No one to be to control the student. So before assessment or examination, we ask the student to fill up the student integrity form. Means they will not be to cheat during the exam, and also not to browse any website, not to ask their friend, because if we don't provide this kind of form, so the assessment or examination will be just wasting our time, because there is no integrity. Because assessment, the function of assessment is to assess the ability of student to achieve the outcome of one specific course. So the student need to fill up student integrity form. And then actually the final assessment we conducted using iTaklim as well, iTaklim. So before assessment, we have to provide exam protocol regarding when they should enter the room, how long it will be, and whether they, how they should answer the question. because in Italim, for IC is quite, for IC, we can do it online, meaning the student can write it directly to the system. But if it's difficult, if you provide, the student need to provide figure, for example, or graph. Yeah. So in Italim assessment system, we just provide the question in Microsoft Word, yeah, and upload to the system. The system will be downloaded and they can write it using a pencil. Yeah, can type it, for example, yeah, and later they will upload it back to the system. Yeah, for MCQ will be easy. Yeah. MCQ will be easy, yeah, because we can just use a facility in a Ta'lim, so the student can use it. And for us, also will be easy in the marking. No need to mark, yeah, because it is already set, yeah. And the performance then later on just we can just download it in Excel file. This is actually what we set in at Ta'lim, yeah, this final assessment. So the student need to fill up the attendance, yeah, and submission of short essay. So in this file, we upload the question. Student need to download it and they can answer it, yeah, and upload back to, yeah, back to this folder, yeah. And we have short essay, yeah, and also long essay, and we have MCQ. Yeah. So in MCQ, we just provide what called the student need to answer the correct answer, yeah, not the best answers, yeah, the correct answers with 4 option. Yeah. So after that, the what call after that, everything will be available, yeah, in the system, and the lecturers need to download it and mark it and just report their performance. And in the IUM, we have also channel that student can evaluate the lecturer, yeah. So in this kind of new model of teaching and learning, this channel will be important because we can improve, yeah? We can improve our teaching and learning in future if COVID-19 is still still with us, yeah? Still with us. So it is very useful, yeah, for us to improve our delivery example, yeah, maybe about our infrastructure and also facilities. I think that's all from me regarding the experience how to survive in COVID-19. In line with the theme of IPTC, yeah, regarding sustainability in pharmacy education, yeah, how to survive, how to produce good quality of graduates, yeah, because especially in pharmacy and medical health, yeah, it is quite challenging for us, yeah, to deliver, to achieve our the outcome, yeah, using online teaching learning, yeah. So especially for clinical, yeah, practical. So, but as mentioned by Prof. Hana just now, yeah, some students like, yeah, online and teaching, yeah, and some students don't like teaching online teaching, yeah. But my son was not, he didn't like online teaching. Yeah, told me, is it possible for me to withdraw for this semester? I just told him that you have to take this. Actually, it is not affecting just only you. Yeah, it is affecting everyone. Yeah, it's affecting the world. Yeah. We don't know actually whether next semester or next year the COVID-19 already cleared. Yeah. So I think this is all from me, Mr. Herman. Thank you very much for listening. And I guess if you have any question, yeah.
Thank you, Professor Tahir. Everyone, let's give a big virtual round of applause for Professor Tahir. But what about you, sir? Personally, as a lecturer, do you prefer online learning? Because in fact, some lecturers, they normally they're very busy. Sometimes they skip the classes, but with the online learning, they don't have the reason to skip the classes. What about you?
At the beginning, I was not happy with online teaching, but now I used to it. And
we have no choice.
but I think there is nothing to replace actually face to face, because there is no what call it personal touch and body language, everything. If you can, my experience in teaching student, normally they don't on their video, their camera, so it's quite difficult to, especially for quite number of students for year 4 in Kuliah Farmasi, 141 students. We can, if we use Zoom, so many layers.
True.
So many layers difficult, and also we don't know actually who is the, and also sometimes we don't know their name, their face. Especially for PT for year 1 student, they don't have any experience, to meet their own lecturer, and they don't have a chance to come to the campus so far.
True.
But according to our plan last time, the priority to be given to year 1 and year 4. Year 1 to know their campus, and year 4 to complete their professional training, in industry and pharmacy, pharmacy, apothec, and also in hospital.
Thank you, Prof. Tahir. We will save the question for you in the Q&A session. So what I learned from you and from Professor Patrick and Dr. Hannah, that actually your university and Wolverhampton really keep the quality of the teaching and the learning, even if we are still in the pandemic situation. Okay, so we're going to move on from the UII experience, Universitas Islam Indonesia experience. In this case, I'm going to invite my big brother, I call him brother, Dr. Saifuddin, who would like to share. Maybe I'm also thinking maybe he will confirm, maybe next semester we'll use Canvas too, like in Wolverhampton. Or actually we have something like iTa'lim, it's called Classiber in UII, but so far I think Google Classroom is still the most popular. Okay, we would like to hear the perspective and sharing experience from UII, especially from Dr. Saifuddin, or favorite lecturer in pharmacy department, who is during this pandemic time. So many students misses him, because he is not only a lecturer, but it's also a source of inspiration. Okay, Dr. Safedin, you have about 30 minutes. You can use the 30 minutes or less if you want to give more time for Q&A later. But I think I'm going to pass the virtual screen to you now. Okay. Ladies and gentlemen, please welcome Dr. Saifuddin.
Okay, thank you very much, my brother, Mr. Herman, for inviting me to this session. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Alhamdulillah, wassalatu wassalamu ala rasulillah wa ala alihi wa ashabihi wa mawala. Rabbi syrah li sadri wa yassir li amri wa hlul uqdatam min lisani yafqahu qawli. Amma ba'du. Professor Ball and Dr. Hannah Morisi, also Professor Tahir from IUM. Thank you very much for joining this session also. And actually, Mr. Herman told so many lines about me. Yeah, I'm nothing. And I will not explain the whole aspect of online teaching learning in UII, but maybe I just would like to highlight one interesting aspect, or maybe only one interesting method we implemented in the Department of Pharmacy UII during the online classes period. which is my dispense. And actually my dispense is not mine. I mean, I didn't actually involve in this project, but because this is quite interesting to be shared to the students and also to all participants. So I choose this topic to be presented during this presentation session. So let me start my presentation. So if Professor Ball and Dr. Hana Morisi, also Professor Taher already explained the whole aspect of teaching learning, both in the Wolverhampton University and also in IUM. So here I just would like to highlight about my dispense, which has been used in the Department of Pharmacy UEE for pharmacy skill practice development. And this software last year was used for the students within the professional pharmacist program, but maybe next year or maybe next semester we will also implement the software for undergraduate students. So before continue my presentation, I'd like to credit that some part of this presentation is taken from the description on my dispense provided by Monash University, and some other parts of this presentation is belong to Ibu Mutiara Herawati, MSc and team, because Ibu Mutiara Herawati and her team got permission from Monash University to use my dispense and already implemented last semester. And I'm sure it's quite inspiring. It's quite inspiring to be implemented next semester or next year in UEE. And also permission to use my dispense for teaching learning purpose at the Department of Pharmacy UEE has been granted by Monash University Australia last year. I mean, Ibu Mutiara contacted my dispense support to get permission to use my dispense, and Alhamdulillah, we successfully implemented my dispense in our teaching learning process. So thank you very much, and I'm really proud to Ibu Mutiara Herawati for her great effort to get permission to use my dispense in UEE.
Yeah, maybe it's a good idea later Bu Mutiara can also join us and share.
Okay, so if maybe there are some questions about my dispense, I will hand over the question to Bu Mutiara and she will answer the question perfectly, because I just now maybe what's that? Not really deep understanding about my dispense, but Ibu Mutiara will explain better than me. Okay, as mentioned by Professor Ball, Dr. Hana Morisi, and also Professor Taher, that COVID-19 pandemic, of course, we didn't expect to have this pandemic condition. Yeah, so suddenly we got so many changes in our life. Yeah, and for teaching learning process in we itself since 2016, we already recommended us as lecturer to implement blended learning method. I mean that at least 30% of teaching process or teaching activity delivered by online. But of course, not all lecturers are happy with this recommendation, yeah, including me. I mean, I'm not an online person, yeah, as mentioned by Brother Herman. I prefer to see my children in the classroom and deliver the lecture face to face and get direct discussion with the children. But of course, suddenly all courses should be delivered online since pandemic occurred, and it make us to be innovative. You have to be innovative in delivering teaching, and also to develop some online method, online teaching material So the teaching process still can go quite well. Yeah. So maybe there is one question I would like to ask the students here. Yeah. And actually, I already asked this question to maybe almost all students. Yeah, got this question during the classes. Yeah, especially when I teach pharmacotherapy of infectious disease. Anybody can answer the question why bacteria and viruses become resistant?
Okay. Anybody you can speak directly or type in the chat room? Kian maybe want to say something, or Hamdi? Anybody?
Anybody would like to answer my question?
Yeah. This is
Why
Dr.
bacteria and
Saifuddin's
viruses become resistant?
strategy for engagement. So maybe somebody can speak.
Hello.
Yes, please.
Hello, my name is Gihat Antar. I am from the University of Bhakti Kencana.
Yeah, Gihat, please.
Yeah, I don't know if my answer will be correct or not, but from my understanding, when people consume antibacteria, yeah, too much, too often, they become resistant. The bacteria itself become resistant to the body, which make the bacteria and the viruses become resistant. Is it like that's what I know? Thank you.
Pardon, can I add some?
Yeah, Hansen, please.
For what I have learned before, why bacteria become resistant to antibiotics is that because when we don't consume the antibiotics well in a period of time, the bacteria produces the beta-lactamase enzymes. So it breaks down the antibiotics ring so that our antibiotics that we consume isn't effective to kill the bacteria. That's from what I know. Thanks.
Okay, thank you very much. A great answer. Okay.
Can I answer on behalf of Dr. Hannah? Actually, Dr. Hannah already sent it to the chat, but it's a direct chat to me. So she said it's due to the mutation for the virus.
Okay, thank you very much. All answers are correct, yeah. But all answers are coming from our perspective as human being, yeah. I mean, not from bacteria or virus perspective, yeah. If we answer from our perspective as human being, that answer are correct, yeah. But if we answer the question from the bacteria or virus perspective, bacteria and virus become resistant because they want to survive.
So you're representing the bacteria, Dr.
Yeah, because it's very, very important. I mean, I always tell to my students, yeah, that even the very tiny creature, bacteria and virus, they want to survive in their life. Yeah, they got some pressure from us, from human, yeah, because we combat the bacteria, the viruses by antibiotic, by antiviruses, but the bacteria and viruses, they struggle, they try to be survived, yeah. So it means that we should learn from bacteria and viruses that even they are a very tiny creature, a very small creature, but they still have a willingness to survive. Yeah. So we are human with a big physically. Yeah. So we have to also struggle to survive in our life. Yeah. So nothing can make us surrender in our life. Yeah. So it means that this pandemic, of course, brings some negative impact in our life, but we still have to survive, yeah, during this pandemic. Yeah. So this the answer from bacteria and virus perspective, not from human perspective. Yeah. Of course, in our teaching process, I'm sure that all students have been already taught by the lecturer that if we as human use antibiotic or antivirus inappropriately, it will make the bacteria and viruses getting resistant. Okay. So back to my presentation. What is my dispense? Actually, my dispense is an online pharmacy simulation that allows the children to practice dispensing in a safe environment. So it's quite good to be implemented during this pandemic era. And the system is designed to allow users to build knowledge, improve cognitive proficiency, and develop professional values and attitude. Yeah. So actually, because just last year, we started with my dispense, and of course, we are still learning how to use this tool appropriately. And actually, my dispense was originally developed by Monash University Australia in 2010. But since 2013, Monash University has worked with international partners to make my dispense available to other schools of pharmacy. And as far as I know, like some school of pharmacies in the US, they already also implement my dispense for their skill practice. And I'm sure Dr. Hana Morisi know better than me about my dispense because Monash University is in Australia, and both Professor Ball and Dr. Hana Morisi, they have long experiences in teaching in the school of pharmacy in Australia. So in 2010, we in UEE started to use my dispense through Ibu Mutiara Helawati. She contacted my dispense support team in the Monash University, and Alhamdulillah, we got permission to use my dispense in our teaching learning process. So what does my dispense do? My dispense actually simulates a community pharmacy where students are able to practice dispensing of prescription medicine, medicine and provision of advice and counseling. Maybe all students already know that during the teaching process, within the whole curriculum, there are some courses, some subjects related to prescription medicine, counseling, and also drug information. But also, my dispense can be used for simulation on drugs and pharmaceutical supply management. So we are in Indonesia. We know that there are some important areas for pharmacists in primary health centers, in the hospital, in the community pharmacies. Yeah. Which is the first one is managerial aspect or managerial area, and the second one is clinical area. So my dispense can be used for both area of pharmacist profession, which is clinical pharmacy and pharmaceutical managerial. So by using my dispense, students can undertake this great exercise. The lecturer and the team will upload the exercise, the material to the system, to the software. And each exercise is a single patient presenting with either a prescription or self-care requirement. And so as we know that as pharmacists, we have to give provision to the patient, either patient presenting with a prescription or patient coming to the pharmacy for self-care requirement.
So this is like a virtual simulation, Dr. Stephanie?
Yeah, maybe in next slide I will show you this kind of simulation.
Yeah, maybe we're curious how real is it? I mean, if we compare with the reality, you know.
Okay, maybe yeah. I will jump after this slide. This slide is an online simulation. So you can see here in the slide, there is a pharmacy simulation. Sorry, I don't need. So this is a pharmacy environment. I mean, both in community pharmacy or maybe in hospital pharmacy. And this is the simulated patient. And actually we can choose an avatar for the patient according to the age and gender of the patient. Maybe I will show you the avatars that can be used in my dispense. Sorry. I cannot move my slide.
Maybe you can escape.
Sorry. Okay. These are the avatars for the patient. So we can choose any avatar from here according to the age and gender of the patient. So there are some maybe young age patient, some old patient, older patient, and also some kid, kid with their mom or their parents. Yeah. And also we can see here that the avatar quite various. We can find also an avatar with hijab. So because maybe here in Indonesia, lots of Muslim female wearing hijab, so we can choose an avatar by wearing this hijab.
Oh, so this is related with the diversity, like what has been explained.
Yeah, so it is quite interesting actually.
Professor Ball.
Yeah, and I think we can also add some more avatar here, but of course I think we should contact the support team of my dispense in Monash University.
Can we put our own
more
face?
Avatar related to Indonesian people.
Yeah. It's a good idea. We can use our own
representing
face.
Indonesian people. I mean, we of course maybe not only the religion, but because Indonesia is quite diverse, in terms of race, so many tribes in Indonesia with some different physical appearance, so we can maybe suggest my dispense support team to add some more Indonesian avatar.
Yeah, there's a little addition from Bu Mutiara here in the chat.
Okay, please, Bu Mutiara.
She said, or maybe you can explain directly if you want to add something, Bu Mutiara, please.
Bu Mutiara, you would like to add some information about this avatar?
Yes, he said with this application, we may mimic the real activity, although some point cannot be changed by any application or method online.
Yeah.
So at least it can help us to represent the reality.
Yeah, I think we should make a stronger collaboration with Monash University, because as far as I know, there is also like an annual symposium about this software, My Dispense. So maybe next year, maybe this year, Bu Mutiara may be maybe one of us from Pharmacy WE can attend this My Dispense annual symposium. So,
Bu Mutiara, do you want to say something? Okay, it's okay. You can carry on, brother. We still have about
no, I think
3 more minutes. Yeah.
It is enough. Thank you. Please, Bapak Saifuddin will explain.
Yeah, we still have about 5 more minutes.
Okay, so back to my previous slide. So My Dispense has a comprehensive databases, either for patient or prescriber and medication. And until now, there are 43 prescribers. I mean, like in Indonesia, doctor who write the prescription, and also 445 patients. Like you see before, there are a lot of avatar we can use in the application, and also over 800 prescription medication. But maybe we can also add some prescription, yeah, if we are allowed to do so. And in addition to prescription medication, there are also 282 OTC medication. I mean, a medication for self-care requirement. So there are so many material, yeah, supplied by MyDispen in this application. So to get into this MyDispen application or software, we should go, we should log in to this application. And of course, to get an account for this login process, we should contact MyDispen support team in Monash University. And as I told before, Alhamdulillah, Bu Mutiara contacted the support team of MyDispen in Monash University, and the permission has been granted to us here to use this application. And as far as I know, UI is the first university, the first institution in Indonesia got this permission from Monash University. So Alhamdulillah, I'm so proud to Bu Mutiara. And as I said, that in addition to the dispensing process, MyDispen can also be used for inventory control, or drug supply management, because for the pre-pharmacy professional program students, they also learn how to store the drugs, how to purchase the drug, and how to make sure that each drug is stored appropriately. So this is an example of prescription process. So the students will get this exercise. This one is for prescription medication. So the student should answer this question, this assessment. And this is the patient, simulated patient.
Are they all in English, Dr. Saifuri?
Yeah, that's the problem. Yeah, that's the problem. The language used in MyDispen is English.
Yeah,
so
it confirms the statement from Professor Ball in the beginning, right? English is very necessary.
Sure. Yeah, that's correct.
Yeah.
So that's our challenge. Our challenge here in Indonesia, how to improve our English, including for myself. I'm still learning how to make my English better and better.
You're a good English speaker.
Okay, so this one is a more complicated assessment for prescription medication. And again, the students have to solve this problem. This is for finding and solving drug-related problems here. Maybe pharmacy students know that one of our duty as pharmacists is to find and solve drug-related problems. And also this one, another example for finding and solving DRP. Okay, so in the Department of Pharmacy UI, MyDispen is used as pharmacy for skill practice in hospital pharmacy block. I mean, we just started to use this application last semester, but not yet for undergraduate students. Yeah, we use it for the student of pharmacist professional program, but hopefully next year we can use this application for undergraduate students. And the four competencies has been practiced and assessed by using MyDispen. The first one is optimization of drug use, and then drug information services, and then communication skill, and the last one is scientific evidences of pharmaceutical services. And these four competencies is 4 out of 7 competencies assessed during the final pharmacist national exam. And this is our students said about MyDispen, because we distributed questionnaire to the students to get their opinion about MyDispen. And for the question that my dispense is easy to use, the answer from the students, around 65% students, they said they agree that my dispense is easy to use. Yeah, it means that the software is not quite complicated to be used by the students. And then the next question, my dispense instruction can be easily understood. The answer, 63% of the students answer agree that my dispense instruction can be understood easily by the student, even that the language used in my dispense is English. And for the question that I like my dispense appearance, I mean, for example, the avatar and also the pharmacy simulation, the student answer that 45% of the students agree that my dispense appearance is quite likable. Yeah. And for the last question, it's quite interesting. Yeah, I prefer to use my dispense for skill practice, not until half of the students. Yeah, I mean only around 40% of the students answer agree. Yeah, to use my dispense over other method for skill practice. Yeah, that I'm sure that the reason behind this, as you mentioned, because my dispense use English as what's it? The
Medium of
language,
instruction.
for instruction, and it's still quite difficult for the students to comprehend all instruction, and maybe still taking time for them to solve the problem, because during the assessment they should consider time constraint for each, for example, for each exercise or each assessment, we provide 10 minutes, to solve the problem, and because they should think so long about what the question is and what the instruction is, so they took a longer time to solve the problem. Even if maybe they can solve the problem, but they took a longer time, than we provide. Okay, so maybe there are still some challenges for the lecturer, for the team to develop a better my dispense for the skill practice in our department. So I jump to my conclusion for my presentation. Online teaching learning is a big challenge, as mentioned by Professor Bol, Dr. Hannah Morisi, and also Professor Taher. It's not easy, even for me, because I'm not an online person. It's quite challenging. It's really hard for me to change my lifestyle from offline to online. And then, of course, continuous innovation and improvement is a must. So we in our department always try to find out some innovative model for teaching material. And I'm sure that we always encourage all lecturers to develop online teaching material to make online teaching process better and better. And of course, we hope that the pandemic will be over very soon. Okay. So thank you very much to Ibu Mutiara Herawati and team for letting me presenting my dispense. Even I didn't involve quite much in this project. And also thanks to IPTC organizing committee for making this event amazingly held, because I know you had only maybe 3 or 4 weeks to organize this event. But Alhamdulillah, with your great effort and also support from Professor Bol, Dr. Hamorisi, Professor Taher, this great event can be organized very well. So thank you very much, and hope to have you both again, Professor Bol and Dr. Hannah Morisi, in Yogyakarta very soon after the pandemic is over. Thank you very much, Dr. My brother Herman.
Thank you.
I hand over.
When is this picture taken?
You can see the time. It's 2019.
September 2019. I see.
And,
okay.
It's 2 years ago. Yeah. Okay. Not 2 years. Yeah. 1 year ago. 1 year ago. Okay. And hopefully this year, once the pandemic over, Professor Bol and Dr. Hannah Morisi will come over here again to Jakarta and see us directly, physically, and already help
And
them.
we have some of you here too.
Yeah.
2020 never happened.
Finger crossed.
Okay, thank you very much,
Okay.
Brother Herman.
Thank you, Dr. Saifuddin, for the great presentation. And previously we had Professor Tahir, Dr. Hannah, and also Professor Patrick. Okay, and thank you to all of the amazing participants. They still survive. We still have about 146 participants. So in general, actually, there are 3 questions from the participants. The first one is regarding about how the gap between the technology and the real practice that they need to have as a future researcher or maybe a practitioner. For example, regarding with the lab skills, for example, that's the first question. And then the second one is regarding about their whether the students are confident they have enough skills or not. We don't know when the pandemic will end. So the question regarding about the proficiency and also their confidence as whether they have acquired enough skills or not, because technology is important, it helps a lot, but it cannot be the only solution. And actually, Dr. Hannah already volunteered to answer one question regarding the lab thing. Dr. Hannah, please.
Yeah, I would like to share with everyone. Can we stop the screen from Saifuddin?
Yes.
Okay, sorry.
Dr. Saifuddin, you
You're
can
right.
Sorry, Hannah.
stop the screen sharing. Yeah.
Okay. So I will show you how we tried to kind of control. Can you see my screen now?
Yeah, it's starting.
Yeah. So what we did, I've got a lot of feedback. What we did is we recorded some sessions live, and actually got the students to. I don't know if I can play it. You guys in the way, but
You can
this
click
is how
the one in the center, the play button in the center.
Try pushing the play button.
Yeah. Yes. It's just pictures were in front of me.
And as soon as the end is exposed, screw
Don't know if you can
firmly
hear the sound.
on.
Yeah, it's
Rubbish to the
audible.
side. You can then invert your vial. Sometimes it helps to hook your little finger underneath the end of the chemo pin just to secure it, and you can then withdraw the correct amount of active ingredient that's going into our infusion bag.
So this is just an example of how we done lab classes where we done the video ourselves, the students see it, and then they reflect on it and still write lab report because we give them the answers. This was a clean room manufacturing.
When that's dry,
it's
and the alcohol's done its job, rubbish.
Yeah. Yeah, you can.
I think another thing that people have to focus on is we have to remind our students that you do not graduate from a pharmacy program as a fully competent, fully skilled professional in every area. It's like when you get your driving license for a car. It doesn't mean you're a perfect driver. It just means you have enough skill to carry on learning for the rest of your life. And if this COVID pandemic means that you come out with 1 or 2 things not covered as well as they should have been, you still have the rest of your life to catch up and acquire whatever skills you missed out on.
Yep.
So you do not graduate as a pharmacist and then stop learning. You carry on learning for the rest of your life anyway.
One of the other things we do as well is avatars, and I used it in Indonesia last time when I came. So you can also have case studies recorded on Avatar, and then so the students actually learn to listen, to take notes. So it's just
I have been experiencing irregularities in my menstrual cycle for the past 2 months, and I am concerned about the cause of this as I am planning to start family soon. I also put on too much weight, which will make pregnancy even harder.
So very important part of our job as a pharmacist is to do a lot of things concurrent, not just listen or just write or just dispense. We need to be able to do all of this at the same time, and also think on our feet and decision make on our feet. So using Avatar to relay the information rather than someone role playing, someone they are used to, make a little bit of difference because the students don't have the rapport with the Avatar as much as they have with me. And on reality of Hannah is 35 years old, female or male, disappear because the Avatar represent the age group, the case, and it's a brand new person. My dispense, the one Sapidin was talking about and Machiara was talking about, it's fantastic, but you will find also that people do other small projects. I developed a game, how to manage stock when I was in CDU, what pharmacists do when they have a cold store parcel, how to order, how to receive the order, how to put things on the shelf. My dispense is fantastic, and I think your students will enjoy it. But you got a good percentage of people travel around the world and become globally active pharmacists, whatever in the Federation of International Pharmacists when they meet, or through migration. So the fact is that they use English. I would love you guys to see it as a positive point rather than a negative point, because sure, it's just to make you globally being able to speak with anyone and everyone. I know our native language is close to our heart, but it's emotional connection and cultural connection rather than scientific progression connection. So I see it as a positive rather than negative.
Sure. Although one of the
Professor Bro, I couldn't hear you.
dispense was created with an enormous grant to underpin it, and it's been you know, those sorts of softwares, whilst extremely valuable, are hugely expensive to develop to that sort of standard. And certainly the way forward to have more of these sorts of facilities and more flexible ones is for us as groups of universities to get together, because we will be able to resource these things much better as groups than as individual colleges.
Sure.
Thank you, Professor Ball and Dr. Marcy. Yeah, technology cannot replace everything. And yeah, again, you cannot master everything, especially as an undergraduate student in the pharmacy program.
Yeah.
Professor Tahir, would you like to add something?
Yes, actually, I agree with Prof. Ball. Actually, when you graduate from pharmacy, means you finish everything. I think, yes, we have to take opportunity to study entire life. Other things that need to be considered is because pharmacy, after they graduate from university, they need to take housemanship and also attachment to a certain, you know, personal place. in order for them to sharpen their knowledge and skill. Yes, they still have time, even though they don't get everything during their time in university, but they still have opportunity to develop their skills outside there. Yes, thank you.
Thank you very much. Yes, please, Dr.
Just one
Marcy.
thing, a last thing. During the pandemic as well, while students didn't have the exposure within the institutions, the teaching institutions, a lot of work experience and jobs become available for students to actually go and hands-on work in hospitals, the community pharmacies, and so on, because they still have better knowledge than the lay person to offer service, especially in vaccination. When we had the trace and tracking process started, you need a lot of people floating around the system. So while they didn't have the institutional exposure, medical students, nursing students, pharmacy students were massively in demand or during the pandemic to actually do other activities, which gives them somehow partial exposure.
Yeah.
Thank you, Dr. Marcy.
Some of our students are doing things like marshalling people at vaccination centres and things like that, because the vaccination programme is rolling very, very fast in the UK. They've just topped 15 million people inoculated. I actually had my first jab this week.
So how do you feel now, Professor Ball, after being vaccinated?
Absolutely fine. I had the AstraZeneca.
The bliss of the age. I don't have mine yet.
Hannah's still in the queue because she's younger than me.
I was listed in the last priority, maybe.
Dr. Saifuddin, would you like to add something? Do you plan to have any a kind of future planning, like having like Indonesian version of My Dispense, like what is questions by Bu Okti?
Thank you very much, Brother Herman, also for the question. Of course, we hope we here in Indonesia we can develop maybe the same application for skill practice simulation. Yeah, because I'm sure it's not quite difficult to develop that kind of software or maybe application, but still we need to find out the way how to build, how to develop, and the most important thing is how to begin. Yeah, how to start creating this kind of application. Yeah? I mean, for example, we here in UII we have lots of human resources with great skill, yeah, with very good skill in IT. And I'm sure if we try hard enough and seriously, we can develop that kind of application, because in this online era, I think it's quite easy for us to find lots of support, yeah, to build this application. So thank you very much, Bu Okti. Good luck with your PhD in Taiwan. And I hope once you come back to Indonesia, to UII, you will be able to support us, yeah, to build this kind of application, because this application is quite related to your field. Yeah, I mean, to your research area and to your lecture area. Yeah. And I found there is another question from Gihad about this application, as mentioned by Professor Bowles. Even this application, the software was built firstly maybe around 10 years ago, but I'm sure that this application always be updated by the support team. So there is no problem. We can always follow the newest version of this application, and hopefully, if we can collaborate more strongly with the Monash University, we can also maybe, as you stated, we can make some more Indonesian avatar and some more Indonesian related prescription medication and some real pharmaceutical problem here in Indonesia. And with the support from Professor Bol and Dr. Hana Modisi, maybe you can help us to get connected with some more Australian pharmacists to build this kind of medication. Thank you very much, Brother Herman.
Okay. Thank you, Dr. Saifuddin. I think we almost come to the end of our session. I'm going to give like the chance to give like a kind of a closing remark to motivate the students, how to stay motivated and to stay optimistic about the future as the students, pharmacy, future pharmacists and also researcher. Please, maybe from Professor Taher and then Professor Ball, and then Dr. Mercy, and the last Dr. Saifuddin.
Okay, thank you very much, Mr. Roman. Actually, the world keep changing, yeah? So you have to adapt with new condition, new situation, especially what happened to us right now. So we have to be resilient, to be to adapt, yeah, and develop ourselves, yeah, with the condition whenever is it, and the future is depend on us, yeah. So we have to develop ourselves, yeah? Let's say the time being we lack of experience in university to develop ourselves in terms of our skill, community, and also clinical practice. We can also, but we can gain it later, yeah. Finish already normal. Yeah. Thank you very much.
Thank you, Professor Taher. Long life learning, right? Okay, Professor Ball, please.
Very similar comments, really. As I said, we have to see the university training as a basic license to start our practice. The other thing I would have to say, looking over my career as a pharmacist, looking at what pharmacists were doing when I first graduated compared to the skills and knowledge we need now, it is inevitable that people will continue to acquire new skills. So anything that they have missed as a result of lost opportunities over the last couple of years, I do not believe it will compromise people significantly. The human spirit is remarkably resilient, and with God's support, one way or another, we will continue to progress.
And I think from me, I agree with both Professor Taher and Patrick, but I would like the students not to look at themselves as being victimized or victims. It is a two-way contract. While we are having hard time to sleep at night because we want to give them the best that we can do, it is also they need to meet us halfway through. It is their responsibility to make sure that they get out of us and out of the big world what they believe we didn't offer them. It is their responsibility to remediate when the world open, to go and find jobs, to make up for what they lost, jobs in pharmacies, even unpaid.
Yeah.
They need to make sure that they meet the institution halfway through, because I can tell you guys, the students in the background, we doing our very best. We're not denying anything, but it is a global pandemic.
Yeah.
We have to provide what we can do. And you need to keep in mind, I'm not a victim. I will survive. I will be the best I can be. I will be the best pharmacist I can be. All what I need is to remediate. ask questions, and find ways where I can learn and survive and become as good as the people who were attending face to face. Shaking hand and smiling directly in your face doesn't teach you to be a good pharmacist. Your learning, your practice, your intention, and your ability to make yourself is what will make you a good pharmacist, not the fact you're attending an institution or not. So I will wish you all best of luck.
Thank you, Dr. Marcy. Lastly, last but not least, Dr. Saifuddin, please.
Thank you very much, Brother Herman. Maybe I just would like to add some word from Professor Tahir, Professor Patrick Ball, and Dr. Namorisi. As I mentioned before, that even from the bacteria, from the virus, we should learn, that even they are a very tiny creature. The virus is the smallest maybe creature in this world, but they always try to survive in their life. So when we get some pressure, we maybe we face lots of problems, but it should make us to get stronger and stronger, to make us better and better. So pandemic is maybe like we can deny this pandemic, but still we have to survive during this pandemic, and always pray hopefully this pandemic will be over soon. Thank you very much, Brother Herman, and also all students or participants for this APTC. Thank you very much.
Thank you very much. We would like to appreciate all of our speakers today. You can show yourself and maybe a virtual and also a real applause, from your screen. Thank you, Professor Ball, Professor Taher, Dr. Marcy, and Dr. Saifuddin for the great and inspiring sharing. And we would like to give a virtual certificate to award you the virtual certificate. Yeah, I think Miss Nurlaili is showing the certificate now. Okay, the first one is to Professor Patrick Ball. Let's give applause to Professor Ball. And then to Dr. Hannah Morrissey. Thank you very much. To Dr. Saifuddin, PhD, and to Professor Muhammad Taher. Thank you very much for being with us. It's really inspiring. We need to stay optimistic. And I would like to thank you all. We already have the photo session, and I would like to give prizes too to some of our participants. And the first one is we have a prize for Gihad from Bhakti Kencana, if I'm not mistaken. Gihad, are you here with us?
Yes, hello.
Okay, you got door prize. I don't know what it is. Hopefully it's a kind of a new gadget to help you for online learning.
Thank
or maybe
you so much.
access to my dispense, we don't know. Or maybe you get a Canvas account. And then the second one is to Hansen. Okay, to Hansen. Hansen, are you here with us, Hansen?
Hello, I'm here.
Okay. And I still have 3 more prizes, actually. Anybody wants to... Okay, I have a very quick question. You can answer it directly or you can chat it. Okay. Where is Wolverhampton University located? Can anyone answer that questions? Where is it located?
United Kingdom.
Yeah, Miss Nur Fatima, is that you?
Yes, sir.
Okay, you got it. And then where is the location of Professor Taher's campus? Where is it?
Guantanamo.
I should give this question to another person, actually.
Okay.
Okay. What is the name of the application used by IIUM? Hamdia Snow. Yeah, please.
iTa'lim.
iTa'lim. Okay,
Yes,
sir.
correct.
So this prize can be for anybody, right? Doesn't have to be for undergraduate student, right? Okay. And one more. My question is, what is the name of the university that shared the My Dispense to UII?
Monash University.
Monash.
Okay. In my screen, I saw Sofiani Latifa. Okay. Maybe I'm wrong, but Sofiani Latifa, I think, answered that question.
Yeah.
Okay. I got already past the 3 prizes. And as the moderator who knows nothing about pharmacy, I will say that actually I learned a lot from the way you learn, and I think it's really inspiring from UK experience, and then from Malaysia, and also from UIE. And I hope that we all we need to stay optimistic that actually learning is a lifelong process, and we can use any tools. And like what Professor Ball said, that actually it's not enough. You can not get everything in one time. You need to follow the process. Okay, I would like to thank you very much for having me in this opportunity. I would like to give the microphone and the virtual the screen to our MC, the amazing Sophie, our student. Please, Sophie, back to you. Thank you very much, Professor Ball, Professor Taher, Dr. Marcy, and Dr. Saifuddin, and
Welcome.
everyone.
Thank you very much.
My pleasure. Yes, Sophie, back to you,
Okay.
Sophie.
Okay, yeah, thank you very much to Mr. Herman, who has guided a very interesting talk show, yeah?
Yeah, maybe you can help Dr. Saifuddin, because you are a millennial and you use a very nice public speaking style like a millennial, because Dr. Saifuddin is from the colonial time. Just kidding. Yeah, Sophie, thank you.
Okay. Thank you for Mr. Herman. Yeah, wow, it's a very interesting talk show, guys. Okay. Previously, I represented the committee to say thank you very much. Yeah, and the certificate for Mr. Herman as a moderator. Yeah. Okay. Yeah, this is certificate to Mr. Herman Felani. Thank you, Mr. Herman.
Thank you, Sophie.
Yeah, it's okay. Okay. Now is the time to announce the winner of the oral competition and the poster competition. And of course, the lucky ones to get the door prize from this morning during the competition until the talk show just now, yeah. And the lucky ones are the participant who asked question about the topic and the same attracted question. Okay, we inform all the participants that will be not double reward, yeah. So don't confuse because in the professionals of the committee are not double rewards, so that the distribution of prizes can run well. Okay. Okay, before I announce the other door prize winners, I will repeat for the Kahoot winners. Yeah, they are Yasirli, and Vicky, Hadromi, Firda, and Rianita. Okay, please contact the committee for more information. Okay, guys, I will announce all the lucky ones who got the door prize from the previous competition. Yes, there are 10 very interesting questions that were selected by the committee and also the judges. Okay. Okay. Okay, guys. In number 10, there Muhammad Raihan Zulfikar. Yeah. Then at the number 9, there Ilya Marelsha Rohila. Okay. And then number 8, Muhammad Ashar. Number 7, Aliyah Nur Ariza. And number 6, Karina Prima. Okay. And number 5, yeah, Kurnia Sandi. Number 4 is Hamna. And the 3 position is Vina Wulan Yuniar. And the 2 position is Zulfa Nurafifa. And the first is Inesia Rahadila. Okay, congratulations. Furthermore, I will also announce the lucky participant who have asked a question during the talk show, yeah, which was also guide for by Mr. Herman. And earlier for the door prize talk show, Mr. Herman. Yeah, okay, I'm not. Yep. Congratulations for lucky friends. The committee will process prize soon. For the lucky ones I mentioned the name earlier, please personally contact me. State the name and send via WhatsApp on my number. Its function is to make it easier to give the prize. Yeah, I will be send my number in the chat column, or If you have other committee contact person, you can also contact them too. Yeah. Okay. Okay, for the next announcement, it will be delivered by Kian Aviati of the IPTC 2021 committee to Kian Aviati. Time is yours. Okay, come on, Kian. Yeah.
Okay, now we will announce the winner of favorite poster. Before that, I want to explain about the criteria of the winner favorite poster by voting likes on February 10 until 12 to 2021 in poster post comments on Instagram at PIO_UII. Who is that? The committee has gotten that 2 winners of the favorite posters get a total prize $25. Okay, let's just call them Dwi Nursita Lestari and Dea Aulia Ramadhani. Congratulations! Okay, Dea Aulia Ramadhani.
Yes. Two winner, yeah, Kian.
Yeah, 2 winner.
Okay, 2 winner.
And next, Dwi Nursita Lestari.
Okay, congratulations ya for Dwi Nursita Lestari. Wow, amazing guys. Congratulations ya. Thank you for your time to Kiana Fiati. Okay. Next. Why? Oh, no. I'm sorry, it's okay. Next, this is something the competitors have been waiting for. Get ready. The head of the Pharmacy Department, Faculty of Mathematics and Natural Sciences, Universitas Islam Indonesia, will be announced the winner. To Professor Yandi, time is yours.
Okay, well, thank you. Okay, guys, I'd like to announce the winner in this activity or IPTC 2021. The criteria include appeal, title, content, purpose, method analysis, outcome, conclusion, organization structuring or use of visual, and style of preparation. Okay, next slide. The result winner of oral and poster presentation competition. Okay, oral presentation competition. The third winner is Jihad Antar Universitas Bakti Kencana. Okay, Jihad will get $50 as the third winner. Okay, congratulations. Okay, next. The second winner is Zahratun Nafiah, Universitas Islam Indonesia. Congratulations. Zahratun will get $75. And the first winner is Hansen Ngaliman, Universitas Surabaya. Congratulations, Hansen Ngaliman. Yeah, the first winner will get $100. Okay, once again, congratulations. Okay, next. For poster presentation competition, the third winner is in Indonesia, Raya Nurdin, Universitas Islam Indonesia. Congratulations. Inessa will get $50. And then the second winner is Iwang Davistiawan, Universitas Islam Indonesia. Congratulations. Iwang will get $75.
Yes, no, it's
And
just what I applaud.
the first winner is congratulations, Zulfikar Muhammad Aditama, Universitas Ahmad Dahlan. Yeah, congratulations. Okay, Zulfikar will get $100. Okay, one again, congratulations for the winners. Okay.
Okay, thank you, Professor Yandi. Okay, again, congratulations for the winner, yeah. And wow, it's amazing. This is some international Pharma Talk and Competition 2021. Okay. Okay, guys, thank you for your participation. We are waiting for the next participation in IPTC 2022. See you on the next agenda. Wassalamualaikum warahmatullahi wabarakatuh. Thank you, Miss Professor Patrick Paul, Dr. Hannah Morrissey, Professor Muhammad Taher, and Professor Saifuddin. Yeah, thank you.
Thank you very much. See you.
See you.
Thank you.
Thank you very much, Professor Bong. Thank you very much, Hannah.
You're very welcome anytime.
See you very soon, inshallah.
Thank you, Prof. Patrick. Dr.
Thank
Hannah,
you.
Prof. Taher.
Thank you very much.
Yeah.
Nice to see you again.
Inshallah, we really look forward to coming to see you again.
Inshallah, inshallah.
Very dear name, Sophie. It's my mother's name.
Pardon, Hannah?
Very nice name for Sophie. It's my mother's name.
Oh, good.
Version of my mother's name.
Have a very good weekend, guys, and see you.
Okay, see you.
Is it okay to go?
Yeah, you can leave now, Professor Ball and Hannah.
See ya.